Good morning, esteemed guests, faculty members, and all the delegates who are participating in this workshop through online and offline mode. I would like to extend a very warm welcome to all of you on day one of this workshop, third annual workshop on animal handling and experimental pharmacology, organized by Department of Pharmacology, IGIMS Patna, under the Aegis of Academic Society of Pharmacologists of IGMS. This workshop aims at providing you with the opportunity to deepen your understanding and upgrade your skills related to animal handling and experimental pharmacology. First, we have the inaugural lecture on the topic Current and Future Perspectives of Animal Research and Drug Discovery. And this topic will be presented by Dr. U.S. Chakradhar Rao, sir, Department of Medical Oncology, JIPMA, Pondicherry. Over to you, sir.
Okay, I'll do it like that, it's easier. Very good morning to one and all. Thank you for this opportunity to share some of my thoughts and insights during this very small gathering. So it's a small gathering, so it's better that we could be more interactive than simply me talking and simply you sitting and then listening and then leaving. So the topic was given to handle here is the current and future prospects of animal experimentation in drug discovery. So where we stand right now in drug discovery research, animal research. So do we really need animals for drug discovery, or we have come over? We are in an age that we don't even need animals to discover drugs. What's your opinion? Anyone would like to say a few insights or thoughts? Okay, still animals are essential. Okay. Any other thoughts? Okay. I think we do need animals because, in fact, the drugs that what we are going to use finally, they are not going to be used in model systems. They are not going to be used in vitro cells. They are not going to be used on computers. They are not going to be used for some healthy human being. So we need to understand the complexity of the mechanisms of a disease, and the holistic model to understand the complexity of mechanisms is basically a complete in vivo model. So animals are still essential, as Professor on the front line said, that they are a cornerstone for drug discovery research. So the stand is still there, so there is an essential role of animals in drug discovery, but where and how we could change these role of animals is something that is evolving from time to time. And most importantly, regulatory requirements, because unless otherwise you show the safety and toxicity profile as well as the efficacy profile of any molecule, is not going to be accepted for a new drug application. Okay, so IND is something that will come before, and then the new drug application should have definitely these kind of data from these animals, and that is where we stand. So which therapies do we vote to animal research? Any answer? Almost everything what we consume today. Okay, none of the drugs have come to the market without animal research. So we do all the medication that we currently use to the animal research, in fact. So just to highlight, we always hear about penicillin discovery. Okay, we always hear the story of fungus grown, mold grown on a petri dish. But what actually brought that observation into clinic is actually, there was a trial, there was a preclinical study in 1940s which compared actually in mice what happened when they have infected with streptococci, and then they gave this mold, and then they tested, and they proved the efficacy in the preclinical models. And then it has become a groundbreaking molecule, and then antibiotics were discovered from there onwards. So that is a stepping stone of converting the laboratory observations into a direct clinical applications. So they do play a clear role. I think we are in the post-COVID era, I would say, not BC or AC, it's like pre-COVID and post-COVID. So we clearly know that how the animal research has helped to progress the research in COVID times, to understand the disease mechanisms, because we know that in the past, there's a lot of research already being done on coronaviruses, how the pathology happens, like what happens to the lungs, what happens to the liver, what happens to the kidneys, what happens to different organisms, including nervous system, on different coronaviruses. We do have a clear understanding. And that has helped us to manage coronavirus infection, as well as the mRNA vaccines that have been developed also had to undergo this preclinical testing. Without that, we could have not received vaccination. Though unfortunately, we do not have much of the preclinical data on long-term toxicities of vaccines that has been generated during these times, and that is where there is a gap. You see, still, in clinic, we see there is a post-COVID fog, post-COVID symptoms, but we do lack this clear understanding of these mechanisms because the data is not publicly made available until now for the COVID vaccine developmentation. So, one most interesting point here is that we do everything, we owe everything for whatever drug development happened to the animals, but the judicious use of animals and the availability of the data that is being generated from all of these experiments should be made available to everyone so that they can learn from these insights and then make some progress ahead. Can we transfer all these results from animals to humans? Any answers? Yes or no? No. Why? Yes, so there is a differences in different systems. It could be primates or non-primates, rodents, etc. What we could understand here is the basic functional involvement of different pathways, organs, and systems. But maybe, for example, the immunological maturation in humans, it's completely different from the immunological maturation that happens in rodents, and also in zebrafish, for example. Because these days people are also using most of the aquatic models like zebrafish models to study some of the observations on cytotoxicity or even immunological mechanisms they are studying in zebrafish. But there are some differences between the organ systems. We cannot completely translate the findings, but at least we can have some fundamental understanding that can give us some direction how we could conduct our clinical studies when we do the phase 1 and phase 2 studies. So it can definitely shed some light on how we can design our clinical studies. So preclinical studies are essential even to plan your clinical studies. So what are different types of animals that we currently use in our preclinical research? So if you see here, I think most of the part is driven by rats and mice. They comprise almost 70% of the preclinical research, and coming to the primates as well as rabbits and guinea pigs, and more nowadays we are using amphibians as well as fish in our research work because it's very feasible. It's feasible to use them, and because it doesn't require much of the infrastructure as well as the ethical regulations to use fish and amphibians. For example, zebrafish up to 5 days of its lifetime, the earlier 5 days, you don't need ethical committee approval to use them for your research work, actually. I don't know what are the regulations currently. I'm not the person from the domain. Maybe some other speakers will shed some light on that. So, as Professor said, do you think that animals are irreplaceable? Are they replaceable? Because anyway, we are going to use drugs on humans. Why can't we directly use them on humans? And then learn from it and then try to use them for humans again. I think that leads back to your also the Declaration of Helsinki on the safety, and you should not use something on humans unless otherwise it is proven. Even though if it's not efficacious, that's okay, but at least it should not be problematic. The safety profile has to be definitely established before the molecule should bring it into the human trials. And also the subjective phenotypes. I think that is most interesting thing that we cannot even study in different other models. For example, in vitro cell culture models and in simulations. So behavior is something that is a complex phenotype. Okay, so all this anxiety behavior, depression behavior, social anxiety, different kinds of behavior is something that we can, for example, ethanol withdrawal syndrome. That is something that you can't imagine studying in a cell culture model. So you need a complex, it's a complex phenotype. So we need definitely an animal to exhibit this phenotype. And social interactions, that is something that is, I mean, people have been done. I think there is one lady, I forgot her name, from South Africa. She studied Gibbs for ages, and she has developed some great insights after observing these chimpanzees throughout her lifetime. And then that has led to more of our understanding of the human behavior also. So the social interaction also something is a complex phenotype that we cannot study in other models that we currently have. But what we could do is to refine the usage, okay? And to, you also know about the 3R principles, right? What are the 3 R's? Which is reduce, refine, and replacement, okay? So first thing is reduce. How we could achieve the reduction in the usage? For example, you are doing an anti-inflammatory study in your lab with using some phytochemical. I think this is most common in our setup, that we study a lot of phytochemicals. Maybe some other person in somewhere else in Madhya Pradesh also is doing some same experiment without knowing that you are already doing that. So basically you are basically doing the same thing but in different places. So you're using more animals. So you need to have a registration mechanism. So that adds 1 R. We currently have 3 R's. So you can have a registration mechanism. So before you start your animal experimentation, you put your protocol there, you make it available in the public domain. So people before they start another experiment in the similar domain, they can look through it, the registered protocols, and see, okay, if somebody's already doing it, just connect with them and then maybe discuss and then try to, develop some collaborative work rather than repeating the same thing. So that's how you could reduce, in fact, by bringing in a registration mechanism. And then what about refinement? So what is about refinement? Okay, so the refinement procedures includes the best usage of the animals. And the best usage of the animals is also, okay, registration mechanism is okay. What about the responsibility of the user, as well as the committee and the members, from the committee to the end user? So everybody has to be responsible in implementing the appropriate guidelines when we currently use animals for experimentation. So that accountability is something that should come from the committee to the end user who is doing the animal experimentation. And then how we can replace the animal models. We do have some other systems that we can use, but before going to other systems, we can even replace by making the data available in the public domain. That comes from your research. Okay, you have a repository where all animal research output is being put there. So people who would like to plan an experiment with 6 animals, maybe they don't need to do 6 animals, maybe they need 12 animals. So they can take this data from already existing public domain, they can add 2 or 3 animals from their setup and then add this data and then they analyze combinedly. So this fair data sharing is something that is very essential. But before that, the robustness should be there, how the data has come. So the people who are sitting here would like to trust the data that is coming from Madhya Pradesh or Andhra Pradesh or from Pondicherry or from Tamil Nadu. How to trust that data? So that robustness of the data should be there. That has to be generated from the end user, from the committees to the end user. Everybody should oversee. So the responsibility and robustness, that is the integrity with which we generate this data. That means it's more accurate, more well designed. That means the committee should first see if the study is well designed, whether it's going to add something new to the scientific domain. If it's not adding something new, there is no point in doing such studies. Okay, so if it is something redundant. So that robustness is something that we need to bring in picture. So now these 3 R's has become 6 R's, along with robustness, registration, and then responsibility. I would put the responsibility on the top of every R, because that is what will help in refining all the other 5 R's. And then in the end, we will judiciously use the animals for scientific benefit. And then all these animal welfare organizations will not trouble you also, not to do animal experimentation. So I think that is what we discussed here. And what about the alternatives? Any ideas about alternatives currently available with us? I still have 6 more minutes. Sorry? In silico computational models, methods, perfectly fine. That is very well taken. Any other models? In vitro cell culture models, perfectly fine. For example, if you are developing an anti-cancer drug, so first you test in the model system. So if you are developing for ALL, you take lymphoblasts, and then you treat the drug with the cells. You treat the cells with the drug, and then you see the viability of the cells. It's a very simple experiment. Any other models that we currently use or can replace animal models? So for simulations, also, we do need some animal data. So that is where that registration and then the fair share data sharing will help, so that the people can use the existing data and then try to model and then simulate. So if you have 6 animals that you are using for one experiment, so in clinical trials, we do have simulations, clinical trial simulations, okay? We create a digital twin, and then we see what happens if we have the same physiological parameters, but what happens to the kinetics as well as the pharmacodynamic parameter that we are measuring in those digital twins. Similar modeling can be done in animal experimentation. So you get the data, you make a digital twin of your animal, and then you can simulate the same data, and then you can reduce the usage of the animals. That's how you can refine, and then you can convince your ethics committee as well as your scientific experts that the experiments that you are planning is more valid and it could add something new to the scientific domain. So as I was mentioning before, the in vitro cell models are something that are unique. We can only gain information on the specific systems, on the specific cell types. But if you want a holistic information on a kind of an organ level information, we do have some organoids like the 3D cultures. So that means your liver has three or four different types of cells. For example, hepatocells, Kupffer cells, and then there is a biliary canaliculi. So if you use cell systems, either you could use hepatocells separately, Kupffer cells separately, binary canaliculi separately. We can't grow them together. So as an organoid, you can grow them, all of them together, so that when you study your drug, you can have a comprehensive information from the organ systems directly from your in vitro experiments, rather than directly moving to the in vivo experiments. So from this organoid cultures or tissue cultures or organ on chips experiments, you can gain some insights that you could use to refine your experimentation in preclinical studies in vivo systems. But they cannot entirely replace the in vivo models, but the observations that you make from these systems has to be validated again in vivo models. But you can reduce the numbers. So I would not go into the details of this experimentation. Basically, this is about studying an organoid culture where some kidney cells were changed in their genotype so that they can develop some cancers automatically when they are grown in culture, so that at the same time the normal kidney cells as well as the cancerous cyst cells you could treat with the drug, so that you could understand what happens to the off-target effects. For example, what happens when you use a chemotherapy molecule, whether it will only kill the cancer cells or it can also kill the normal cells. And that is also essential in our case when we have the technology of developing targeted therapies, because targeted therapies are oriented towards only specific target, and the target could be expressed on different organ systems and different cellular, and these kind of organoid systems can help us to generate some data that we can use for drug development. And these are the different technologies. For example, CRISPR-Cas9 gene editing is something that has evolved the animal research domain to a next level, because in the past when we have to develop genetic models, it was very difficult process to develop a genetic model. But these days, within a fraction of a week, you could have these animal models developed, genetically modified animal models developed and delivered to your doorstep to study your hypothesis or the question. So to conclude, I think animal usage is still relevant. That is where we stand currently. And I think recent COVID experience is something that has also opened the eyes of many of these endorsers of animal experimentation who endorse stopping of animal experimentation. We have learned quite a lot that it's very difficult to understand with the current existing alternatives to animal models to completely replace animals, but we can still refine animals. We should not directly go into animal, but maybe try to use these alternatives before you go into the animal models to have a complete, comprehensive, holistic understanding of the drug mechanism of action and its toxicities. And these are the aspects, the take-home messages that I would think that we should start practicing it. We already discussed this about data sharing, registration, and then having proper design of the experiment, that is robustness of the data that has to be overseen from the committees to the end user, and most importantly, mandatory training programs. Because in Europe, we are not supposed to touch the animals unless otherwise you have a certification saying that you are well trained to handle animals. Then only you are allowed to go and perform an animal experimentation. So such kind of legislations, even for designing an animal experimentation, you are not supposed to design an animal experimentation unless otherwise you do a certification course of 2 weeks, which is run by animal welfare organizations plus the academic societies and scientific organizations coming together. They form a society called RESOLVE, which is like European level animal experimentation training, FELASA. You can Google it when you have time. And they will give 2 levels of certifications, and those users, those people with those certifications only are supposed to operate on animals. So it's like continuing medical education. So the programs like these workshops are something which kind of, alternatives to those kind of certification programs. At least you should learn how to handle animals more appropriately, judiciously, so that you could do justice of using all these animals for your experiments. Thank you very much, and I'm happy to take if you have any questions. Any questions? Okay, how many of you would like to go for alternative to animal experimentation before you start animal experimentation? No one? Okay, we have only 3 out of 20 people or 30 people, odd people we are here. Why? What is the limitation of not going to alternatives? There must be some reason why you don't want to go for alternative, right? There must be some limitation. Most trained expertise and infrastructure maybe to do, but maybe the simulations and in silico thing is something that you could always attempt at, because that is something that does not require much of the infrastructure except some good working computer and internet connection where you can use some of the tools already available in the public domain. Okay, but that's a point well taken. I think we also need to, my suggestion to the organizers is maybe to add upon to the animal handling workshop to an alternative, how to handle these alternative methods, what they require. Some insights they could try in these people who come here so that they can go back and then maybe they start exploring in their own ways how to merge these 2 things together. Yeah. Thank you. Thank you for patient listening.
Thank you, sir, for sharing your valuable knowledge and insights on the topic. Now, I would like to request Professor Dr. Harihar Dikshit, sir, Principal, NSMCH Beta, and President of ASPIGIMS, to kindly felicitate Dr. U.S. Chakradhar Rao, sir. Thank you, sir. Moving on to the next session for today, which is on the topic Committee for Control and Supervision of Expert.
Start.
Okay. We now begin with the inauguration ceremony of third annual workshop on animal handling and experimental pharmacology. It's my honor to invite all the dignitaries on the dais. Professor Dr. Om Kumar Sir, Dean IGIMS Patna. Dr. Basla, kindly present a bouquet to Professor Dr. Om Kumar Sir. Dr. Ruhi, kindly present bouquet to Professor Dr. Harihar Dikshit, sir. Dr. Bhavna will now present bouquet to Professor Dr. Vikas Medhi, sir. Ms. Suchitra, kindly present bouquet to Professor Dr. Lalit Mohan, sir. And Akanksha, kindly present bouquet to Professor Dr. Manish Kumar, sir. Okay. Basla, kindly present bouquet to Dr. U.S. Chakradhar Rao, sir. Dr. Bhavna, kindly present bouquet to Dr. Sunil, sir, AIMS Patna. Dr. Basla, kindly present bouquet to Dr. Nitish, sir, Naipur, Hajipur. I would now like to request Professor Dr. Harihar Dikshit, sir, Principal NSMCH Beta and President ASPIGIMS, Professor Emeritus of Department of Pharmacology, IGIMS, to kindly come on dais for the welcome address. Over to you, sir.
Thank you. At the outset, I want to give thanks to the organizing committee for giving me this opportunity to give a welcome address. Respected Dean, today's Director, Dr. Professor Om Kumarji, Professor Vikas Medhi, PGI Chandigarh, a lion figure of pharmacology, Dr. Lalit Mohan, Professor and HOD, Department of Pharmacology, Dr. Manish, he is the organizing secretary also, and he is a professor of pharmacology now. He got PhD also. Dr. Chakradhar Rao ji, Dr. Hitesh, Dr. Nitesh, Dr. Sunil, and all the participants, colleagues, scientists, PGA students, my juniors. A very warm and cordial welcome to all in this hall. It's a moment of great pride and joy for me to stand before you as the President of Academic Society of Pharmacologists of IGMS on the occasion of our third annual workshop on animal handling and experimental pharmacology. This event represents not just an academic gathering, but a celebration of scientific curiosity, ethical responsibility, and collaborative learning. Animal experimentation had always been a cornerstone of biomedical science, which was in the morning, just before this session, well emphasized by Dr. Chakradhar Rao. From understanding fundamental mechanism of disease to evaluating the safety and efficacy of new drugs, animal models have guided generation of pharmacologists. Yet, with the progress of science, we are also witnessing a paradigm shift towards replacement, reduction, refinement, rehabilitation, and reuse of animals, embracing both technological innovation and human ethics, well explained by Dr. Chakradhar Rao in the morning session. So this workshop, we aim to provide young pharmacologists with hands-on skill in animal handling, understanding of experimental design, and above all, a sense of ethical consciousness in conducting biomedical research. I just, before this inaugural session, I have a talk with Dr. Vikas Mehdi and discuss about the idea of alternate modalities to be organize a workshop on alternative methods also, as suggested by Dr. Chakradhar Rao. I take this opportunity to express my heartfelt gratitude to our Dean, who is also today's director of this institute. I also express my heartfelt gratitude to the director in absentia, because he is busy in other works also. Dr. Om, whose consistent support and vision keep motivating IGIMS to achieve higher academic standards, and I also want to give extend my deepest appreciation and warm welcome to Professor Vikas Mehdi ji. I also wish to thank Dr. Lalit Mohan, who is carrying forward the great legacy of pharmacology and working hard for keeping the flag of Academic Society of IGMS higher and widespread. Why I have chosen widespread? Because we are going to organize a national conference in 2026 at BHU of ASPIGIMS, and he has already collaborated with KMC Manipal, BHU, RAIMS, IGMS itself, and Netaji Subhas Medical College and Hospital. So that's why I have chosen the word widespread. Now the sapling of ASPIGIMS is growing, and very soon it will take shape of butt bricks, which was my vision. ASPI के फॉर्मेशन के पीछे मेरा यही विजन था, ये एक बट वृक्ष बने, ज्ञान वृक्ष बने, जैसे बोध गया में है। और ये मैं बहुत खुश हूँ और अपने आप को गर्वान्वित महसूस करता हूँ कि ये आने वाले दिनों में बट वृक्ष के रूप में ले लेगा और उसका ग्रोथ निरंतर होता रहेगा। I also wish to appreciate Dr. Manish for your hard works done to accomplish my vision of Central Animal House of excellence with breeding facilities. I must congratulate you for getting promotion on the post of Professor Pharmacology, and also for getting PhD degree. I was criticized by my friends and colleagues for the conception of animal house initially, but now they have realized its importance. मैंने कहा था उन लोगों को, हंसले घर बसता है। जिस समय मैंने कंसेप्ट लिया था कि एनिमल हाउस बनाना है, एक छोटे से आउट हाउस में बनाना शुरू किया था, उसमें रखना शुरू किया, उस समय जो लोग क्रिटिसाइज, क्या या एसी में जो फैकल्टी को एसी मिल नहीं रहा है, आप रैबिट को और चूहे को एसी में रखोगे, बड़ी मजाक उड़ा। मैंने कहा था ठीक है, कोई बात नहीं। आई हैव फेस्ड मैनी चैलेंजेस, मैनी एलिगेशंस, लेकिन मैंने कहा था उस समय भी, हंसले घर बसता है। गांव की कहावत है, हंसले घर बसता है, जिसको आप ज़्यादा हंसी कीजिएगा वो बसेगा। आज आई एम प्राउड ऑफ दैट। वेरी वेल इक्विप्ड Central Animal House is here, and one of the best in Bihar also. I think I am not comparing with other animal houses all over India. I also wish to our faculty members of the Department of Pharmacology, the organizing committee, and all the participants for their dedication in making this event a success. I must appreciate the presence of Professor Hitesh. I think Shukalyan is not here. Anyway, all of you in the Department of Pharmacology, your enthusiasm reflects the true spirit of ASPIGMS, all the life members, advancing science with purpose, integrity, and growth in medical studies. As we begin this workshop, let us remember that the essence of experimental pharmacology lies not just in procedures, but in principles, not just in data, but in discipline, and not just in outcomes, but in our responsibility toward both science and society. Very important, society. With these thoughts, I once again welcome our chief guest, Professor Om Kumar ji, guest of honor, Professor Vikas Medhi ji, and all attendees to this meaningful academic event. May this workshop spark new ideas, strengthen our ethical foundation, and inspire every participant to pursue excellence in research. Thank you for patience hearing. Thank you.
Thank you, sir, for your invaluable words of knowledge and wisdom. Now I would like to request Professor Dr. Vikas Medhi, sir, Professor, Department of Pharmacology, PGIMER, Chandigarh, to kindly come and say a few words.
So, good morning everyone, and respected Dean, and Dr. Om Kumar, and Dr. Deskit. Congratulations to become a principal, and Dr. Lalit, and Dr. Manish, and all the faculty members, So I'll just begin that how should we think, So if you look at toward the eastern part, there is a single GLP lab that's in Kolkata. That's a Chris Biotech. Northeast, there is no GLP lab. But we are very proud that when I actually went to Japan to represent India in just before the COVID Feb, and I could see that China was running after OECD people to get the recognition. So during that time, we become a vice chairman for a OECD, and now we are the chairman of OECD, and that's a proud moment. And today we have almost 58 labs, which is GLP approved lab. So if you go back a couple of years back, before 2011, our lab used to go to Belgium, Denmark, Sweden, get an approval, so that our study can be submitted to US FDA or EMA or Japan. But today we get a lot of studies from China and other countries. They do the studies here, and later they submit to US, EMA, or Japan. So one more, important point is, I just wanted to make, I thank Dr. Dixit to building up, animal house. Because, Punjab, I'm taking an exam for last 10 years. And I told Dr. Y.K. Gupta, I'll take you to a couple of places. So I took him to 3 places. And when you interact with the MD student during the exam, you ask a simple question: can you tell me the difference between mice and rat? Okay? Not able to answer. So do you think I can blame to the MD student? Answer is no, because they don't have exposure. A lot of people, they believe that animals use are not allowed. But that is not true. You have to create an infrastructure as per our guideline. So if you go for a national guideline, CPC-SCA, now name has been changed, CC-SCA, or global guideline, if you look at that ARRIVE guideline, accordingly you have to make it with OECD principle. Like Ayush Ministry, they requested me to build up their GLP lab. So I have been helping them. I call all the Ayush Center to Chennai, took lecture from morning to evening. Now, one of the lab has been identified is in the Gwalior. So I went there, I look at all the facilities, I'm helping them to develop. Today, any Ayush product, I'm also helping them for a preclinical study guideline. So once you do as per the regulatory guideline, if you generate the data, all the toxicity data, it will be accepted globally. And that is why it is very important that our students should be trained as per the GLP, as per the OECD GLP, you know, and then only it will be easier that once they join the industry, or if the institute decided to go for a product development, it will be easier for that. And that is what the motto is, that, you know, today you see that when you talk about animal experimentation, I'll tell you another story. ICMR has created a big facilities, Genome Valley, okay, to cater the whole country for all transgenic animal, from smaller animal to higher animal. So when I recall the first meeting, it was, the money was given from PMM House. So there are two people from New York and, you know, Europe. They said, why don't you give this money to the NGO? And that day I said that I think India got an independence in 15 August 1947. Who are you to direct us, our money to debit to another NGO? And that's how this facility has been created, and now it is also GLP accredited. So all the medical colleges, when we look at that, we need to create an infrastructure. Obviously, there is some pressure is going on that we don't want an animal experimentation. You can directly go to the human. So we have a concept of microdosing, or you have a concept of phase 0 study. After COVID also, we created a human guideline, healthy human model guideline. But still today, we do not have an alternative. So we have to go for a conventional one, like we have an NDCT rule. Some of the method has been adopted if you go for an amendment of the NDCT rule in 2023. But our students need to be trained. So I request all the students who are doing MD, please visit a GLP lab. You know, we have 58 plus GLP lab, at least one lab, at least one GLP with drug testing lab. You visit that, you know, one NABL accredited lab, a GMP facilities. You'll be very clear after doing a MD that how we start from, you know, Sarhaj says next year, it will be an alternative. So there is a society of alternative to animal experimentation. And that should be also trained. Like you have been doing in silico study during COVID. During that time, we published 78 papers with computational. We developed our software because we brought a software and they did not extend for 3 months. Then we decided to go for developing our own software. And right now we are giving it free to everybody. Anybody can use it. So then in silico, in vitro, so next time, because I was little encouraged last time when I came, you know, I was taking lecture till 6:30. I thought that after 5 o'clock everybody will leave. But they were there here for till 6:30. Then I got repeated call, can you come here and take a session for animal experimentation? And that's a kind of encouragement. Today if you say that lack of facilities, people are not having an expertise, we started our own program from PGI, that is NBRL training program. How do extract that, you know, animal behavior to do a human? And that is program. So all of you are invited for that program. So we do it 6 monthly, all the animal experimentation program. So we have another program for animal experimentation, you know, NACERA, you know, National Workshop on Animal Experimentation and Alternative. So you can join us, and that I wanted to make it because if you stop today, another 10 years, there'll be nobody on expertise on animal experimentation. And what you're going to do will depend on Europe and US. And that is what people wanted to make it, you know. So that is why you have to be trained. I took that initiative. So we are going to train that people on animal experimentation, on GLP, on, you know, all these alternative also. So that we have to do it. So thank you very much for inviting me, and I'm going to contribute. Thank you.
Thank you, sir. Now I take the privilege of calling on DICE Professor Dr. Om Kumar, sir, Dean IGIMS Patna, to kindly come and say a few words. The lighting of lamp is symbolic of dispelling the darkness of ignorance and illuminating the light of knowledge and wisdom. It's my honor to request all the dignitaries present on stage to kindly perform the lamp lighting ceremony, marking the official commencement of our deliberations.
Very good morning, all of you. Respected Professor Harihar Dikshit, ex-HOD Pharmacology, IGIMS, and presently the principal of Netaji Subhash Chandra Bose Hospital, Behta Medical College. Very respected Dr. Vikas Medhi. I must tell you that as a dean, I am participating in faculty selection and faculty promotion for 2 or 3 years. So I am witnessing experts from all the subjects, starting from the broad specialty to super specialty. And I have found none as superior as Dr. Vikas Medhi as an expert. So much so that I was forced, when he was asking questions to the candidates, I was forced to take a note on my notebook. So I have noted almost 100 questions during that interview. And we are all fortunate that he has agreed to come to IGIMS. So this is a great opportunity to extract maximum from a person like Vikas Medhi. I'm very thankful to Dr. Lalit, who is very keen to continuing the good work which has been started by Dr. Professor Harihar Dikshit. I was listening to him that he has just started the animal house. Then he has started this association of pharmacologists of IGIMS, Academic Association of Pharmacologists. So beginning may be very humble, but if you are trying and you are trying to grow that society or that very small things, ultimately one day it will grow in a situation which, as Dr. Medhi was saying, that we are having a very good animal house, and this association is also guiding. And this is very important to organize such type of meetings, because it is not optional for a teaching institute like IGIMS, and neither it is a tick box requirement for promotion or selection of faculty members. It is an ethical mandate of any teaching institute that such type of activity, CME, workshop should be done. So at the outset, from the core of my heart, on behalf of my regular director, Professor Binde Kumar, I welcome you all and congratulate you all for this, organizing this very good workshop. When we talk about experimental pharmacology, we are talking about very foundation of modern therapeutics. Every breakthrough drug, every novel molecule that saves our life today, from antibiotics to anti-cancer drugs, has its origin in the meticulous, ethical, and humane use of laboratory animals. Understanding pharmacodynamics, toxicology, and mechanism of disease begins here, in these controlled and responsible environments where curiosity meets the compassion. I was seeing the program of this workshop, and the inclusion of panel discussion such as From Bench to Bedside Without Animals, Myth or Reality, is both timely and visionary. As the world moves towards refinement, reduction, and replacement, the 3R principles, we must balance innovation and responsibility. Technologies like in silico modeling and organ-on-chip systems are exciting, but the biological complexity of living organism still holds irreversible lessons from traditional medicine. Workshops like this prepare our young scientists to bridge the ethical and scientific gap. IGIMS has always believed that excellence in medical education is not just about acquiring knowledge, but about cultivating wisdom that respects life, values evidence, and upholds the ethics. I am proud to see our students actively participating in hands-on demonstration. This was very heartening that all the resource person, even students, senior residents, are participating in this workshop. To the students and young researcher here, remember the hallmark of good scientist is not how much he discovers, but how responsibly he discovers it. May this workshop ignite you a lifelong curiosity for science and deep respect for all living beings that contribute to it. I am sorry for preponing your program because Honorable Governor is visiting our campus for his treatment, so I have to be there. So I am again giving my apology to the organizers for change in the schedule. And let me conclude by congratulating Professor Harihar Dikshit, Professor Manish Kumar, Professor Lalit, Professor Hitesh Mishra, and the entire organizing team for their tireless effort in making this academic event a success. I am also thankful to the resource faculty who has come from Pondicherry, Dr. Rao and Chakradhar Rao and Vinod Nair, and from AIMS also. So thank you once again for giving me opportunity to be here. Thank you very much.
I now request Professor Dr. Hitesh Mishra sir to kindly come on the stage also for book release. I now request all our distinguished guests to kindly release our workshop book, Preparatory Manual on Animal Handling and Experimental Pharmacology, making a significant milestone in our efforts in the field of animal handling and experimental pharmacology. Thank you, everyone. I now request Professor Dr. Lalit Mohan sir to kindly come on the dais for vote of thanks.
Distinguished guests, esteemed faculty, dear participants, and fellow organization, I would like to extend my heartfelt gratitude to each one of you for making third annual workshop on animal handling and experimental pharmacology a grand success today. I am delighted to share my DM trainee student, Dr. Palvi, and Dr. Vajla, PG student, Dr. Bhavna Anil Barma, to got grant from DHR and MRU. Not only faculty are getting grant from different funding agency, also my department PG student is also getting grant from different funding agency. Sir, it is due to motivation or due to PG orientation program, and many of the workshops related to conducted for research methodology. That is a motivation from our director and dean. Professor Dr. Om Kumar, Dean IGIMS Patna, has always supported to academic activity and backbone for promotion of each and every faculty of the IGMS Patna. My two of the colleagues also promoted in this year, Professor Manish and Dr. Sukalyan Saha. We thank to Professor Dr. N.R. Biswas, Vice Chancellor, Balaji University. He may be present on online later on, and former Director IGIMS Patna, and Professor Dr. Harihar Dikshit, previous HOD IGMS Patna, Principal NSMCH Beta, for their vision and encouragement, which have enabled our department to run various courses successfully, including allied health care courses, MBBS, MD, DM, and PhD. So we have all the courses that is related to pharmacology in our department. We appreciate the administrative support for encouragement from Professor Dr. Vibhuti Prasanna Sinha, Deputy Director Administration and Chief RU. Today we had privilege to having distinguished resource faculty, eminent pharmacologist, Professor Dr. Vikas Mehdi with me. From PGI Chandigarh, Dr. V.S. Chakradhar Rao, from JIPMA, Dr. Vinod Nair, Director Precision Medicine, Oncology, JS Associate, Gurgaon, both of them friends from my PG time. And it's glad to share Dr. Pramod Manji, HOD Pharmacology, AIMS Patna has worked before at IGMS Patna. We appreciate your valuable contribution. The organizing secretary, Professor Dr. Manish Kumar, co-organizing secretary, Dr. Pankaj Kumar, and the entire organizing team, including faculty members from the Department of Pharmacology, IGIMS Patna suggest Professor Dr. Hitesh Mishra, Dr. Kalyan Saharai, Dr. Adil Ali Sakur, and veterinary doctor Dr. Manoj Kumar. We have started breeding facility in animal house now, as well as junior resident, senior resident, supporting staff for your time, for your tireless efforts and appreciated. A special thanks to Dr. Sajid Hamid for constant work on the releasing of book, for publishing of and releasing of book of the souvenir, and Nidhi for anchoring of this event. We thank MyCalPharm software for providing a platform to demonstrate complex experimental procedure. We also acknowledge the financial support from NSHM Beta, and future I also wish to get support from NSHM Beta. IT Solution Delhi, Gintech Delhi, and AD Instrument Delhi, which helped us conduct this workshop. We appreciate encouragement for all HODs of pharmacology from Bihar and other parts of India, including Tibbi College and Ayurvedic College. We have MU with the Tibbi College and even AIMS Patna, they are using our animals. We extend our thanks to engineering cell officer and staff for their help and assistant to print the electronic media for covering this event. Thanks you to everyone involved in publishing the preparatory manual of animal handling and experimental pharmacology. Let us continue to advance the science of the pharmacology and experimental medicine together. Thank you. Last but not least, to all the participants, your enthusiasm and engagement have made the workshop meaningful. We hope you back valuable knowledge and insight. Thank you to all.
Thank you, all the distinguished guests. Moving on to the next session for today, which is on the topic Committee for Control and Supervision of Experiments on Animals, Rules and Guidelines and Hands-on Training to Fill the Form. B. It will be presented by Dr. Manoj Kumar Sir, Veterinary Officer, Department of Pharmacology, IGIMS Patna. The other team members include Professor Dr. Manish Kumar Sir, Professor Pharmacology, IGIMS Patna, and Dr. Pankaj Prabhakar Sir, Scientist 1, Pharmacology, IGIMS Patna. Over to you, sir.
Good morning, all of you. Good morning, all of you. I am going to deliver talk on the Indian Act, that is CCSEA, Committee for the Control and Supervision of Experiment on Animals. In that, for the research purpose, we need to fill Form B. So we have to hand-on demonstration. I think everybody has got the Form B. Okay. So I will start my talk with Mahatma Gandhi, the father of the nation code. He says the greatness of a nation and its moral progress can be judged by the way the animal treated. So why any act is necessary? Like CCSEA, why it is necessary? Because still the need of animal experimentation is exist. There are so many discoveries based on the animal experimentation itself. So when we are talking about the new discovery, new medicines, any pathophysiological studies on animals, there may be chances the animal may get some kinds of serious injury that will perhaps his life, like persistent injury or some kind of pain. That's why to balance this animal welfare as well as scientific our data, we have to act. In first, the act is Prevention of Cruelty Act, that is PCA Act, that was passed in 1960. This act having 4 chapters, chapter 1, chapter 2, chapter 3 and chapter 4. What we are talking today is CCSEA that comes under the chapter 4. In that chapter, there are 6 sections are there. As we see, from 14 to 20. In that section 15, that describe the CCSEA, that is Committee for Animal and Supervision of Experiment on Animals. Earlier this name was like CPCSA. In 2004 there was amendment because of the act having no prevention word is there. So committee has decided to change in the act, and now we are knowing this as CCSEA. So CCSEA is the apex body to regulate the animal experimentation. In this act, earlier there was no that much clarity and not that much awareness, but after this Menka Gandhi, under chairmanship of Srimati Menka Gandhi in 1998, there was one act added, that is Breeding Act, we will discuss later, and this committee has formulated so many guidelines. So main objectives of this committee is giving the registration, inspection as well as monitoring of this animal house through the IEC, local committee at the level of institution, we will discuss later. So in this section, we will see the registration. By knowing the registration number of any animal house, we can judge whether this organization is private or government. As like see in this, GO means government organization. If written as PO means private organization. Then there are so many types of registration. Like in first, RE means research for education. There is no breeding. If we want to take any animals from any breeders, we have to see in this column. BI is in-house breeding, but no trading. Means he will use there only. Inside institute he can supply to her student or researchers. For trading, see in this column, third column, fourth column, that is research for education, in-house breeding as well as trading. For small animals, if SL is written, that means small as well as large animals. So this type of things we have to enter in Form B. When you are looking in Form B, we have to put the registration number from there. We have to procure animal or in which we are going to perform the experiment. We have to write their name as well as registration number and date of the registration or renewal of the registration. So as we already discussed, Breeding Act was passed in 2001 and 2006. At the central level, there are 19 members team is there. In current days, Dr. Abhijit Mitra is chairman and Dr. S.K. Datta, Joint Commissioner of Animal Husbandry, is the member secretary. So these 19 members team will decide any change in regulation of the animals guidelines. So what are the guidelines? First and prior most important is the animal which are going to perform the experiment. that should not subject to the any kind of pain or suffering. Means during experiment and after experiment or before experiment, we have to take care of this. And no unnecessary sacrifice of the animals, as well as we have to take or procure the animal from registered breeder only. These are the few guidelines. And when we are planning any studies on an animal experimentation, we have to go from the lower species to the higher species, not directly go to the monkey, or we have to start whether this animal models are, that depends upon the suitability of our experiment, whether that is mice or whether zebrafish or Drosophila or C. elegans. There are so many invertebrates model is also there. So we have to see which type of studies and what is the exact model. Then we have to start. And these are the various, under this act, various rules and guidelines are there. So we can go, you can go through the CCSCA website. There is all the guidelines are there. So under these guidelines, there are two important is veterinary care. Means when we are procuring animals from outside the institute, we must have to keep in quarantine periods. Quarantine is essential to know whether our animal is free from diseases or not, whether they have many symptoms of disease. So we have to take the animals which are healthy and free from the diseases. So veterinary care is very, very important. For small animals, quarantine period is 1 week to 1 month. Like in tuberculosis studies, we have to keep animals for observation for 30 days. Otherwise, 14-day acclimatization period as well as quarantine period is enough. In case of large animals, we have to keep for up to 6 weeks. As already I have discussed, animal procurement. When we are procuring animal from any outside institute, we have to brought that animal. During transportation, we have to maintain the AC vehicle, or we have to keep these animals in proper cage. We will see in later slide why in proper cage, and number should be as per the guidelines. Because if you are procuring too many animals, that may cause stress and there may be some mortality. And during transportation, we have to provide feed and water. Nowadays, for water, there are water jelly are there. So it will be used for the if prolonged journey is there, we can use the water bag. That jelly-like material are available in market. So another guideline in this, which are given by the CCSCA, that is layout. What type of animal house structure will essential. So in this 50 to 60, first is that animal house should be away from the human activity. That should be in separate area. And 50 to 60, most of the 60% of the area should be taken care by the animal holding rooms. That is, animal holding room should be up to the 60%, and rest of the 40% we have to divide in experimental area and non-experimental area. In experimental area, we have the OT, pre-OT, post-OT, necropsy, these types of things. In non-experimental area, we have the feeding storage, bedding storage, we have the quarantine room, we have the sick animal room, we have the office, like this. So we, apart from this, in layout, when we are planning for an animal house, we have to take care of these things also. Because so many times animal house is made in the beginning and there is very narrow, this one, corridor. So animal equipments like IVC, if you are in a later stage, if you are planning for this IVC, the IVC machine will not go through that corridors. So we have to think in early stage we have to plan at that time of making animal house, there should be wide enough, at least 4 feet, 4 to 5 feet, 4 to 6 feet wide corridor is essential. And building material must be concrete building, because the sound we will discuss in later. Less than 85 decibel sound is essential to maintain in animal house facility. Just like this, animal doors. When we are planning animal house, animal door must have, there is a glass window to see from that, observe the animals from outside, not to disturb if you have the breeding facility too. So now you see, these are the 5 criteria which is essential to maintain in animal house. Like temperature. Temperature must be between the 18 to 29 degrees centigrade. Based upon the species, there is some variations are there. Relative humidity 50 to 70%. If your relative humidity will change and temperature change, in young pups there may be chances of ring tail disease. There will be corrugation, and later stage that tail will be slug off from the animals. So we have to avoid this infection. Similar to that, ventilation, 12 to 15 air cycle change per hour is essential. If this cycle is not in proper way, what will happen? The ammonia levels, as we know, the rat urine contains lot of amount of taurine. So ammonia production is more. So when ammonia production is more, that animals may come into stress. So we have to minimize the stress. So that ammonia level is less than 25 ppm per million. But for that is our limit. 25 ppm is our limit, because that is for human. For animals like rat and mice, it is less than 5 ppm. So we will imagine at the time when we are entering an animal house, we are feeling a smell, means that is up to 25 ppm. But when the ammonia level is reached 5 ppm, animal may get discomfort. So sometimes when we go to the animal house, a rat having this red pigmented deposition on the eyes, that is phenomena is known as chromodactylia. That is porphyrin, red pigment is normal phenomena, but the secretion increases when the animal are in stress. So we will identify this. Another is light intensity and frequency. So 325, less than 325 lux is essential to maintain in animal house. If intensity is more, there may be chances of blindness, because albino mice has more chances of developing cataract. So another thing to avoid this in incandescent bulb, we have to rotate. Means upper, within 1 week we have to upper case to lower and lower case to upper, move. So every animals will get the proper. Another important thing is that light cycle, means 12-hour light and 12-hour dark cycle. That is essential, because light intensity and frequency also hamper the reproductive synchronization. If that cycle will change, the reproductive performance will be disturbed. So we must have take care of these things. And I have already spoken the noise level. Noise level should be within the 85 decibel limit. So overall, what we have talking about this Animal Welfare Act, that is mainly depend upon this 5 freedom, 5F. The first, freedom from thirst, hunger and malnutrition. Our animal which we are going to keep for animal experimentation, that must be free from hunger. Means we have to provide ad libitum feed and water. And another important thing, when we are talking about the nutrition, we have to also think the variation in species wide, like guinea pig. They need the vitamin C, that is 10 milligram per kg body weight. In that ratio we have to think because that vitamin C deficiency is there. Next, the freedom to express the normal behavior for that species. How they will behave normal? We have to provide the proper case. Like if we are planning to keep the monkeys, there must be some enrichment. We will talk in later slide. Enrichment is also essential, like jhula, like hanging, because they have to express. If you are keeping mouse having the normal activity burrowing in the deep. So if you are not keeping too much bedding, like 1 millimeter or centimeters, then he can't able to burrow. So we have to give the social interaction also to that animals. Freedom from fear and distress. Similarly, freedom from pain, injury and disease. Freedom from discomfort due to the environment. So we have already discussed the temperature range and all other things. We have to maintain in this. If our animal is freedom from this 5F, then only we will get the good animals. When we get the good animals, our data will be good. So I have already in under 5S, this is the housing system, adequate space for free movement. If we are keeping cats in a rat cage, we are keeping the dogs. So many times, if you are not giving the proper space, the movement will be not proper. That will hamper the animal's movement and leads to the animal in stress, and later on that stress convert into the distress. Food and water, that food should be palatable and should be free from the toxins. We have to think about this. Bedding material is also free from the toxin, and that should have the absorbent capacity. So the urine, as the behavior of this rat and mouse, he will toilet in corners. So ammonia level will be more if that will not absorb. So nowadays corn cob is used most widely because they have the very good absorbent capacity. And depend upon the ammonia level or depend upon the density of the animals in that case, we have to change the frequency once or twice in the week. So now in that CCSCA there are two important things that is most commonly I have seen in so many institutions. If they are doing research on any genetic engineered product or any transgenic mice or any alteration modulated models, the Form B or project must be passed through the IBC, Institutional Biosafety Committee. Then only you have to put in Institutional Animal Ethics Committee. It is essential. So what is the IAC? IAC is nothing but Institutional Animal Ethics Committee. It is the local representative of CCSCA, because CCSCA can't possible to inspect all the animal facility throughout the India. So he has invested his power into local body. So he can decide there whether this project is suitable, feasible or not. And every experiment should be performed under the well qualified person. They have the duly qualified in that behalf, that degree holds in veterinary medicine, science or laboratory animal science of any university. Like already Chakadhar sir has mentioned, in European countries they have the FALASA, Feline Association of European Laboratory Animal, they have the certification course C and D. After that course only, 10 days course for veterinarians and 8 days course for the normal MSc graduate or PhD scientific personnel. So without that course, we can't enter even the animal house facilities. Similar to like that, in American society, there have the program like diplomate of American College of Laboratory Animal Medicine. Only that degree holders can perform any experiment. This is essential because we have to understand, as sir already, Chakdhar sir has explained, we must have to keep the balance between science as well as the animal welfare. In our India, that much not issue of this one animal welfare, but in European countries, there is so many chances of interference of NGO. If you are any mismanagement in animal handling, they will suddenly, persons will come and they want to stop that animal facility immediate. So must have to take care of animals with humanity. So what is the quorum of this IC? IC have mainly 5 internal committee. Internal means the 5 members must be from that institute. That is 1 biological scientist, 2 scientists from the different discipline, 1 veterinarian who is involved in the care of the animals, and 1 scientist in charge of the facility in that establishment. Out of these 5 members, they have to nominate 1 chairman or 1 member secretary. Sometimes the head of the institution, who is non-biological person, but he wants to be the chairman, then we have to keep 6 members in internal and 3 members that is nominated by the CCSCA government of India, that is come from the outside as the representative of CSCA. So he will see whether we are doing the experiment with proper care or not. In that, one is main nominee. His main nominee is unavailable and he has given in written, then a link nominee will act as the main nominee. Otherwise, there is no need of link nominee. One is a scientist outside and another is social aware nominee. So this validity of this IC will be for the 5 years, and half of the members will be changed during the renewal procedure. When we are applying for the renewal, half of the members will be changed. So what is the key responsibility of this IC? When the IC meeting is called upon by the member secretary under supervision of chairman, he will see the protocol. Means first is review of protocol. Means what the researchers or scientists has submitted the form B, they have to go to this with proper, like in when we are seeing this form B, there are so many columns are there. The IC must have to go through this, whether this animal have already data is published, research has already done somewhere else, or what animals you are taking, whether this is statistically significant or not, based on this experimental design or all the other things. And another is harm benefit analysis, whether this studies having some role for the human mankind or not. If that study is not just like a sacrifice of animal just for the sake of one paper. And after in this, there are so many things what sir already have told, 3 years. So in this, they must have to see this alternatives. So we have to encourage alternatives. If there is no data in public domain related to your research, then you have to plan the pilot study, not directly go for the 100 animals, we have to take care and start the experiment. We have to first take 3, 5 like this animals, and we have to do that pilot study. From that data, if you see whether this molecules having some potency, then we have to go for the final experimentation. So during this procedure, when the project is approved, after approval, IC has to monitor this all the projects, what is the conditions is there, what are going on. So in this, what sir has already explained, one is refinement, that is more concerned in European countries, that is also taken care of in India, in mostly in private sector. I have not seen in government sector that much, but this is the data what is available in public domain shows that the animal enrichment, like when you are using the zebra fish, we have to give the plantation, some rocky materials, so animals will feel we are in natural environment. So when their feelings is natural, the data will generate also natural, more reliable. So in like in mouse or rat, there are so many models, igloo and other things. So many times we see the red color, because animals is red, rat and mouse is color blindness. Okay, so only they have the rod cells. So most of the times when we keep these materials, we have to keep the red only. So this is the essential part for the refinement. This is the how the animal research protocol will be this one refined based on the scientific justification, animal welfare consideration, risks and benefits analysis, and ethical consideration. One important things for this cancer research, most of the times they are planning cancer research and they have not defined the human endpoint. That is also essential. So IC committee must have to see this, whether this person have defined human endpoint or not. Means when we have plan, like suppose that one cancer model is there. If you are taking mice and we have to define whether this size of the tumor means 5% of the body weight, 10% of the body weight, then we will slaughter this, euthanize that animals. Because unnecessary keeping long time, the animal may get more pain. So when our scientific means output will be confirmed, we will euthanize that animal, so the animal will get the less pain. This is the another work of this IC, means they have the regular inspection of that animal facility, as well the animal protocol, review the progress, means the researcher should submit his report in the next IC meeting, and if there is any change, if you are taking biopsy mice in protocol, you have started work, so this is not available, we have to start albino means albino on Swiss albino. That is not correct. We have to any deviation in protocol which we have submitted in IC meeting, we must have to take approval. So these are the another things what we are doing today is important for this IC. They must have to promote the training program, workshop or continuing education, so that the many researchers who is not knowing, so know the basics and implement the ethical concern for the animal experimentation. So IC meeting, all the members should maintain the confidentiality of that meeting about the any projects, and minimum 6 members is required, essential for that quorum, meeting quorum. Presence of main nominee is essential to validate that, and social anomaly at least once in a year. If available in all the meetings, that is very good. Otherwise once in a year, because this person is only responsible for this, seeing that because they are, CCSA is thinking like that they are the lay person, whether we are providing proper care to the animals or not. Annually, at least once meeting, if institution having the less than 40 protocol, if more than 40 protocol, we have to go for the 2 meetings in a year. IC will be the approving authority of experiment on a small animal only, not for the large animals. If you are planning for any experiment on large animals, we have to put in IC first. IC will see the protocol and it will recommend to the CCSA, and final approval will be given by the CCSA. Member secretary has all the right for conducting meetings as well as keeping all the records, and they have to submit this minutes of the meeting within the 15 days of the meeting, and PI and co-PI will submit the study in Form B. Based on that Form B data, the committee will approve or disapprove or approve with some recommendation. So these are the 5 types of forms available: type 1, type B, C, D and E. Form B is for the animal experimentation. So we will discuss this. How we will fill this Form B. In earlier we have seen this name and address of the establishment. Everybody see in that name and address of the establishment where this animal experimentation will be done. We have to keep this in, like we have written here, Indira Gandhi Institute of Medical Science, Patna. Next, second is in section first page, registration number. So next is registration number. Means here our is government organization, RE Research for Education. Now breeding facility is also there. So BI and date of registration. So next, third, name and address and registration number of the breeder from which we are procuring. So we have to keep here the registration number, just like I have shown in previous slide. Like if you are taking from private organization or government organization, whether they have the trading facility or not, we have to keep here. And place where we have to keep this animal for experiment, we have to put here, like central animal house or departmental lab where we are performing this. Place where experiment is to be performed, like I have written in central animal house or departmental lab, we have to write this. Next, 6 is date and duration of experiment. So after this IC, we have to keep the exact month and year after IC meeting. Not like days, 60 days, somebody has written, I will keep animal for 60 days. Not like this. We have to write the month and year after this IC meeting. Because after approval, you are supposed to authorize for the performance of this animal experimentation. Next, 7 is type of the research. Whether this is the basic research, whether this is the regulatory research, whether this is the contract research. We have to write like this. And in last, we have to sign there. PI should sign there with her or his designation. In section 2, we will come to the section 2. In section 2, first is name of the title. We have to write the complete titles, not short form. We have to write in complete whole titles, what are there in your protocol. Then second is principal investigator, researchers or advisor. We have to write here the PI name. Okay, principal investigator name. If a student are there, MD student or some student is doing any project, they have to write her guide name or his guide names. And in next, his designation, department, telephone number, email ID, or experience in laboratory animal. Means they must have the experience of 40 years, 30 years, 18 years. But how many years of animal experimentation experience, that should be written here. In third, list of all individual authorized to conduct. तो सर ये बता देते हैं ना ठीक. So in third, we have to enlist all the individual. Normally here somebody is written like 1 or 2 who is going to perform experiment. All the person's name should be included here, not the person who is involved in research. So name, designation, department, telephone number, email ID and experience. 4, 5, 3, 2, 1, whoever is there, we have to enlist here. Then fourth, funding source. We must write the funding source, whether this is from the institute fund, whether from the DBT, DST or any other funding source, we have to write there. And not only budgetary code. Sometimes they have written like budget code, we have funded from through this budget. No, we have to write all the ministry or all the details of the funding agency. Fifth is duration of the animal experimentation, initiation of the project and completion of the period. We have to write both. Next is detail of the study plan to justify the use of animal. We have to write all the literature of review, why we are doing this research, why we have selected these animal models, what are the differences, not sake of the information only, no, this is the animal model is essential, because of that we are using the animal, because animal model is essential for this study. This is not the exact reference. We have to go to the PubMed index or any search index and we have to put their reference, whether we have search, whether any alternatives is there, whether this experiment has already performed or not. Based on that, we have to give that also in reference. Then in 7, we have to write the species, strain, age, which strain is there, what is the age, what is the weight, what is the gender, whether male or female both are there, we are taking, or only female are taking. Then D is year-wise breakup of that experimental protocol. In which group you are taking how many animals, and year-wise, because IC will give you approval for 1 year only. After submission of that data for 1 year, then he will permit for next year. So you have to give the complete breakup of group-wise breakup. It is rationale for animal uses. That's why I have already told why this animal uses is necessary. We have either cell culture, sometimes so many molecules are there. So we have a screen on cell cultures and we have find this out of 60, these 4 having the good potency. That's why we are taking these animals to validate in animal models. Like this, we have to give the reasoning. And whether similar studies has been conducted on in vitro models, we have to search it, and if not there, we have to write. We have searched this, and that is the reference you have to keep in this form B, attach in form B. Then only the IC will give you approval. So these are the important things, because sir, as Lodi Chakradhar sir has told in a speech, So many times there are so many unpublished data is there. So a study will repeat in other institution also. So we have to avoid this. For this sake of purpose, nowadays CCSE has issued one guideline. Every animal experiment should be published in public domain. Whether if you are that is not published, sir, 10 minutes. 10 minutes. Sir. Okay, we will discuss this one later on, because up to that time you will fill this form whatever possible. We will discuss after this one comment. Thank you.
Thank you, sir, for your engaging session. I would now request Professor Dr. Vikas Bedi, sir, to kindly come and felicitate Dr. Manoj Kumar, sir. I now request Dr. U.S. Chakradhar Rao, sir, to kindly come and felicitate Dr. Pankaj Prabhakar, sir.
Thank you, sir, for such a nice presentation. Now we will proceed with a panel discussion on topic from bench to bedside without animal, myths and or the reality. The moderator for the session is Professor Dr. Haryar Dikshit, sir, Principal NSMCH Beta or President ASP IGIMS. Over to you, sir.
Good morning, everyone. It gives me immense pleasure to welcome you all to today's panel discussion on a topic which is more relevant from bench to bedside without animal, myth or reality. So before that, the starting of the discussion, the background which I want to share with you, let me begin with a story. Many of us know the CD28 superagonist antibody TGN 1412 episode in 2006. 6 healthy volunteers in a human trial of monoclonal antibodies, in which dose was 500 times smaller than that found safe in animal studies. In such situation, developed a catastrophic cytokine storm within minutes, despite extensive preclinical testing that had shown no warning signals in animals, even in non-human primates. The event forced regulators, scientists, and ethicists to rethink the entire chain of preclinical evidence. 2 decades later, we are witnessing the rise of new approach methodologies, which is in short named NMA, organ-on-chip systems, stem cell-derived organoids, in silico toxicology, and AI-assisted prediction, all aiming to mimic human physiology more precisely. But enthusiasm must meet caution. AI tools need high-quality human data to learn from, and those data sets are still sparse. organ in chips cannot simulate a whole human body. Organoids lack functional vasculature, innervation, or immune components. With that background, let us invite our panel to examine what is truly possible today, what remains aspirational, and how we train the next generation to bridge this critical gap. For today's discussion, we have 5 eminent subject experts from different background and experience. We are privileged to have Professor Vikas Mehdi physically present here as a esteemed panelist who has profound understanding of regulatory issues. Also online, the most visionary academician, administrator, Professor N.R. Vishwas-ji, who is currently working as Vice Chancellor of Sri Balaji Vidyapith, Pondicherry. There are many achievements. He delivered Srimati Rukmani Animal Annual Internal Oration, and is a teacher of, he got Teacher of Excellence Award also in 2023. He delivered prestigious Professor Hirendra Kumar Chatterjee Memorial Oration at 91 reunion of Founder and Days celebration at R.G. Kar Medical College, Kolkata on 19th December 2023. And we have all seen his great work for the development of इंदिरा गांधी इंस्टीट्यूट ऑफ मेडिकल साइंसेज. आप कुछ काम करो, मैं उसका रिजल्ट दूंगा. जैसा कि सुभाष चंद्र बोस ने कहा था, तुम मुझे खून दो, मैं आज़ादी दूंगा. थैंक यू सर. यू आर हियर. प्रोफेसर विकास मेहदी, प्रोफेसर एंड हेड डिपार्टमेंट ऑफ फार्माकोलॉजी, PGI Chandigarh. He is a regulatory expert and he is omnipresence. You will find in every conference, workshop, he is present, and he is so humble, so down to earth. He is fine human being. Professor Upu Gundari Satyanarayan Chakradhar Raoji is from Department of Medical Oncology, JIPMER, Puducherry. And he is a founding member of Pharmacogenomics Research Network, member of Clinical Pharmacogenomics and Implementation Consortium, CYPLL Nomenclature Expert Committee member for PharmaVar, an external collaborator of Cancer Research Platform in Pediatric Oncology and Hematology of University of Geneva. Also here, Dr. Vinod Nair, he is Director, Oncology and Precision Medicine, JS Associate, Gurugram, Haryana. And Professor Nitesh Kumar is Assistant Professor in Pharmacology, National Institute of Pharmaceutical Education and Research, Naipur, Hajipur. Welcome to all of our panelists. First, I would like to invite Professor N.R. Vishwas because he is going to another meeting immediately. So, your views, kindly express your opinion on, from your experience, what are the potential drawbacks of animal-based preclinical studies in current scenario of drug development? Technically, where do these fail? Thank you. Sir, I invite you, Chakradhar Raoji, come on physically.
Hello.
Sir,
Can you
floor
hear
is
me?
yours, sir. And
Yeah.
the question, I repeat, from your experience, what are the potential drawbacks of animal-based preclinical studies in current scenario of drug development? Technically, where do these fail? Sir.
Dr. Dixit,
Yes, sir.
thank you very much for telling good words about me. And I see there are other people in this panel discussion, including Vikas, Dr. Rao from JIPMER, Vinod is also here, Dr. Nitesh from JIPMER, Hajipur, Naipur. They will continue all this. Unfortunately, I have to attend the Vice Chancellor conference that is already started at 11. I was told by Dr. Sukalan that this panel discussion will be at 10 o'clock. So I suited and fixed my timing accordingly. But anyway, I wish they will do the job properly. What I want to just briefly tell that we can't do with animal experiment, though sometimes it is found that 90% of the data we generated through animal experiment, particularly the safety and toxicity studies, that does not reflect in when we translate to human, because still side effects happened. Even on 500 means 500 times less the dose of the animal dose causing side effects, very serious side effects, even causing death. But still, we need to do animal study, and that's why, with a lot of efforts, we made the animal house in IGMS. You are present that time, I was also that time. And we have to do animal experiment, and we have to train our PG. Even when I go sometimes now also as an examiner, the PG, they don't know how to handle an animal, how to catch a rabbit, rat, mice. So this is very unfortunate. So my view is that animal experiments should go. Of course, all these other new technology is having some fruits, some results, but still we cannot do away with animals. So the question was that this from bench to bedside means without animal experiment, we do in vitro study in the laboratory, and directly with that data we will go to human trial, or that means bench to bedside without animal. It's a myth. It's a myth. I support and I am in this favor that it's a myth. That means still we have to do animal study, though that data may not truly reflect. And as you know that Phocomelia, this episode, that in the animal study it was nothing found, but suddenly it happened in human with thalidomide. So it's not totally reflecting all the data, all the result what we get in animal study, but still we have to do it, and that is my punch word. Thank you very much for giving me opportunity, and I have to attend another VC meeting, that is very urgent meeting, and I wish my best. I wish this of your all efforts what you are doing here great success. Thank you very much for giving me this opportunity. Thank you.
Thank you, sir, for your expression of thought. You are correctly said that 90% data does not translate, and still you said about the topic is without animal it is myth. Still we have to do animal studies. Thank you for sharing your views, sir. Thank you. So, I would like to begin with the gaps in preclinical methods of the issues. So, I invite Professor Dr. Vikas Maiti ji. What does bench to bedside without animals truly mean in modern biomedical research? You have to explain all this around this.
So, thank you very much. Just I wanted to say that when you develop a drug, it's for human, but do not forget that you need a drug for veterinary also. Okay? So when we had a meeting on FDCs, fixed dose combination, so I'm there since 2011. So we invited the veterinary group for deliberation of fixed dose combination for veterinary. And that they said that this is the first time somebody had called us after independence. So all the drug we develop We develop for human as well as for veterinary. And we have a concept that this product should be, at least we had a G20 meeting, So one world health. We work on one world health. Product should be safe for human, safe for animal, safe for environment. Now coming to preclinical studies, that we need to understand that all the preclinical studies we do so that we can utilize for first time in human, in a healthy human volunteer. So if I look at that, depending on clinical use, you generate the data. I wanted to use for 10 days, I need acute toxicity data. But that data also give that information about, in case of overdose, in case of accidental, homicidal, suicidal, how you are going to manage, how clinician will go to manage. Okay? So if you take an example of paracetamol, we know up to 4 grams is safe. But if you go beyond 8 grams, it is going to fail the liver. So you take a precaution that we have an N-acetylcysteine. So all the drug, it given indication for a clinician that how you are going to manage in clinic if there is overdose, suicidal or homicidal, whatever the reason means. Second point is we do all the preclinical studies because we wanted to use the drug for a month together. Beyond 1 month, we go for a repeated dose toxicity studies, at least 14 days, twice in a week, 28 days, 60 days, 1 month, Then, 90 days, 1 year. So what do you wanted to learn from that? If you use the drug of 10% of the lifespan, how it is going to behave, human body, that we generate and recommend. So in acute, you generate a NOEL and LOEL, no adverse event at that level and low adverse event at that level. And when you do repeated dose toxicity study, you confirm that, NOEL and LOEL. So then, depending on the clinical use, if you go beyond 6 months, you are going to ask that long-term toxicity data. Long-term toxicity data, that is for 2 years. Mice and rat, they live for 2 years at least. So you are going to give the drug and see that first question you are going to ask is, can it cause mutagenicity? Because you are chronically taking a medicine. Can it cause mutagenicity? Mutagenicity does not mean that it will develop a carcinogenicity. So you also do carcinogenicity testing. So if you look at all together, you do phase 1, you do phase 2. By the time before you start phase 3 study, you are going to ask, regulatory is going to ask that, can you keep the data for long-term toxicity data? But over a period of time, we also learn that a lot of new drug that had to be withdrawn, like cisapride, terfenadine, because if you look at the WHO list, these are the drugs, it causes concomitant medication, it causes QT prolongation. Okay, so we also develop a special branch on safety pharmacology. Is it safe? You give a drug, can it affect the CNS? Can it affect cardiovascular system? Can it affect, respiratory system? All GIT, all the system we check. Then you might be asking the question, I develop a drug, can I use in pregnancy? Pregnant woman. So you have a segment 1, segment 2, segment 3, and then for male fertility also, somebody participated in phase 1 study and, he's only 18 years, he likely to participate in a phase 1 study. Later he went and got married and doesn't have children, and that is why we do all reproductive study. So if you take an example, I'll just give you an example, phenytoin. Phenytoin causes pseudolymphoma, from day 1. Because when you sacrifice the animal, you are looking at all the vital organ. And second one is 47 organ, each and every, even lymph node. So from day 1, it can cause pseudolymphoma. And that given indication that when you plan for a phase 1 study, you take all the precaution. If it is liver toxic, you are going to ask, is it permanent or is it temporary? Is it reversible or irreversible? Any damage, any event take place, and that's how you look at. Now, let me look at during the COVID time. It is extensively used. All this, animal use during the COVID time is hugely increased. So today, if you say that, if you go to any of the forum, experimental biology, where all the branches are there, so you can see that how extensively they are doing animal research. Obviously, it is for productive. So if you do any study, this should translate into the bedside. It should say that I developed a product, I wanted to do this, nobody has done that, and that is why. Obviously, if you take it, Dr. Biswas has already said regarding thalidomide. actually what happened in 50s when they developed, they did it in mice and rat, but they also did in rabbit, and rabbit they could find teratogenicity. And what they did, they hide the result. They hide the result. And later when one pediatrician had written a letter to Lancet, that became a signal. People came to know that there are 10,000 baby born with phacomelia. And the court has asked for a data, and they could find that they already knew that it causes teratogenesis. What we learn, so that is why you say today you do a smaller animal, mice and rat, and you do a higher animal. Because looking at this incident, it says that you need to first question, that answer to be questioned, is there any species variation? How the drug is behaving in smaller animal, how the drug is behaving in higher animal, and that is why you do. I'll just give you an example. During COVID time, we had to go to jungle to get the monkeys because we have only limited 4 places monkeys available. Today we are working on Zika virus. We want a monkey from African origin. We don't have. I mean, there is one vaccine for contraceptive. We wanted to have a monkey, but we don't have a monkey. Then we had to go for an alternative marmoset. And that is how, we need to have all this expertise. As I said that we have a lab, but particular spaces, if you go for it, like if you say COVID, what do you need for a COVID? You need a drug which can be immunomodulator. So what kind of animal you go? You at least biopsy mice, hamster, preferably. And that is why all the facility, that is why that, ICMR has created a bigger facility where you can procure the animal, you can do the study. I think you need to look at that how strong our preclinical development. Obviously, we also promote to go for an alternative one. As you understand, you say what preclinical, but we also use is non-clinical. What does it mean? How the non-clinical word come? Because you have an alternative, like US Commissioner has signed, you need not have to do the preclinical study. But one more important point is, like last time when I had gone to Glasgow for IUFAR meeting, one of the ladies stopped me. And she asked me that because do you have a guideline for, clinical trial for male, female, and others? You understand others? So in preclinical study also we look at how drug is behaving in male, how drug is behaving in female. Why this point is important, till 1994 we did not have a provision for evaluating drug in case of a female. You only recruit healthy male volunteer. Okay, so that means we do not know how the drug is behaving in female, and they say you must include, all male and female, provided you take an adequate protection for pregnancy. And that's how, it can reflect. If you ask the question, can I use for a geriatric population? We don't have a data. Most of the drug, when you buy it, it said that we do not have any data below 12 years, below 5 years, below 2 years, or geriatric population. And that is why, all preclinical data need to be generated, and we can use AI. If you take an example, we do a lot of stroke model. We have varieties of stroke model, and there you also look at construct, phase, and predictive validity. But when you go to clinics, all the drugs are failed. What is the reason? So any of the drug, when you use for a neuro, your question should be coming, does it cross blood-brain barrier? Is it targeted? And you can use the AI that how much it will be in a clinical situation. If you take an example of autism, Alzheimer's, you can use the AI. How much it is, people are using AI. So that you can say that when you develop a clinical protocol, okay, this dose is sufficient, even for cancer also. And that is how we have to predict. So it's not conventional animal experiment, use the new technology. And if there's an alternative one, like you said, start from, proof of concept. You do in silico. And when you generate the data of in silico, it does not mean there is no evidence. It is called zero evidence, but it will give a direction that what is the mechanism of action, what dose will be effective. So you get all the data. Based on that, you generate the in vitro data. Then it will be very good data to decide for in vivo studies. And that's how you go for all regulatory toxicity study and say that you have enough data to support that you can go for a clinical trial, So if you take an example, you refer it to monoclonal antibody. So they said that they had done in monkey 500 times more, but it is unpredictable. You have a type B reaction. Even if your dose is increased, less dose, it can cause anaphylactic. They had in gangrene. Second issue was coming is, French study. It's a cannabinoid study. They lost a couple of healthy human volunteers. And that is why today if you say, when you go for a clinic, you do phase 1, complete 1 patient, then you go for the next patient. Don't recruit 12 to 14 patients. And that's how that all this incident, when it happened, we learned a lot of stories. Like yesterday, day before yesterday, there is an application. They wanted to use the drug for, local application, like vaginal. They said that we had done orally, so we can go for a vaginal application. Then you say that, it is totally different. You have to generate the local toxicity data. Today you say that any drug we develop, why we go for a rectal tolerance? Because if the drug is proposed to use in children, you cannot give it orally. You cannot give it IV. So at least you can give it rectally, it can be appropriate. So these are the justification is there, all the data you generate, and then you say appropriately, probably it is safe with certainty, with the dose and duration, and that's how we have to look for that. But I said again and again, new technology, let's adopt, let's validate, let's apply. Thank you very much.
Thank you, sir. Your punchlines as you delivered your inaugural speech, Atmanirbhar Bharat, and similarly, you had just now told about one product, one health, one world. So the vision of our Honorable Prime Minister, one health, including everyone, not only human beings, not only animals, even the environment. Second, very well talked about the guidelines, importance of guidelines of male, female, and others. As you said, we can use AI, as sir has said, and animal studies are relevant, but you must promote others, alternatives also. You can utilize AI for, and as he said correctly, he exposed the thalidomide crisis, because responsibility and integrity is important, because thalidomide crisis was due to hiding the data. So that is important in your research publication. Don't hide. So thank you very much, sir. Next, now it is, let us enter into the scientific and technical aspect. My question to Dr. U.S. Shakardhar Rao ji, how reliable are current non-animal models like organs on chip? Next, आपका regulatory भी है, sir. Sir, yeah. So, how reliable are current non-animal models like organ on chip, organoids in silico models, or AI simulations compared to animal testing in predicting human outcomes? You have talked about it in the earlier session also. So, predicting human outcomes is very important.
Thank you, Professor Dikshit. And thanks to Professor Mehdi, Bikash Mehdi, because he has explained many of the important aspects that I'm going to maybe repeat, but in a different way. Before that, maybe I would like to give one example of our own study, what we have done to understand some inhibitors of drug metabolizing enzyme. You may all know UGT2B10. It's a glucuronidation enzyme, right? So that what it does in the body is to glucuronidate many of the molecules. Some of the immunosuppressants that we use in stem cell transplantation setting in pediatrics, there are some of the substrates, and some of them also could inhibit this enzyme. But what we don't know, ideally how you would study an inhibitor. So first, ideally, either you go and administer a substrate, and then you give a putative inhibitor, and you see the metabolism pattern in an animal, or maybe you can extract microsomes, liver microsomes from an animal, from liver. For that, you need to sacrifice the animal, or otherwise you have a recombinant microsomal preparations that are coming from liver cells and human tissues biopsy samples that you could use in vitro, and then you could test for metabolism. Okay, you give substrate and you treat the microsomes with inhibitors, and then you look for what's happened, whether the metabolism is actually happening or it's inhibiting the metabolism, because you measure those levels of the parent drug as well as the metabolite by other analytical methods. It's a laborious process, and also it has a involvement of animals or other sources of obtaining these biological material microsomes. How to avoid this? Can we do a little simulation to understand if the inhibitor that we think could inhibit are the ones that really could inhibit this enzyme? Yes, we could do like in silico simulations. So that's what we have done. So we know that some known inhibitors of this enzyme already that are available in the literature. So what you could do is you could just take the structure of this UGT2B10 enzyme, which is available in the public domain, and then you take this molecule structure and you see how both of them are interacting together in the presence of the substrate. Okay? And then you have some understanding of known inhibitor, how it interacts with the protein. And you also take some other negative controls, which are not an inhibitors, which are some random molecules that can never interact with this enzyme. And then you understand the interaction pattern. So you have a kind of a blueprint for positive control, you have a blueprint for negative control, and then you test all your molecules that you think could be putatively inhibiting your molecule. And then you see where they fit into, close to the positive control or close to the negative control. And then you have 100 molecules, maybe you could narrow down to 10 molecules that you could test again in the in vitro systems or in animal systems. That's how instead of testing 100 molecules directly on animals or in vitro systems, which require some biological material, maybe you could cut down on the resources as well as wastage. You can also refine your methodology. And from there you can go into the animal systems and maybe into the human systems of giving microdoses and then giving inhibitor in microdoses and testing for metabolic patterns. So that is something that we could understand. And to answer your question to the outcomes really in the humans, we know that from animal preclinical systems, the actual translation into the human is not 100%. We know that already from the beginning that we started using these systems. But these systems are well established for a long time. So we have about translation of 70% from the findings from preclinical to the actual human studies. But when it comes to different aspects of the drug development, from pharmacokinetic behavior understanding, pharmacodynamic behavior understanding, drug safety profile, these understandings or translational capacity might vary from the animal systems to the human outcomes. For example, take pharmacokinetic behavior, okay? Absorption, metabolism, and distribution and excretion, if you would like to study. Some of the in silico models, and as well as these organs on chips, they can outperform animal models, actually. To understand only pharmacokinetic behavior, I'm talking about, not the other PD behavior or the combination of both. So just to understand the PK behavior, maybe you could try to adopt these organ on chips models, understand from it, and then you come to a clear dosing for animals, and then learn from it. That's where you can refine. And we do have some lot of literature in the public domain which have even now replacing these animal models to understand pharmacokinetic behavior alone. And when it comes to efficacy, for example, the pharmacodynamic behavior models, so the organoid models, okay, the 3D cultures, so these are more efficient in understanding, because what we are looking at is a specific organ-related effect or tissue-related effect or a pathway-related effect. That is something that we could clearly gain insights by these organoid models. But what comes as a bottleneck is translating this information, learning from these organoid models, and then putting into a complex picture of the in vivo systems of the humans. And that is where it becomes a big problematic, especially when it involves some immunological component and then endocrinological component. For example, Professor Mehdi has already mentioned, like whether you are performing the testing in both males as well as females, because the systems could be different, the approaches could be different. You know that there are a lot of endocrinological component different between both of these species. So it is very important to understand that when it comes to when your phenotype, when your outcome of interest has an involvement of immunological as well as some endocrinological component, I think preclinical models can never be replaced by these other systems that we currently have. Maybe we might reach a stage where we could also develop something to understand them very well. But at this point of time, I think we have a very good models for pharmacokinetic understanding, organ-on-chips, and for efficacy models, organoids, 3D organoids. And then, of course, AI can integrate the information that we can learn from these different systems and predict us something to use as a refine design, to refine our design for the animal studies. And from there, we can get greater insights, and then that could make a real progress in the development of drug development process. And it is the case, it is the state of the art as on today in many other pharmaceutical organizations. So this is the standard care of practice these days. Thank you.
Thank you, Dr. Chakradhar Rao ji. Well expressed about the importance of simulation with negative control, and % you can achieve that. And particularly for pharmacokinetic action in silico model, outstanding you have said about that. Preclinical never can replace the alternative methods. So in nutshell, you are expressing that these methods are complementary to animal research. Thank you very much, sir. Next, Dr. Vinod is here. Dr. Nitesh, what are the ethical implications of continuing animal research when alternatives exist, and the ethical risk of relying on incomplete alternatives?
Thank you, sir, for giving this opportunity to discuss about this matter. I can say that alternative models are there. Then CCSEA used to say that you have to replace it. That is the one criteria. In 3 hours, 1 hour is replacement. So if you have method, you replace it. So that should be approved method. But one more thing used to come. Shall we replace everything with that? Suppose antipyretic testing, antipyretic study like the water testing used to be done in the rabbits, and a number of rabbits were used a lot. So the factory were looking for some alternative model, and they found out lal test is gold standard. They followed it. So they replaced rabbit with that. But suppose if you are going for drug discovery and development, you have to develop a drug that is antipyretic. Can you go with only lal test? There may be other effect, there may be side effect, there may be toxic effect. Can we go for that? Will it be sufficient to just go for with that lal test and clinical study? Not possible. So a comprehensive study is required. When you are going for drug discovery and development, then it will be required to go with the animal study also, or even whatever alternative models are there that you have to follow. Yeah, alternative models will help you to reduce the number. Like suppose earlier people used to go with 5 compound, 10 compounds in one go. Now using Schrodinger software or GastroPlus, you can go with 1 lakh compound in one day. You can screen 1 lakh compound and then you come out with 1 or 2 compound. Suppose if I can say about alternative medicines are there, like 70% of the people depend on herbal medicine. In those herbal medicine, one product, that is one extract, used to have sometimes 500 to 600 component. Which one is more efficacious? Which one is more toxic? So considering those points, alternative methods will help you to sort it out, and you have to go for animal studies, or you can say that alternative medicine both. So ethically it is very early to say that you can replace completely. Wherever multiple time uses are there, you follow that guideline. They are telling that replacement, wherever replacement is possible, you go ahead with that. Still 3D cultures where it's not completely approved. So 3D organoid cultures are there. Some of them are approved, but still many are not yet approved. even US FDA is finding it difficult to approve one model properly. So it's very
FDA, EMA view non-animal testing data.
Yeah. So thank you very much. I just wanted to take one. I have been right now with Austrian collaboration. So you are working on Alzheimer's. So you do all the behavioral parameters. So normally when you go to our ethics committee, they will give maximum number as 6. So when I took the data to Austria, so my collaborator said that I am not be able to do behavioral parameter with 6 animal. We need at least 14 animal. Okay, so that's how. But when you look at all the regulatory guideline for, generating regulatory toxicity study, so minimum number is 5. And for academic study, we said for a statistical reason, we are going for a 6. Okay, so that need to be justified. Then you say one rodent or other rodent. Most of the study, they say one of the rodent. But we do with, our regulation is by Drug and Cosmetic Act 1940 and 1945. So that was that schedule A to Y. And the Y actually talk about how to generate all the preclinical toxicity study and clinical trials, schedule Y. On 19 March, schedule Y is replaced with NDCT rule, new drug clinical trial rule, on 19 March 2019. So all recommendation from schedule Y has been incorporated in NDCT rule. So today you say that schedule Y is no more. It has been replaced with NDCT rule. But for preclinical study, everything it is written in schedule Y still exists. So accordingly, you have to do the study when you do all for a drug development process as per GLP or OECD study, we follow schedule Y. Okay, so that's how we generate. But there is one amendment in 2023 that if you have an alternative method, and when you talk about alternate method, then you have to ask that any regulator has approved that. Because most of the time when you go for a drug development process, it's not only India. You wanted to market in US, you want to market in Europe, all across the globe. So that is why we have to follow our guideline, as per Drug and Cosmetic Act or 1940 or 45, Rule 1945. But at the same time, if you work on biosimilar, then you have to generate the data as per biosimilar guideline. It was developed in 2010. Then again, the latest one is 2000. It's going to come up. Okay, so all the product you have to follow that guideline, particular guideline. All the new therapy like CAR T cell therapy, these are the new. So then accordingly you have to generate. Today you say that I remember we got a proposal in 2009 IND, and that was on curcumin eluting nano formulation for drug eluting stand. So we were discussing that we don't have a guideline for a nanoparticle. Like you say, any drug, if you use beyond 6 months, you have to generate a long-term toxicity data. But nano problem is it is written in the tissue for a long time. So that means even if you use for short period, you have to generate a long-term toxicity data. Later, 2014, we come up with a guideline for nano. It was adopted by, DBT, adopted by, DCGI office. So we can refer that you follow this guideline. Now, let me tell you, from a regulatory term, you have a guideline and you have a schedule. Schedule Y or NDCT rule is a rule, okay? So that means guideline, like you said, CPCSE or CCSE is a guideline. But this guideline is attached to Animal Cruelty Act. They're sitting in Dubai, they're sitting in New York, Indian company. So all, we are contributing. We are second largest now, so we are contributing. So that's how we have to go. So just read the regulatory guideline, accordingly you do it. Accordingly you do it. Thank you very much.
So the message is clear that you have to follow the guidelines, and all the preclinical studies you have to follow Schedule Y. Am I clear? Yeah. Alternative, you are having research on alternative methods, you have to follow both the global and national guideline. Thank you, sir. Now I invite Dr. Vinod. Dr. Vinod, are you here? Vinod
Yes,
Nair?
Professor Dikshit. I'm on Zoom, Professor Dikshit.
Yeah. He is an expert in precision medicine and oncology because the future of medical science is on precision medicine. My question is the future prospect, Dr. Vinod Nair ji,
Yes,
how cutting-edge precision medicine could redefine drug development pipelines, perhaps even making animal use obsolete after a few decades. Second question, which is the very important, is it realistic to imagine a zero animal research world in the next 20 to 30 years? Dr. Vinod?
Yes, sir.
Kindly.
Am I audible?
Yes.
Okay.
Yeah, go ahead.
So animals got medicine to where we are today. And at least in the next decade, I do not personally believe we would be in a animal testing free world. It's going to be more like we're still depending on animals, but the extent of dependence is going to be much more limited than what we have today. Now, regarding the precision medicine parts, I'd tweak it a little, the question a bit in a different way. We have always known that COX-2 inhibitors are effective, or COX-1, COX-2 inhibitors are effective in the management of inflammation or inflammatory disorders. The use of in vitro tests identifying or specifically measuring the inhibition of these inhibitors have been in place for more than 2 decades now, and they've already been used. And most of the laboratories, since the time when we were doing our PhD, used to start off with the in vitro test, have a proof of concept in the in vitro system, and then take it to animal models, thus reducing the number of animals that you would eventually end up using, because the in vitro concentration is to kind of give you a sense of what blood or plasma concentrations you're kind of aiming for. Now, coming to this question around the reliance on animals, the FDA recently came up with this guideline around the need to reduce a lot of animal testing that's happening right now. And one of the first molecule types that they've targeted are the monoclonal antibodies, which makes a lot of sense because the maps are designed against a particular target, and the cross immunogenicity ends up being a big hassle in terms of efficacy testing in the animal models. That's a way forward in the right direction. But one of the key questions that still remains is, there are still going to be off-target effects. Now, can these off-target effects be picked up through animal experimentation? At times it is, and at most of the times it's not. So what is the way forward? Right now, we cannot be in a world where you are conducting a in vitro set of assays and directly translating that to humans, because there would be a few things that are not elucidable as of now through in vitro and in silico techniques. So that part right now is still a very big gap as to how do you kind of move from in vitro, bypassing the entire preclinical animal testing and directly go into the humans. The other thing here is I focus a lot on oncology and drug development in the oncology space. And one of the challenges that's usually been associated with animal models of oncology is that it relies heavily on the xenografts, and it does not necessarily mimic the true disease progression and the metastasis that's seen in humans. Another reason is that most of the times the animal models that end up being used involve much more healthier animals as compared to the incidence or the age at incidence of many of these cancers in humans, which is usually about the age of 60. So that's why most of the data that you get in the clinical phases or preclinical phases does not actually translate into efficacy when it gets to the clinic. Now, the other angle to this is, since most of the drugs or monoclonal antibodies or even the small molecule inhibitors that are being developed right now are targeting these specific mutations, the true replication of this particular disease state in the animal through an endogenous development of cancer is not possible. And as you mentioned, the xenografts do not actually do justice. And FDA's decision on moving away is basically an acknowledgement of this difference that exists between a preclinical model and the human etiology. Where we would end up 30 years down the line, that's a big question even now, because as animal data, as in vitro, in silico experiments, data is being generated, that's actually adding to the pool of the knowledge base that exists right now. And over years, if we get to a point where this knowledge base is accessible and can be tapped into, then through QSAR and structure-active relationship kind of modeling, a lot of the need for animal experimentation can be done away with. But a bigger question for me right now is, the pharma has been doing it for a while, and they would continue down this path. The regulators are going to now increase their dependence on these non-animal methodologies, with thrust coming in from activists. So the question that ponders here is, as pharmacologists, where are we and what is the direction we are headed towards for adoption of these? Because if we continue down the same path where we are right now, heavy reliance on animals, a lot of the stuff that we are doing right now, developing right now, might face hurdles. And the individuals who are getting trained today, they may not be able to directly translate this into practice when they get into their professional journey.
Thanks, Dr. Vinod Nayarji, for your futuristic view. Still, we hope we translating into the actual form is having a great gap. And if we continue the same journey, at least we can hope we can replace animals in coming days, because that is more challenging. You have to create many environments. So now the session is going to end now. Today our discussion makes one thing clear. This is not a battle between animal research and new approach methodology, but a partnership in evolution. As I said earlier, it's a complementary, right? NAMS, organoids, organ-on-chips, and computational models have brought us closer than ever to studying human biology in human systems. They are transforming toxicology, drug screening, and early mechanistic research with remarkable precision. Yet, Biology is still more than the sum of its parts. Complex neural networks, systemic immune interactions, endocrine loops, and the subtle orchestration of organ communicating in real time remain beyond the reach of current in vitro and in silico systems. These are the domains where carefully designed animal studies still provide indispensable insight. The wise path forward is not replacement overnight, but intelligence integration using NAMs wherever they are validated and reserving animal experiments only where the living organism's complexity truly demands. Thank you all for panelists for giving enlightening us for their views, and it was a balanced discussion. And thank you our audience for patience hearing. Thank you. Thank you. Thank you all the panelists, Dr. Vinod Nair. Thank you.
Thank you.
Dr. N.R. Biswas, Dr. Mehdi, and Dr. Rao, and Dr. Nitesh. Thank you for your kind words and expression, making this workshop, a panel discussion, very important, fruitful. Thank you very much.
I would like to call Professor Dr. Haryar Dikshit sir. I would like to thank Professor Dr. Haryar Dikshit sir and all our distinguished panelists for sharing their expertise and knowledge. Now we would like to felicitate Dr. N.R. Biswas sir, Vice Chancellor Sri Balaji Vidyapeeth Pondicherry through online mode. We all also would like to felicitate Dr. Vinod Nair sir, Director Oncology and Precision Medicine, Gurugram through online mode. Now I'd like to call upon Professor Bikas, Dr. Bikas Mehdi sir, to kindly felicitate Professor Dr. Haryar Dikshit sir. Thank you, sir. I would like to request Dr. Sukalyan Sahar Roy sir, Additional Professor, Department of Pharmacology, to kindly felicitate Professor Dr. Bikas Mehdi sir. Thank you, sir. I'd like to call upon Professor Dr. Haryar Dikshit sir to kindly felicitate Dr. U.S. Chakradhar Rao sir. Thank you, sir. Now I'd like to request Dr. U.S. Chakradhar Rao sir to kindly felicitate Dr. Nitesh Kumar sir. Thank you, sir. Now we have a small tea break for 10 minutes, following which we would request all the delegates to kindly move to Department of Pharmacology, 3rd floor, for various sessions to be conducted sequentially in the pharmacy, CAL, demo, and skill lab. All the participants have already been assigned some group number, and you will be rotated sequentially in all the four groups. So, thank you.
Recording stopped.
एक्ट और इससे फॉर्म बी से संबंधित अगर कुछ आप लोगों को लग रहा है तो तब तक डिस्कस कर लेते हैं, या कुछ इस तरह से एनिमल एक्सपेरिमेंटेशन में कुछ आप लोग को लगता है जानना चाहिए तो उसमें डिस्कस कर लेते हैं हम लोग। फॉर्म बी आप लोग को कंप्लीट समझ में आ गया है, क्योंकि फॉर्म बी जो है वो एनिमल रिसर्च का बहुत ही मतलब बेसिक और इम्पोर्टेंट चीज़ है। पहले क्या था, जो इथिक्स में प्रेजेंटेशन के लिए पीआई को बुलाया जाता था, अब प्रेजेंटेशन के लिए नहीं, आपके फॉर्म बी के आधार से ही वो जज कर लिया जाएगा जिसको अप्रूव करना है या रिजेक्ट करना है। तो फॉर्म बी प्रॉपरली भरा होना चाहिए। तो इसके लिए क्या है, जैसे अभी डिस्कशन में भी आप लोगों ने देखा होगा, एनिमल मॉडल सेलेक्ट करना बहुत ही बेसिक और इम्पोर्टेंट स्टेप है। जो आपकी क्या स्टडी है उससे सिमिलर मॉडल लेना है, ठीक है। आ गए हैं सारे आदमी, आइए मतलब
स्टेशन 1 से स्टेशन 2 मूव कर जाएंगे।
ओके ओके। Good afternoon. So basically in practical session we will see the how many types of laboratory animals currently used and what are the differences, how we will identify their sex differentiation, how we will identify male or female in each species. So most of the animal research is done on mouse model. Most commonly 70 to 80% of the animal research is conducted on the mouse. In mouse there are so many outbreed strain and inbreed strain. Outbreed means they have mother or father of that will be from different genotype, so heterozygosity will be more. But when we are thinking about this immunological studies or cancer research, we have to procure the animal inbreed strain because so many homozygosity between this, so our result will be better. In our lab in Central Animal House, this is albino mice. In albino, this is the bulb C strain. There are two albino strain are there. One is bulb C, another one is Swiss albino. Okay. So as you see in that presentation, this is water bottle, and water bottle having the nipples. From there you will suck the water, and for feed we have to provide the feed pellet. This is also very important because these two animals comes under the family rodent, and rodent words arise from the rhodri, Greek word rhodri. Rhodri means gnaw, because this animal having the peculiarities that incisor, one pair of incisor that is continuously growing. Throughout the life incisor will grow, that is hypsodontic type of teeth. So that's why this animal regularly gnaw, it means teeth ring, to maintain his teeth in balance. That's why we are providing this pellet feed. Otherwise when we are giving the soft feed, incisor will grow. To maintain the incisor in proper shape, we must have to supply in feed pellet, hard form, with proper nutrition in balanced, what is called balanced nutrition. So this case is Why we are using nowadays this type of case, transparent case? Because one of the important factors what we have discussed is enrichment. Enrichment means one enrichment is food, space, this bedding, everything comes under the enrichment. We have to provide as much as possible the good environment to the animals, so our data will be correct. Otherwise there is so many physiological differences. When animal in a stress, all the parameters will be changed. So in this bulb C mice, how will differentiate in these two, bulb C and rat, mouse and rat? This is based on this is the body length and this is the tail length. So tail length is greater in case of mice. As Mehdi sir has asked, their basic difference in rat and mice is body length and tail length ratio. We must have to see the tail length is greater in case of mice. Okay. So next is the tail of rat and mice. What is the difference? The tail of the mice having the hair, you can zoom it here, the presence of hair throughout the tail, while in case of rat we will not find any hairs. So these are the two basic differences in rat and mouse. Next is coming to the sexing. In both cases we have to see anogenital distance. Anogenital distance means anus to genitalia. We have to see the distance. If distance is less, that is female. If distance is more, that is male. In neonatal or when it becomes the adult, then we have to see the testicles or we can see the mammary gland, that is easy. But in neonates, we are able to see these two difference. Apart from this anogenital distance, see in this case, zoom it. See, this is a female. In female, there is clear hairless line is there. That is known as pubic line. Okay, this line is present only in case of female. So based on this we can identify whether this is the male or female. So now see this is male because anus and genitalia distance is more and there is no pubic line. All the portions is covered with the hairs. So based on this we can differentiate this is the male or female, because this is one minute. Now this becomes keep. So now because of that much stress, and we will already discussed, noise level is more than 85 decibel. So in animal house we have to keep silence. That's why the animals becomes more in stress, in aggression. That's why they are jumping. And one of the important behavior of this bulb C male. So in case of this is the V star strain. In V star this is the albino V star. So when animal are in stress, they will in this rat also same anus and genitalia distance. This is the male. Testes are already visible. So in these two species, the sexing will be done based on the anogenital distance. Another is pubic line, hairless part present in that. Now another important thing, when we are deciding any experiment, we have to take the weight of that animal. Sex, weight, as in you can see in form B, we have to mention whether what is the strain, what is the age, what is the sex, and why this animal model is needed. So like if we are planning to study any gallbladder carcinoma, can we use the rat? No, because we have to know which animal having which organ, because like if in lungs or liver, like if you study ECG pattern, we have to take hamster, because the ECG pattern is more similarly hamster. So based on this, if we are going for reproductive study in higher species, we have to take consideration of monkey. In India, bonnet monkey is most common, because similarity to the human beings. Nowadays in mouse model, because the 99% gene sequence is known, with the help of the CRISPR-Cas9, we are able to design the whole genome sequencing and any disease model. Like in I think 6 months back, one boy of 3 years old had treated with this one CRISPR-Cas9 technology, and they have survived I think more than 8 months. Now we have to think about this. As per guideline, we have to maintain the animal in healthy position. So first we have to know what is the healthy animal. When we are called this is healthy or this is unhealthy. For healthy, they have the normal behavior of grooming. I have already told. Another is hair coat will be full shine, means there will be full combing like behavior they will show. And another thing is in mouse, grooming behavior. They must have habit of grooming each other, means like just like combing. So when you go in morning to the animal house to see your animals, you will find, oh, this animal having isolated in corner. Uniformly distribution is there in cage. If animal comes in one corner, means there is some problem is there. You have to identify whether this is relative humidity or temperature problem or air changes problem, like I have spoken there, means ammonia level. If ammonia level is more, the porphyrin secretion rate pigment is more common seen in rat, but that secretion increases when animal are in stress condition. So we have to look that situation in animal house. Another is, if you are grouping animals after adult, like 3-4 males in one cage, and again you are separated, one males in another group, one males in another group, there must be chances of fighting each other. So you have to avoid this, and if it is necessary, then we have to put some old bedding material into that cage, because they have the well developed smell. So he will understand, oh, this is from our colony only. So in this species, they will mark their territory, male will mark their territory with a taurine secretion. So he will recognize, oh, we are from the same colony. Otherwise they will fight each other, and in morning you will see no animal is there, both are. So tail injury, most of the cases tail bites are there. So tail injury is most common. Apart from this, sometimes you will see there is hairless area appears on the ears. This is due to excessive barbering, due to some changes in animal house, either ammonia level or either something else. This behavior are most common in mice rather than rat. So most of the cases in pharmaceutical research, they are using the SD rat. Do you know SD rat? One of the strain is Aspergillus. SD rat because they are little bit less aggressive than the Wistar rat. So people are using SD rat. These rats are outbreed strain, similar to like whatever we have explained in this one, mice. One more important thing is when you go in the next session, he will demonstrate all the drug administration techniques. In intraperitoneal gavage, as already because Mehdi sir has told, when you are planning any studies, you have to see the further implication in the human beings. Based on that, if you are using that drug in oral route, you have to try in animals in oral route. Because of that only, the translational studies will be less. Because so many times, because it is easy to interpret, if you are giving intraperitoneally, how much drug will reach to the blood-brain barrier, how much go to the ovary, how much go to the testicles, we don't know. That's why when we are planning any study, we have to go through the literature, and based on that, we have to decide. Another thing is, never start a study before you are giving the acclimatization period in your lab. When you are procuring animals from any other labs, you have to keep in acclimatization. Because these animals are kept in animal house, as you see, so many hesitation are there in animals, aggression are there. Because of that change in palace, we have to keep for at least 7 days. Before 7 days, or otherwise we have to keep for 14 days in acclimatization. Then only we have to start. And so many times people will not know from where, which parts we have to take the blood, what is the volume of that blood. In form B, you see, you have to write how much volume, and volume wise the site will be different. In case of mouse, if you are taking less blood, 0.1 ml blood, then you have to take from the tail vein. But when you have to take more blood, greater than 0.1 ml, means more than 10 microliter, then you have to go for retroorbital. For this, you have to use anesthesia. And in anesthesia, you have to go to the guideline of CCSCA. Which type of anesthesia is there? Which type of experiment you are performing? If you are performing the experiment for longer duration, then you have to use the inhalation anesthesia. Most commonly is isoflurane. Most of the lab using. For induction 3 to 5% and for maintenance 1 to 2%. If you are your experiment for the only 30 minutes or 20 minutes, then you have to go for ketamine, gelagin or ketamine, gelagin both cocktail. So based on this, in form B, you have to be very cautious to select the animal models. First you know all the behaviors, normal behaviors. Then you have to decide which type of study, which animal is selected. And one important thing is, in case of rabbit, in these cases we have similar anogenital. This rat, mouse and hamster, only anogenital distance is there. While in case of guinea pig, then you will see, you have to see the Y-shaped genitalia. And in rabbit, when you push, the penis will project, otherwise both you will look like similar. Similar to blood collection, you have to take from the ear vein in case of rabbit. In case of rabbit, we have to take blood from ear vein. The marginal ear vein is well developed in case of rabbit. So we have to take blood from the rabbit ear vein, not from the tail. Okay, like when we are using, we know that atropine esterase is secreted in rabbit. So never we go for that type of studies. So every species have certain criteria. Like if we are going for this osteoarthritis, we have to use higher species, because when you are going for a stem cell therapy, we don't get that much concentration of the bone marrow from rat and mouse. So depend upon your study, you have to think and then you have to implement in your research. One or two more things in this. How we have to handle this species. For animal handling, first this is the tail. So pick the base of the tail. If you pick from here, it may cause injury. So take from here and see when animal is just pick from this and like this. Okay. So when you are giving gavhaj also, you have to take the distance from this gymphoid cartilage to the mouth. That much length only. Otherwise it will pierce the stomach. Another important thing, when you are going for the intraperitoneal, you have to see the inguinal region. This is the umbilicus. If you closely visible, the skin pattern is divided into two parts. This is the umbilicus. So below the umbilicus, left or right, you have to use this. And another methods mostly used in European labs, like this. If you are not able to handle by this, for intraperitoneal, because in this also the intestinal content will be pushed lower, so you will easily inject the intraperitoneal injection. Thank you. If any questions, you can ask. See, this, you have got this manual. Now this manual will be given to you. See, like BCS score, okay, body condition scores, and Grimmsey scales. Body condition scores, through that you can easily assess the condition of whether your animal is healthy or not, whether this is obese. Obese is also not good, or emaciated is also not good. So we have to take the animals with 3 scores. Okay, similar to that, to identify the disease conditions, we have to use the Grimmsey scale. That scale tells us that when animal is in unhealthy condition, their whiskers backward direction. Normally you have to see the whiskers, here is there, tactile hairs is there, so that will be forward projected, but in stress that will be backward projected. And ears also becomes the backward direction erect. So based on this we can judge whether our animal is healthy or unhealthy. Like injury, I have always, because that in our facility nowadays there is no healthy animals, so you can see in photographs so many bite injury is there. So to avoid this, we have to use the bedding materials, old bedding materials into the new cages, so that they can smell and think like that, oh, we are from the same environment. Okay, thank you.
2 मिनट आप पहले यहाँ.
कोई क्वेश्चन है तो
Thank you, sir, for this wonderful session. Now, I'd like to request Dr. Hitesh Mishra, sir, to please felicitate Dr. Manoj Kumar, sir.
ये लोग को इंटरेक्ट नहीं किए हैं. You have to interact.
Thank you.
Sir, please felicitate Dr. Basra.
ये भी 1.25 लाख का ग्रांट. 1.25 लाख का 1 लाख का. अच्छा, 1 लाख का ग्रांट. कांग्रेस.
Thank you, everyone. Now we can move forward for next session.
Yes.
Next one. आप लोग से आप पूछ रहे हो दूसरे से. कैमरा से शूट करेगा.
यही वाला लेके जा रहे.
यही वाला लेके जा. अरे वही बोल रहा. बुलाने आप स्टैंड लिए. चलो. HDMI.
Good afternoon. I will discuss about the what is animal handling and what are the different routes of drug administration we are using in the biomedical research. Apart from this, I will also demonstrate the how we should withdraw blood from the different sites in the rats. Coming to the animal handling, as we since the morning we have listened that.
वहाँ नहीं दिख रहा.
है इसमें.
नहीं वहाँ नहीं दिख रहा.
अच्छा. नहीं आ रहा है. Since morning we are listening that the animal is very important and we cannot replace the animal. That's why the animal handling is a very you can say foundation of our biomedical research. Proper animal handling is very necessary, otherwise we will not get the desirable result, and that result cannot be replicable. What is animal handling? It is a terms in which describes how a person works with, respond to and interact with animal within their surroundings. The use of appropriate skilled handling is essential to ensure that animal readily accept you, actively seeks human contact, and procedure we are carrying out efficiently. Bad animal handling is likely to develop anxiety and which can amplify the stress among them. That's why animals should be approached in the relaxed and confident manner, and it should be regularly handled so that they can reduce their stress. With the proper handling we will get reliable results which can be replicable by the other people or scientists also. With the bad handling we will not get reliable results, and that result cannot be replicable because that can induce stress among them, due to which you can say that the protein or the enzymes which we would like to get expressed, apart from that during the stress condition, that other enzyme can be also expressed. These are the methods for the animal handling. This is the restrainer which is used for holding the animals. By the scuffing method, in this case the loose skin at the back of the neck we hold and we use for the dosing or and during the animal handling also. This by this scuffing method we can get exposed abdomen part. This is the V-shaped holding, and this is the cupping method. These are the methods for the handling the animals. It can be used for the rat as well as the mouse. There are different routes of drug administration, and dosing methodology is important during planning or execution of experiments, and during planning our experiment we consider what is the pharmacokinetics of our therapeutic agent or drug. On the basis of this we decide that which route of administration is required. The drug which cannot be administered by the oral route, then we cannot select the oral administration. In that case other route of administration can be selected. On the basis of the route of administration, volume is selected. What could be the maximum volume we should administer to the rodents or animals. Frequency of administration is also depends on the dosing, and obviously the stability of the substance which we are going to administer to the rats, it also determines the dosing. These are the different routes of drug administration: oral, commonly used; parenteral, in this case the IV, IM, IP, intraperitoneal, subcutaneous is used, and others are topical, rectal and intrauterine. In the rodents, oral, IP and SC is commonly used in the most of the cases. What is the oral administration? Oral route is very convenient and relatively safe, and both solid and liquid form of the drugs can be used by this route, and with the help of the metal gauze we do the oral administration or oral drug administration. In this case, by the left side of the animal's mouth, we directly put our gauze along the hard palate to the mouth and and insert it toward the back of the throat. This is a small video you can see here. This is the Vistar albino rats, and with the help of the tail we always hold we take the desired amount of the volume which is recommended in our in our study. You can see here, if there is any bubble or something, then we should remove it. And one thing is also very important: our gauze should not be the smaller one, otherwise the drug or the therapeutic agent will come out. and always the curved gauze is used. With the, you can see here from the left side, this was the left side of the rat, and gently I removed the metal gauze. Recommended gauze for the rat it is a 20 to 16 gauze is recommended, and the less than 10 ml per kg is recommended. For if we have a rat of 250 gram, then in that case the maximum volume would be the 2.5 ml only. If we have a mouse, then in that case the 24 to 20 gauze is recommended, and in this case also the volume should be the less than 10 ml per kg. If we have a mouse of 25 gram, then in that case 250 microliter is recommended, not more than that. This type of metal gauze is recommended. It may happen that our supplier will provide this type of gauze, but the state is not recommended, only the curved one is recommended. In this case the common complication could be that it may cause the damage to the esophagus, and the drug will go into the trachea, which may cause the casualty or death. It may cause death. That's why the careful and gentle passage of the gauze can be used, so that it can reduce this issue. Now coming to the intraperitoneal administration, this is the very common method to introduce the small drug into the small animals. By this route only the sterile drug is used between the, and this in this case the drug is administered into the peritoneal cavity, and animal is held in such a way that the abdominal portion is accessible with the needle, and the animal should remain immobile in that case, and the plunger of syringe is pulled back to check the whether the location is appropriate or not. In that case we will feel negative pressure when our needle will be the IP, intraperitoneal cavity. This is the tuberculin syringe which we commonly use for the IP, and this is the site, or you can say it is the, if we divide the abdomen into the 4 quadrant, then we always select for the right handed person fourth lower part abdomen quadrant, and the maximum recommended volume is the 2 to 4 ml for the rats and the 2 ml for the mouse. On the basis of the weight it may vary. This is a small video. This is the 4th quadrant of the abdomen. You can see here I am checking whether we are feeling the negative pressure or not. By pulling the syringe. Coming to the subcutaneous administration. In this case, the drugs which are highly active are expected to get slowly are administered by this route. A small volume of drug can be administered only. It may 200 to 300 microliter only. For injecting the drug below the skin, a fold is created in the skin by pressing it between the thumb and the forefinger of one hand, and the needle is inserted at the base of at 45 degrees centigrade. You can see there degree. By putting our syringe like this, we always give the subcutaneous, and preferable site for the this SC is between the 2 ears or the dorsal side near the spinal cord. This is the site. Here we can see here between the 2 ears we can fold the skin with our finger, and we can inject the administered desired drug. This is a small video. By holding the skin and we administer the drug, and this is the dorsal side. In this case the 2 person is required. One will hold the animal. You can see here this is the dorsal side near the near the spinal cord. We gently administer the drug. Blood collection in rodents. from the even morning, it has been asked several times, or we have been recommended that all the procedure should be approved by the IAC, because it regulates or it recommends that which procedure we should follow or not. That's why the blood collection method should be approved by the IAC, and the method should be least painful and stressful. Blood collections should be done under the anesthesia only, and these are the factors which determine what would be the appropriate blood volume and the technique while collecting the blood sample. It depends on the species of the animal, whether it is a rat or mouse. Size of the animals is a important factor. Age of the animal, frequency of the sampling, and obviously the training and the experience person is required for this method, and suitability of the anesthesia. It depends on your protocol which anesthesia we should use our study. Recommended volume, it has been recommended that the 10% of the total circulatory volume can be withdrawn in one occasion, or you can say the 1% of the animal body weight is recommended. If we have a 25 gram mouse, then in that case the 250 microliter is only recommended, not more than that. If we have a rat of 200 gram, then and we have to withdraw the blood sample at the point of time within the 14 days. In that case, only the 1.5% body weight we can withdraw the sample. For example, if we have a rat of 200 gram, then in that case 1.5%, it means that the 300 microliter or 3 ml is recommended, not more than that. We should only withdraw the blood 300 ml. These are the common site for the blood collection: tail vein or vital sinus, jugular vein, temporary cannulation can be done, blood vessel cannulation, tarsa vein, marginal ear, which is commonly used in the rabbit. For the blood collection from the tail vein, obviously anesthesia is used. We should dip the tail into the warm water about the 40 degrees centigrade, and with the help of the 1 ml tuber clean tube, we always withdraw the blood sample. And this is a dorsal site, dorsal vein, lateral vein, and this lateral vein. These three sites can be used for the blood collection from the tail vein. Yeah, you can see here I am using the dorsal vein here. This is a short video. From the dorsal vein, I am trying to withdraw the blood here. If you need the less volume of blood sample, then in that case tail vein is required. Otherwise, if you need the more volume, it means that the 1 ml or 500 microliter, then in that case a retroorbital plexus or retrosinus is required. Here this is the site behind the eye, retroorbital sinus is there, which with the help of the small capillary tube we withdraw the blood. By a fine capillary is inserted by rotating it. Once the plexus is punctured, blood will come out, and the desired amount of or desired volume is collected in the tube. And after the withdrawing the blood sample, we should wipe that area with the sterile cotton and should press against the eye, so that the blood flow will stop. In this case, caution should be taken that a repeated blood sample is not recommended in this method. Obviously skill is required, otherwise the rat or mice may can lose their eye, and 2 weeks should be allowed between the tube bleeding. This is the small video. You can see that by rotating the capillary tube, I am inserting. As per our study protocol, we always collect the volume of the blood which we need. Okay, thank you. Please come forward so that I can demonstrate you.
आनंद पकड़ोगे ग्लव्स पहन लो
This is the re-stainer. This is the re-stainer. On the basis of the body weight, we select this re-stainer to keep our mouse or rat. It is for the 200 gram to 300 gram. Our rat is here about 100. That's why it is too large as compared to this animal. This is the cupping method. Okay, just like by this way we can hold our animal. This is the scruffing method. Yeah, and by this we can get exposed our this abdomen part, and during the IP, this is the abdominal part. We divide just 4 quadrants. Yeah, first, second, third, and fourth. For the right hander, it is very convenient side. Fourth and just beside the near the mid this region. Okay. And for the if you are a lefty, then in that case you can also use this one. Yeah, but it should be the lower, not upper side. Just it is for the demonstration. You can hold this animal also. Yep. Base, yeah.
काट नहीं दे काटेगा
आराम से
काट लेगा
तो टेस्टर टेल का पकड़ो एकदम तुम ये हाँ थोड़ा सा
काट लेगा
बस
ज़्यादा देर तक इसको रखना नहीं है ऐसे इसके लिए एक सजा है
जेंटली टच करो हैंडल विथ केयर
पकड़ो
हाँ हाँ चलो
हाँ ठीक है उसी स्क्रपिंग में you can see here with the help of this I can give the drug orally. Yeah, this is the left side. Just you can see it is too long for this. Okay. If you feel resistant, then in that case we should not insert. It may cause damage to the esophagus. Okay. And for the SC, we can use this side. Here, this is the dorsal side. We pull this in like this way, and on the base of this fold here. Okay. Or most convenient if you don't have any assistance or any other person, then for singly you can use this side also. Okay, this like this way by holding the skin. And obviously for the blood collection, this is the dorsal vein. You can see here. This is the dorsal vein and this is the lateral vein, both side. In this case, this is the dorsal vein, है ना? This is the lateral vein. 3 sides can be used for the blood collection, and obviously this is the capillary tube which we use for the blood collection. This is the sterile one. Under the light anesthesia, we will keep our animals like this way. On the 90 degree, we will insert the capillary tube by rotating this one. question, please? Yeah, you can hold.
What if capillary breaks inside the sinus?
It will not break. If you are expert or if you have expertise, that's why expertise is required. Otherwise, they can lose their eye. That's why if you are not expert, in the form B, you will see there is a name of PI or expertise is required. They are asking the how many years do you have experience. If we don't have experience in the rodents, then in that case IC will not approve that study protocol. It is necessary that you have a expertise.
And why ventral tail bend is not is due to convenience or any other
reason? Convenience. Blood will not come out because in that case now only 50 microliter, not more than that we can withdraw. For the OTT, for example, or insulin tolerance test, it is a period of 120 minute. On the every 30 minutes we need the blood sample. Then in that case, retroorbital plexus will not help us. In that case, we always use tailwind.
My question is,
yeah.
Why dorsal bend? Why not ventral bend? Ventral tail bend.
Because on accessibility is very poor. Yeah. And we in that case, we will not get any sufficient amount of blood.
Okay.
Both is the reason. Because by using this vein, we get only 50 microliter, not more than 50 microliter, 25 microliter, 10 microliter sample only. And the any question, please? ओरल हो गया आपका, IPSC हो गया, 3 कन्वेंटली वी यूज फॉर आवर स्टडी पीरियड, स्टडी प्रोटोकॉल.
सर वो गोज जो दे रहे हैं कैसे पता चलेगा कि वो मतलब स्टोमक मतलब ओसोफेगस में नहीं जाके वो मतलब ओसोफेगस में जाकर
हाँ, जब आप दे रहे हैं ना, इंसर्ट कर रहे हैं तो आपको रेसिस्टेंट नहीं महसूस होना चाहिए. एकदम जेंटली चला जाएगा. यदि जेंटली जा रहा है तो ओसोफेगस में, अदरवाइज थोड़ा भी रेसिस्टेंट हुआ तब हमें कोशिश नहीं करना चाहिए. हाँ, तब बाहर निकालिए आप. और इसको सबसे इसीलिए मैं बोल रहा हूँ, मतलब ऐसा कोई साइंस नहीं है कि उसे, लेकिन जब आप लेफ्ट साइड से डालेंगे ना, ये प्रैक्टिस की बात है, तो आराम से ये पूरा विदाउट रेसिस्टेंट इट विल गो इनटू ओसोफेगस. इफ यू आर फीलिंग एनी रेसिस्टेंट देन प्लीज डोंट इंसर्ट.
सर लेफ्ट साइड से सिलेक्ट किया है, इजीली जा सकता है.
इजीली जा सकता है. हाँ,
लेफ्ट साइड से सिलेक्ट करते हैं
इसलिए. जस्ट एक दिन में आएगा नहीं, धीरे धीरे प्रैक्टिस करने पे ये सब चीज़ें. ओके ओके, थैंक यू थैंक यू.
Please have a seat. Thank you, sir, for this wonderful session. So now I'd like to request Professor Dr. Manish sir to please felicitate Dr. Patel sir.
Thank you, sir. Thank you, sir.
I request sir to please felicitate Dr. Amrendra Raja sir. I request sir to please felicitate Dr. Dewey.
इधर.
Thank you, sir.
Thank
now
you.
You can move forward for the next session.
So you all are PGs here, right? Have you handled faculty? The PG, okay. So have you handled the animal experimentations, like you know how to handle the animals, rat and mice? So basically we'll be here, we're going to demonstrate the screening of anti-inflammatory drugs. And so you know this, are you familiar with this? Okay.
जो शेयर करके चलाइएगा ना सर, वो कभी ऑनलाइन लॉग पीपीटी शेयर कर। नेट है? हाँ नेट है।
Join.
हाँ join करें सर।
ID concept 874 19 1777 48 do not connect audio, turn off my video, nahi join. Password 527 163.
शेयर स्क्रीन किया ना?
एक मिनट
नहीं
अब क्या करना है?
Recording in progress.
हाँ, इसमें वो भी दिखाई देगा ना?
हाँ।
क्या नाम है? इसमें वो भी दिखाई देगा ना? इसमें वो भी दिखाई देगा ना? क्या रहा है?
चालू है?
इसमें वो भी दिखाई देगा ना?
बंद कर दीजिए ना।
इसमें वो भी दिखाई देगा ना?
बंद है। एक मिनट रुको। स्क्रीन शेयर कीजिए। पीपीटी कौन सा? स्लाइड्स को कर दीजिए।
हटाना
रिपीट हो रहा है
तो आएगा ना?
यूट्यूब वाला प्रोजेक्टर में
So we're going to demonstrate here, we'll be demonstrating the carrageenan induced poiedema model in today's lecture. Here we'll be having the small demonstrations on carrageenan induced poiedema model. So before that demonstrations, you know, we will be discussing briefly about what is inflammations, different phases of inflammations, and then we'll see different models or inflammations, predominantly in vivo models, right? In vitro models are different. Whatever models we'll be discussing, that is for the in vivo models. And then we could only able to highlight or demonstrate the acute model, because as it is a times constraint, because we cannot show the subacute phase of inflammations because it take more than, I think, weeks, right? Acute phase we can assess for hours, subacute phase can be assessed within a weeks, and for chronic phases at least for the month. So it is not feasible. So first we will start with the introductions. What do we mean by inflammations? Inflammations is nothing but a pathophysiological local response of living tissue due to injury, due to any agents. It could be the physical trauma, it could be chemical related injuries, it could be the microbial invasions, it could be the radiation therapy. So because of that reason, that will be injuries. So inflammations is a major cause of acute as well as chronic debilitating disease, and it represents a major cause of morbidity in today's era modern lifestyle. So we have, based on the durations of actions, we have three inflammatory processes, that is acute phase, subacute phase, and chronic phase. As I told, in acute phase there will be for 1 hour, within an hour we can see you can appreciate the inflammations. If you inject, just one example is carrageenan induced poedema model, right? We'll see just after the insertions of carrageenan, you will get within hours, you can see the inflammatory release of inflammatory mediators, right? And then we can assess the percentage of inhibitions. Whereas subacute phase, it will resulted in at least for the week you have to subject the animals for the treatment, and then we can see the respective responses. And then chronic phase, we can see for the month. If you are treating the subjecting the animals with the months, we can see the different changes. So if you see the hemodynamic changes in case of acute phase, so there will be a local vasodilatation and increase the capillary permeabilities in case of acute phase. And subacute phase, there will be a infiltrations of leukocytes and phagocytic cells that will occur in the subacute phase. And in case of chronic phase, there will be a tissue degenerations and fibrosis occurs, right? Now coming to the inflammatory models, we have different inflammatory models. Predominantly we will be discussing about the in vivo models, right? So here I have highlighted certain inflammatory models. We will classified all the models in a 3 group. One is acute models, subacute model and chronic models. So here we can see acute model, that is UVB induced erythema model. Predominantly the animal species which you use, that is a guinea pig, and site of applications could be the depilated skin. Then we have carrageenan induced poedema model that we are going to demonstrate here today's class, right? Histamine, then histamine induced poedema, xylene induced erythema, then arachidonic acid induced erythema models, and we can see the different animal species going to use for this screening of these drugs. Then we have croton oil induced erythema, xylozone induced erythema, papaya latex induced erythema, pleural exudations, and these all models are common in the acute phase of inflammations. Predominantly जो सबसे ज़्यादा हम use करते हैं, we predominantly use for the carrageenan induced poedema is most commonly used acute acute drug for the screening of anti-inflammatory drugs, right? Now coming to the subacute phase of inflammations, we use the carrageenan induced granuloma pouch models, where we gonna use the rats and dorsal surface will be involved. Whereas formalin induced, this is the most commonly used formalin induced poedema model. We use the rats for this, and subplantar region will be involved for this subacute screening of phases of inflammations. Then we have chronic phase of inflammations for screening of anti-inflammatory drugs. So that most commonly used model is cotton pellet induced granuloma model. And mainly rat predominantly used for this screening. But apart from the rat also we can use the mice, because the size of rat will be bigger and we can appreciate the better tissues for the inflammation. So better if you have the rat, the best choice is the Western albino rat for this exercise. Then we also have the glass rod induced granuloma models, glass rod granuloma model. Rat and mice can be involved in this exercise. Now I will highlight 3 models, one from the acute, one from the subacute, one for the chronic models. Right. So from the acute model, carrageenan induced poedema model. What do you mean by carrageenan? What is carrageenan? Carrageenan is nothing but a phylogenetic agent. It is non-toxic, but the sugar containing sulfated sugar it is contain that mainly activate the complement system, and that complement system resulted in release of autocoids like histamine, serotonin and other mediators, and that need to be assessed, and because of that reason there will be edema. So we have to use the freshly prepared carrageenan, and that need to be subcutaneously inject in the plantar part of a rat paw. It could be right, it could be left, and then there will be swelling or edema formations, and that need to be assessed by the using the digital plethysmometer. This is the instrument called digital plethysmometer. Now what is the principle behind this inflammatory response? So you can see based on that the time you will inject. Just after the insertions of injections, you will get there is a release of histamine. That will be, you can see biphasic response, inflammatory response can be noted here. So initial phase, within an hour you will get, there will be a release of histamine and serotonin, right? And after that 3 to 5 hours there will be a release of PGs or prostaglandin, protease and lysosome, and this level can be estimated by the different markers. So instrument this called digital plethysmometer, right? Previously we used to use that manual one. Nowadays we have that because of the error and bias, we use the software. There is a 3-phase digital detector, then data acquisition system, and that data acquisition system is connected with a specific software present in the laptop or device. So whatever, suppose you inject the freshly prepared carrageenan subcutaneously in the plantar part of rat paw, and that resulted in formations of edema, and then you have to dip into this water level, and there will be displacement of the water, and that need to be detected by using the data acquisition system. And this data acquisition system transmit that data, whatever water has been displaced, that data will be transmitted to the software in your system. I'll show the interface of the software. So we cannot say it is bias. Previously, when we concern about the manual one, so we can able to temper the data, but here as there is an involvement of this software, so we cannot change whatever data I will get, that will be recorded into your software, right? And then apart from, so next is animal models we're gonna use. Animals I use SD mice, predominantly SD mice, and then we have Western albino rat. We can use that. SD mice also can be used in the hyperlipidemic, any screening model also, diabetic model also we can use, right? Alternative to the carrageenan, we have formalin. If you don't get the carrageenan, if you lack of carrageenan, then we can use the formalin, mustard oil, snake venom, dextran, and polyvinyl pyrrolidine, right? Now procedure, we can briefly will discuss. So you should have 3 group, right? Control, standard, and test sample. Control include the normal slime, the animal group, and n equal to 6. Minimum we have to take 6 rats to avoid the statistical bias. Then standard drug should be you can use diclofenac sodium also, you can use the normal indomethacins also, and test your test sample, whatever you are going to subject or unknown sample. Now before starting with the experiment, you have to overnight starving. You have to subject the animal for the overnight starving, because maybe there is a chances of food drug interactions. So better you starve the animal one prior night, and then divide into 3 groups: control, standard, and test. Then all drugs to be given 1 hour prior to the procedure. Suppose tomorrow morning, early morning, 10 o'clock you are going to start. So 9 o'clock you will be giving the all drugs by the oral route, right? Then one mark need to be made in the ankle joint of each rat, because just you don't know how to till which extent you are supposed to dip into the water, right? So to avoid that conditions, you have to mark with a marker. And then one point to be noted here, you have to prepare the freshly prepared carrageenan, right? Carrageenan need to be freshly prepared. Sometimes we do that mistake. Tomorrow you are going to start, so one day prior we will make, and tomorrow morning so it will be easy for me to just assess, right? So better you freshly prepare, and the dose include and concentration 0.05 ml of 1% carrageenan need to be injected subcutaneously in the plantar region of paw. It could be right or left, and then that need to be dipped into the water, and then there will be displacement. So you can see, you can appreciate from previous pictures I depict there without giving, just before giving the carrageenan, and this one right pictures I depicted the after the inflammations after injections of the carrageenan. So now the inflammations quantified in term of ml, that is water displacement, as if there will be swelling. So if you put that part in your leg part, inflamed paw into that water place, so there will be displacement of water that will be detected by the data acquisition system and transmitted to the software, right? Then evaluations, we can evaluate it by, so this is the time phase we can subject the entire experiment, 0 hour, and just after the insertions, just after the injections of the drug, then 3 hours, 6 hour, and 12 hour, right? And then with this formula, BC minus BT by VC into 100, with this formula we can assess the percentage of reduction or percentage of inhibitions. Both can be assessed from these pictures, right? Then we can see here, I will show you that software, this so So you can see here that this software, this is the interface for the software. So here, suppose sometime the people say you are tampering with the data. So you are gonna suppose first you will be injecting the drug carrageenan in the plantar region of rat. Then the soil leg need to be dipped. Then what you have to do? First you have to connect the software, this software with that this is software interface, right? So you have to fill this all, like department name, scholar name, your name will be there, date and timing. Two things will be very important here, because date, timing, because you have to use 0 hour, I think, 2 hour, 6 hour, 12 hour, 24 hour. So here also timing also they will be, you will get that in chart, you will get the timing also, exactly in the second. Then after dipping the inflamed leg, right, and then this animal weight is mandatory, weight is mandatory, and then you have to click onto the this site. नहीं cancel. Then you have to click after dipping the inflamed leg into the this water. You have to click when there is a changes occur in the digital acquisition system, data acquisition system. So if you click onto that, here that data will be recorded, and that data you can take a print also, print chart, and this you can add into a thesis also. This is authentic data. And okay, slide में ले जाना, slide. And you will get the data like this, right? You will get the data. You can see the name of your in charge or your guide. Then time is very important, because sometimes you get confused with the time. Manual instead of taking the manual report. And this you can put in your thesis also, this chart, right? And this calculation I already have explained. Right, now coming to the next model, that is formulate induced poedema model. Right, here the rat we're gonna that animal we're gonna use as a rat, Western lavender rat. The site of application is subplantar region, and then procedure, the same procedure we're gonna follow here. After you have to subject the animals for the overnight starvations, and then you have to make group of control standard and test. Then here 30-minute here all the drugs to be given 30 minutes prior to the procedures. They are 1 hour prior, here 30 minutes prior you have to give the procedure, and then 0.1 ml of 2% of formalin need to be injected in the right hind paw, and it need to be continued for 7 days. And after the 7 days, with a caliper we can calculate the thickness of the paw because of the inflammations. And then with this formula we can calculate the percentage of inhibitions, or you can calculate the data. So this is the subacute models of inflammation. So coming to the last model, this is a very famous model, that is cotton pellet induced granuloma model in case of chronic inflammations. What we are going to do is in this cotton pellet induced granuloma model is that we have to use the dry cotton pellet of weight 20 milligram, right? So that need to be subcutaneously implanted in the axilla and groin region of rat, right? So then you have to subject the animals for 7 days with the consecutive days with all drug, control, standard and test, and then after 8 days you have to sacrifice the animal and you have to take out the cotton pellet. So there we can see after the 7 days or 8 days, if you get the cotton pellet, so there will be a granuloma formation, tissue formations, freshly formed granuloma formations occur. So that we can take out from the cotton pellet, and that need to be, you can go for the histopathological analysis also by putting this freshly prepared tissue in a 10% formaldehyde, and also you can subtract from the previous one data, and we can this is the evaluation process. By using this formula we can calculate the percentage of inhibitions by WC minus WD by WC into 100. This is the reference, right? So as I told, there is three part of this digital potentiometer. One is recorder and data acquisition system, and this is a software, and then you have to inject the freshly prepared carrageenan subcutaneously in the plantar region of paw, and then need to be inflamed paw need to be dipped into the this water, and the whatever water displacement will occur, that need to be detected, and you have to interpret into your system, and that resulted in, you can take out the experiment, that resulted in complete explanations of this carrageenan induced paw edema model, right? So if you have any doubt, you can ask. Thank you.
Thank you, sir, for this session. Now I'd like to request Dr. Sunil Kumar sir to please felicitate Dr. Arthur Mandal.
Thank you, sir.
I request sir to please felicitate Dr. Trisha.
Thank you.
Thank you, sir. Thank you so much. We can move forward for the next session.
और भी है क्या? और भी है?
एक मैच और है सर, वो हम कर देंगे इसके बाद।
रोड को जस्ट ऑब्जर्व करते हैं यहाँ पे।
तीनों करना जरूरी है, नहीं तो आप कंपेयर कैसे करेंगे?
तो स्टैंडर्ड वाले का पर्पस क्या है मैसेंजर में?
स्टैंडर्ड तो आपको कम करेगा ना, ये कंपेयर कंट्रोल नॉर्मल स्लाइड, उससे कोई बायस
Okay, so good afternoon everyone. Myself Dr. Nitesh Kumar, working as Assistant Professor and Head in Department of Pharmacology and Toxicology, Naipur, Hajipur. A brief about Naipur, Hajipur is like, you can say AIMS and IIT is there for medical and you can say engineering side. So Government of India has made like those institute of national importance in pharmacy side, and we have one institution in Naipur, Hajipur out of 7 institutions in all over India. So this is my research credential where I have published more than 180 papers with the cumulative impact factor, book chapters, H index and other things are here. Based on this recognition, I got listed in top 2% scientists in Stanford Elsevier list, and I have received a funding of almost 3 crores and currently guiding almost 3 to 5 students and co-guiding others, and papers are in various area. So with this research brief I am here to present you about wound healing model. So coming to wound healing model is a model which have been tried from long time, and people are suffering from this from long time also. You know, sometimes the wound used to be so bad, people have to cut their amputation used to happen. The leg, hand, they used to remove it because the gangrene formation is there and they cannot cure it, so they have to cut the hand, they have to cut the fingers, they have to, you know, in diabetes they used to cut the digits also, one by one all the fingers are chopped off just because they don't want to allow the gangrene to spread in the body. So all those things used to say that we need to manage the wound, and for that research is going on from the long time, but still we don't have good medication for this purpose. For that reason, today we are discussing about few models which can help you to develop certain medication which may solve that problem. So the few models are there which are in the alternative side, that is in vitro model, and preclinical the main model, that is animal model. So these 2 different models I am going to discuss. For the preclinical animal model, we have 4 different type of condition. Sometimes the people used to have just one cut, that is known as incision wound model. Sometimes the skin is removed, gone, so that time that is we used to call as a excision wound model. Then sometimes the wound is deeper and inside there is some pockets and all are there, so that is known as dead space wound model. And next is like burn used to happen, because of burn also wound is there and not healing, so that is known as burn wound model. All these 4 conditions are working differently in different population. Normal people is fine, but for few people they are special, like people on the treatment of a steroid. So there the wound healing will not be faster, it will take time. So those conditions are happening because the steroids are given and not allowing the healing to happen, because you know all those barriers are suppressed, immunity is completely suppressed, and that healing will not be faster. Diabetes also, the wound healing will not be faster. So in those conditions also the same 4 model can be tried. Either use dexamethasone and then finally delay the wound healing, or induce the diabetes and then finally you try to develop this model. So here there are 2 different intention. You want to close the area, fine. So if minor incision is there, it will close it without a scar. That's why people used to have cut, they will run to hospital, get it sutured within 4 hours, 24 hours, so that the scar should not form. That is the second intention, that healing should happen without the scar formation. So a treatment you can develop where you can prevent the scar formation, that is fibrous tissue. So in both the cases there are 2 different type of cells which are coming into the picture. The first cell is because of, you can say major problem is epidermis or epithelial cell line. So epithelial tissue, that is keratinocyte, they are closing it and finally the scar is not forming. In the second one you can find out some granular tissues are also coming into the picture, that is fibroblast cell line, fibroblast. And these fibroblasts will cover the area, and then finally scar will form. So that is one problem. So you can, your intention might be to prevent this scar, so you don't want collagen to get stabilized, because once this scar will form, they will not go. The scars are forming because of injury everywhere in the body. Liver is getting problem, more cells are dying, finally they will form a scar, and that is known as liver cirrhosis, the irreversible condition. Similarly, the skin is having bigger wound, that will form the scar, irreversible condition. So these scars can be healing without scar, that can be the secondary intention, that allow the regrowth of epidermis, not allow maturation of collagen to happen too much. Then for that, the two different model, first one is alternative model, what used to be tried nowadays, that is in vitro study using two different cell line, fibroblast and keratinocyte. So you want to proliferate it. So if you will see these cells used to grow, most of the time epidermal cells, they will grow on the basal cell line, or you can say epithelial cells used to always grow on a support, that is basal cell line. Basal lamina used to be there, over that epithelial cells will grow. So here also keratinocytes can be grown in a plate, petri dish, and when you are growing on a petri dish, their surface is modified in such a way that it will give support to the cells, and the cells can grow on that surface. Fine. So the cells are growing, keratinocytes are growing, and they will cover the full area. Once they will be covering the full area, then finally they will not grow, because the cells have two different way to suppress their growth. If they will come in contact, they will not grow. If they are not in contact, they will grow. This contact inhibition used to happen. So they will form a monolayer, and once they are touching, that's what you can see, when the skin cells are growing, and then when they are coming closer, they will stop and a scab will form and the skin is proper. They are not, overgrowth is not there, it's closed. So that thing happen with the cells also, they will try to form one layer. After that, if something happen, that means the cells are going for a contamination phase, or you cannot use that cells. So we used to say that 90% of confluency, then you have to subculture it, otherwise the cells will die, or cells will go into the problem. So we are using those cells and making one layer, and after that we are scratching it. Once you are scratching, the cells on the borders will start growing, and they will try to fill the gap. Okay, and this model is known as scratch undase. So you are making a scratch and trying to see that how much time is taking to close. So this closure of the gap, that is one model what we are trying, that is the rate of epithelialization, you can assess by that, and then finally you can go for various mechanistic study. So that can be done by flow cytometer, Western blot, RT-PCR, so other things you can try over there for mechanistic study. So that's about the in vitro study, how much time the wound closure used to happen, that you are finding out in the cell line, and you are growing those cells. So you have the flexibility, you can go with the rat origin cells, you can go with the mouse origin cell, you can go with the human origin cells, you can select the cells. These cells are, human cells are immortalized cells. Immortalized means they are infected with the virus so that they can grow for multiple times. They will grow for longer time. Sometimes the cells can grow for years. There was one lady, Hela, she was suffering from cervical cancer, and some of the people, they removed the tissue and kept in the lab. They provided proper media, somehow it was growing. It was alive and the cells were growing. And that cells was used in that lab. Finally, it went to other lab through the colleague or through the request, and they started using for screening of anti-cancer drug. That started spreading from USA one lab, and now it's all over world. You can find out now each lab, if will be accumulate, it is in tons, not 1 ton. It is in tons. That much of cells are there in all over world. The Hela died long back. The cells are still alive. That is immortal cells because of cancer. Similarly, any healthy cells can be taken and through the infection of virus, they can be made immortalized. And because the cells, normally the cells, how many times it will grow? It will grow for 25 cycles. That is known as Hayflick's number. After that, it will go for aging because their telomere is shorter, it will fuse, it will induce apoptosis, and cells will die through apoptosis. So we are using cells from various sources. The sources are in India. If you want, you can take it from NCCS Pune, which will give you only for 4,000 rupees. One cells for 4,000, and then you can grow in your lab. But the same cells, if you are trying to go from ATCC, that is from USA, you have to pay something minimal 60,000, 70,000, because nowadays we have vendor here. Otherwise, we used to import it, and it used to cost around 3 lakh to 4 lakh rupees. The difference in those cells are the passage number. They are immortal, but with every passage, every passage of the time when you are subculturing it, there are certain changes in the morphology, certain changes in the function. So if you want fresh cells, people used to get it from ATCC. If you want some cells, they used to get it from NCCS. If you want cells, just cells, you are taking from certain labs, contacting them, I want the cells, they will provide you whatever passage number is there, they will mention and give it to you. So it will be a little longer and may not give a proper result sometime. That's about the in vitro studies, what can be tried. Now coming to in vivo studies, there are 4 different models as we have discussed. Cut, skin is removed, dead space is made, burn is induced, and these 4 models we will see one by one. The first one is excision wound model. So what is important point here, you have to cut the skin which will have epidermis, dermis, subcutaneous fat, all these things need to be removed. For removing that, you have to, what you have to do first, remove the fur, otherwise it will create problem. So first you are trimming the skin, removing the fur. Where to remove it? These are the animals, they can scratch. Whenever problem is there, they will not think that, okay, no, I should not touch it, otherwise it will create problem. They will just scratch it, and the wound will be bigger and bigger. So you have to make wound in certain places where they cannot touch it. Though after that also you will have problem. Sometimes if they have problem here, they will turn and they will try to rub it, that problem may happen. So you have to keep the bedding proper. The second thing is that you have to try here because they cannot touch it. Whatever they are moving their head or they are putting their hand, they cannot touch it. The second problem may happen if you are putting 2 animals together. I will scratch yours, you will scratch mine. So that problem will happen. So you have to keep it properly away from separate animal in different cages. Otherwise, the wound size will go on increasing. So that problem may happen. So you can use mice, rat, rabbit, pigs. The number of wounds you can decide. So you have to cut it. So how much size you want, that you have to decide. So you can decide the size. There are various biopsy punches. You can use those punch 1, 2, 3, 4, whatever size you want. So try those sizes, or you can use a ring, and with that ring make a circle. I had this size of ring only, so I will make this circle only. Fine, you make it, and as many circles you want, you make it and cut the skin and remove it. So whatever you are making, that should be proper, because you are making in one animal, you have to make in the other animal also with the same place, same location, with the same size, with the same condition. It should be uniform in all the animals. So you are making the wound, and after saving, making the wound, and then finally once the bleeding is stopped, you are putting in the cages, individual animal in one cage, and then finally you have to trace the wound. The day when you have made it, that day you have to trace it. So either you can use OHP sheet or you can use butter paper, and in the butter paper you can use permanent marker and circle it. So every 3rd day you have to make a circle, and by that you will find out, okay, the wound is getting closer, the the diameter is reducing, and there will be a day when it will form scab on the surface and it will fall. The day it will fall, that will be the end of your model. So what you are getting from this: hemorrhage, inflammation, granulation tissue formation, reepithelialization, angiogenesis, remodeling, percentage of wound contraction, percentage of epithelialization, and days by then the scab is falling, and then histopathology of skin you can go. So like that, what you are doing, you are making excision, then finally day 3 tracing it continuously. You can see the drug, it was in the diabetic animal, so the drug is producing more closure because they are controlling the diabetes, so the wound closure is happening. So you can see clivenclamide is not a drug for wound healing, but since they have controlled diabetes, so wound healing was faster. So both can be possible. So you have to decide your hypothesis how you want to go. So these are the two models. So first model is the excision one. You can see the granular tissue formation used to happen. So that's why histopathology, you can contact with the anatomy department. They will help you in making the histopathology. Incision, you can make the incision and then finally you can check how much was the epithelialization and granular tissue formation, that you can see by histopathology. Based on that you can compare also. Here each day when you are checking the diameter, you can find out percentage of wound contraction, that is initial wound size minus a specific day wound size divided by initial wound size into 100. So that will give you percentage of wound contraction. The next is the period of epithelialization, that is the falling of scab. The day the scab will fall, that is the period of epithelialization. Next one is incision wound model, where same thing, you are removing the fur, making the incision. Based on the size of animal you can decide the incision length, and once you are making the incision, again the same problem, you cannot make it direct on the center. If you are making on the center, then finally the wound will not get closed. So flank side, both the side of the ribs you are making one incision. You can see here, it's not on the center line, it is on the side. So on the side you are making the 2 lines you can make, and once you have made the cut, then finally you have to suture it. 7th day remove the sutures when you have seen that almost it is closed, then finally you are removing the sutures. Then you have to find out how much was the strength of the granular tissue, the strength of the wound. So that we can check it using a towel clamp. You can clamp it, or you can use the tooth forceps, or you can use something like scissors, which is blunt end, and lock is there. So with that you can lock the skin from both the sides, and one side you can make it static with certain pole or a stand, whatever stand is there in the back side, you can use those stand. And then finally next side you need a pulley. So this one will be tied with a thread and tied with one bottle. The bottle is empty, and put it over the pulley and fill the bottle. So as and when you are going to fill the bottle, the weight will increase, and that force will be applied on the skin. So the force which can break the skin, that will be the tensile strength. So the better drug will have more tensile strength, and normal should be lesser. That is the end point we are expecting, but what will come, that you have to see from your study. So this is one image where instrument setup is there. Traditionally most of the medical colleges have tried this. So animals are anesthetized, and sometimes they are it's the end of the study, so they are euthanized. Animals are euthanized. Both the sides they are tied, and with the pulley the water is filled and the bottom, and then finally the volume is measured. The time it will break, that will be the tensile strength of the wound closure. Next is the dead space model. You are making the incision, that is fine. After the incision you are making a suture. That is normal incision model. But here something more is done. You have to make a pocket. You are making a dead space. And in that space you have to put one polypropylene tube. That is, you can take a tip for the micropipette, whatever tips are there, Eppendorf tips are there. So that tips you can take it, and 2.5 centimeter size you can take it, which is same size, same diameter is there, uniform diameter. So those area you can cut it, sterilize it, put it in the pocket and close the wound. Once you have closed the wound, then finally you wait for 10th day. After 10th day, that what you have done in the earlier. So 10th day you are again checking the tensile strength. Once it is broken, your study is over, but you are taking out that polypropylene tube. Now that tube is completely covered with collagen. Fine. That what is, whatever space is there, that will not be empty, it will be covered by fibrous tissue. So the fibrous tissue will cover it completely. Those fibrous tissue also you can check the strength, that how much was the strength. That much collagen is there, that much strength will be there. So you can find out how much was the strength of that collagen which has covered that tube. So the tube which is covered, those tissue you can take it out, you can check the strength, and the next thing what you can check is 5-hydroxyproline, which will stabilize the collagen. Next is lysyl oxidase, which will help in the collagen synthesis. So this you can try in the dead space model. The last is the burn wound model. In the burn wound model, you have to create wound, and that wound you are creating by burning the skin. For that you can use either hot metal plate. You have to take one coin also, you can take it, a proper coin which you can use for all. Heat it, proper heating will be there, transferring also should be there for same for all. So you can place over the skin. And then next thing what you can try, you can take a ring also. In that you can pour the molten waxes, and then finally you can create similar type of things like you have removed the skins. Now here you are burning the skin. So the same area you can decide, that area, that much of damage you can create on the skin in the limited area. Now next what you have to do, again same thing, tracing the wound size and finding out that how the closure has happened and when the scab is falling. So same procedure will be followed for the burn wound model also, like excision wound model. That's all for these 4 models and in vitro model. Any question?
Thank you, sir, for this session. Now I request Dr. U.S. Chakradhar Rao to felicitate Dr. Nitesh Kumar.
Thank you, sir.
I request sir to please felicitate Dr. Bhavna Verma.
All the participants are requested to move for lunch.
All online participants, now this is, we have a break for lunch. We will continue this after lunch at 2:30.
हेलो, ठीक है, ठीक ठीक। इसको पैकेट में रख लेते हैं। स्क्रीन शेयर कैसे होगा यहाँ से?
स्क्रीन शेयर कर दीजिए ना सर, का वहाँ आ नहीं रहा।
हाँ, यहाँ से।
हाँ वहाँ ऑन है सर, वही।
अच्छा, ठीक ठीक ठीक।
इसको ऑन कर देते हैं।
देखिए यार, करंट वरंट ना लगे, है ना, ध्यान देना है थोड़ा सा। 9818906956।
एक मिनट रुक जाओ, एक मिनट। हाँ हेलो, आओ जी। हेलो, उसका कैप्चर कार्ड वगैरह
So in this session, basically we are going to demonstrate the anti-epileptic activity of different drugs using animal model of seizure or epilepsy. So let us start with what is seizure or epilepsy. So seizure or epilepsy, it is a paroxysmal event that is characterized by abnormal, excessive and hypersynchronous discharge from the aggregate of CNS neuron, right? So as we know that it is characterized by there may be loss of consciousness and also there may be some abnormal type of body movement. So basically it is caused whenever there is an imbalance in between the excitatory and the inhibitory neurons. As you can see here that in case of seizure, what happens that there is excessive excitatory activity, okay? So any drug that is capable of inhibiting the GABA-like substance, it will be ultimately responsible for causing seizure. This is the rationale behind using PTZ-induced seizure model, okay? So in case of PTZ-induced seizure model, we are using PTZ, which is a chemical substance that is acting by inhibiting the GABA and ultimately it is responsible for causing seizure, right? So as you can see here that different type of seizure activities are there. Some are partial, some are generalized. So there may be partial seizure, there may be absence seizure, there may be generalized tonic-clonic seizure. And we have a drug for all the different categories, right? So as you can see here that we are having different categories of drug for the different type of the seizure. Now the next question is that if we are having all these different drugs, then what is the need of all these animal models? Can anybody tell me just one line? That इस तरह के मॉडल की जरूरत क्या है जब हमारे पास सारे के सारे लगभग मतलब है सीजर आप देख रहे हो, इसके लिए अलग अलग कैटेगरीज के मेडिसिन हमारे पास अवेलेबल है, तो फिर हम इस तरह के मॉडल की बात क्यों करते हैं? ओके, लेकिन अभी तो अवेलेबल है ना, ठीक है? सो बेसिकली ओके, सो बेसिकली द मेन प्रॉब्लम इज द प्रॉब्लम लाइक acute side effect. So these are the common side effects that are very commonly encountered in the day-to-day practice. And also some side effects are there that can be seen when a particular drug it is used for a long duration of time, okay? So you can see that there is a problem of acute side effect, there is a problem of chronic side effect, and also some drug-resistant epilepsy are also there, right? Where the existing drugs, they are not capable of treating that particular condition, right? So these are the different problems due to which there is a need of newer anti-epileptic drugs. So we are constantly searching for such a molecule or such a drug that is having limited or the negligible side effect, and that too is capable of treating the different type of epilepsy, or you can say epilepsy like the pharmacoresistance epilepsy, right? So epileptic models, they may be either anti-epileptic drug testing, it can be done either by using in vitro model or by using in vivo model, right? In case of in vitro model, we are basically using tissues, right? We are using like hippocampal tissue, we can use neuronal tissue, and also different type of models are there in the category of the in vitro model, where we can test a newer molecule or a newer anti-epileptic drug testing can be done, okay? So the most important thing regarding the in vitro model is that we can find out that what is the exact mechanism of action, right? So basically this is the most important thing regarding the in vitro model, is that we can elucidate the mechanism of action of any particular drug for a particular type of epilepsy. So this is regarding the in vitro model, and this is the only most important thing regarding the in vitro model, is that we can find out the exact mechanism of action. Now coming to in vivo model. So in vivo model, we are having electroshock seizure type model, we are having chemical induced seizure model, we are having kindling rat seizure model, we are also having genetic rat model of epilepsy, okay? So basically what we are doing that we are simply inducing epilepsy, okay? So it can be induced either by giving electric shock, right? Or it can be induced by using different type of chemical substances. So any drug that is capable of inhibiting GABA or other substances, they are capable of inducing seizure-like activity. So it can be induced by using chemical substance in the form of PTZ. It can be induced by using strychnine, picrotoxin, biguculin, and all these different drugs. So this is regarding chemical induced seizure, where we are using chemical substance for inducing seizure. It can also be induced by giving a small amount of chemical substances in a daily dose form, or using a small amount of current. That is, in kindling rat model, what we are doing, that we are using a smaller amount of chemical substances or a smaller amount of electrical current to induce seizure, right? We are also having genetic rat model of epilepsy. So basically, genetic rat model of epilepsy, they are those rats that are very sensitive for the seizure, okay? So it can be induced very fastly and very rapidly in case of genetic rat model of epilepsy. So now the next thing is that why we need animal model, okay? So basically, so many things are clear to you that there is a need of newer anti-epileptic drug. So we can discover new anti-epileptic drug. We can also find out the spectrum of activity, that is the spectrum of anti-convulsant activity of the newer anti-epileptic drug by using these different models. And also we can find out that whether the efficacy of the newer anti-epileptic drug, it has changed over a period of time or not. जैसे कि आपने देखा है कि epilepsy या seizure वाले condition में कभी-कभी होता है कि लंबे समय तक medicine देना पड़ता है, okay? So basically, we can also find out that whether the efficacy of any molecule or any newly discovered molecule, it has changed over a period of time or not, okay? So that is why there is a need of animal model to get all these different answers. So what are the characteristic feature, or what are the features that should be present in ideal animal model? So basically, whatever drug testing we are carrying out, so it is meant only for the human use, right? So the first thing is that the nature of seizure that we are inducing in case of animal, it should be quite similar to that it is seen in the human being, right? Second thing is that, like the human being, there must be EEG correlation between the epileptical activity, okay? So EEG correlation, it is also a very important thing that is required for an ideal animal model. Now, so basically this is the usual approach to the anticonvulsant drug testing in animals. So first of all, what we will do that before inducing seizure, first of all we will use our newer drug, or you can say any anti-epileptical drug, right? So first of all, we will introduce the drug either by intraperitoneal route or by the oral route, and thereafter, after a period of 30 minutes, what we do is we induce seizure by using any of the different methods, that is either by using electrical stimulus method or by giving chemical substance, as for example PTJ inside the intraperitoneal cavity, and thereafter we notice that what are the changes that are taking place, okay? So these are the different parameters that can be assessed. So basically, after inducing seizure, what we do is that we looked at whether there has been any change in the threshold, whether there is a change in the EEG pattern, or whether there is a change in the incidence of seizure after using any newer anti-epileptical drug or the existing anti-epileptic drug, right? Now coming to maximum electroshock model. So the pioneer work in this type of model, it was done by the Toman and the Goodman. So the purpose of this test is basically to induce a very intense type of physiological type of seizure in the animal, that is a rat or the mice. So basically this model is quite similar to grand mal epilepsy that is seen in case of the human being, right? So after inducing seizure by using the electrical current, what we do is that we observe all the different stages, okay? So there may be no seizure activity, right? If the drug is very much effective in that condition, no seizure activity can be seen. Also there may be extension of the forelimb, okay, without involvement of the hind limb. There may be full extension of the forelimb accompanied by the partial extension of the hind limb. There may be complete tonic extension of the hind limb beyond 90 degree, and there may be post seizure, that is post ictical depression state, or in certain cases there may be death also, okay? Now just remember that what will be the end point? It is the hind limb, that is the tonic hind limb extension, it is supposed to be the end point of this type of seizure model. So the observation of drug effect, it can be measured in the form of prolongation of the latent phase, or there may be decrease in the duration of the tonic phase. So basically what we can do is that we can calculate the percentage protection, that is how much protection this particular or a newer antiepileptic drug is giving. Okay. So suppose we are using 10 animals, and out of 10 animals the number of animal with tonic hind limb extension is 10. In that condition it will become 100%. That is, your drug is providing 100% of protection. Okay. Seizures जब भी हम induce करते हैं, that is 100% seizure protection. We can also calculate the ED50. ठीक है. So the dose that protects about the 50% of the animal from tonic hind limb extension, it is supposed to be the ED50 for that particular drug, right? Now the advantage is that it is not a very time consuming process and there is a very good reproducibility. Okay, आपने IGIMS में किया, आपने AIMS में किया, तो basically the reproducibility of this type of experiment or this type of the model, it is very good. ठीक है. The disadvantage is that this is not an ideal model for the different types of seizure. यदि हम absence seizure की बात करते हैं तो फिर हम इससे उसका आंसर हम नहीं ले पाएंगे. Okay, so basically this is a fit model for the grand mal type of epilepsy. On the other hand, it is not suitable for the other different type of the seizure. Okay, and also the mechanism of action, it can only be determined by using in vitro testing. Okay, so by using in vivo we will not be able to get the exact mechanism of action. Now coming to PTZ. So regarding the PTZ model, the pioneer work, it was done by the Mason and the Kappa in the 1972 in the mice. So basically PTZ, it is a chemical substance, or you can say it is a tetrazool derivative, and the mechanism of action of this PTZ, it is by antagonizing GABA. ठीक है. तो GABA जो कि inhibitory substance है उसको inhibit कर दे रहा है. तो ultimately it is responsible for causing seizure, and the type of the model that we are getting after using PTZ, it is petit mal seizure or petit mal epilepsy. So basically these different phases, it may be noted after injecting PTZ inside the peritoneal cavity of the mice or rat. So there may be no activity, there may be hyperactivity, there may be myoclonic jerk, there may be for limb clonus, there may be generalized tonic-clonic seizure, or there may be hind limb tonic extension. Okay. Now similarly we can use different type of chemical substance for inducing seizure. So you can use a bicuculin that is supposed to be acting at the level of the GABA, that is by antagonizing GABA, it can precipitate seizure. So depending upon the nature of the chemical substance, we have to choose animal accordingly. Okay, so in some cases we have to choose SD variety of the rat in order to get the desired result. So depending upon the nature of the chemical substance, we will have to choose the nature of the animal. So it can be induced by using chemical substance like the strychnine that is acting at the level of the glycine and ultimately responsible for causing seizure. Similarly, picrotoxin is there that can be used for inducing seizure, and also we can use a pilocarpine, or we can use a combination of the lithium plus pilocarpine. Okay. So first of all, in case of lithium pilocarpine, first of all pre-treatment it is given with the lithium, and thereafter it is followed by the pilocarpine in the dose of 30 to 40 milligram per kg body weight intraperitoneally, right? So the conclusion is that there is not a single model that is capable of answering all the questions, that is the question of any specific type of the seizure. So we are having model for grand mal epilepsy, we are having model for absence seizure. So we will have to use different type of the model for the different type of the seizure activity. So this is the conclusion regarding this particular type of animal model. Now coming to demonstration of anticonvulsant activity of sodium valproate by MES model. तो MES model के बारे में just हमने discuss किया. तो basically these are the our requirements, that is it can be carried out with mice. The drug that can be used is sodium valproate. Sodium valproate, it is a well established anti-epileptic drug, and there is a syringe of 1 ml. The equipment is electroconvulsiometer, there is an electrode, and also there is a need of a stopwatch, right? So the procedure is that first of all we will divide our animal into different groups, that is each group it will contain 6 mice or rat. one important precaution that is very much important is that prior to starting experiment, what we will have to do is we will have to check that whether a particular mice or rat, it is showing tonic hind limb extension or not prior to performing this experiment. Okay? So only if an animal is showing tonic hind limb extension, in that condition, only those animals we will have to choose for this particular type of experiment. So you can see that we have divided our animals into different groups, that is A and B. Okay? So this type of division is important because it prevents mixing of the animals, right? So in case of group 1, we will simply give normal saline by the intraperitoneal route. In case of second group, we will give a well-established antiepileptic drug, that is sodium valproate, and the dose is 300 milligram per kg intraperitoneally. Okay? So basically this is the electroconvulsiometer. तो इसमें अलग अलग पॉइंट बने हुए हैं कि करंट हमें कितना में सेट करना है या फिर ड्यूरेशन कितना चाहिए. तो बेसिकली इट इज गिवन फॉर अ पीरियड ऑफ अबाउट 0.2 सेकेंड. ओके, तो इफ यू आर यूजिंग रैट the current will be different. In case of mice, the current will be different. Okay, so it is 50 milliampere for the mice and it is about 150 milliampere. It is given for the rat. So what we do is, you can see here that this is the earpiece. What we do is we simply put this earpiece on the pinna of the rat or the mice. ठीक है, ये हमारा instrument के साथ it is connected to this particular instrument. Okay, and thereafter by simply pressing this particular knob, a fixed amount of current for a fixed duration of time, it is delivered through the pinna of rat or the mice to the animal, and ultimately this one is responsible for causing seizure. ठीक है. So these are the different stages of convulsion. That is, there may not be any seizure activity, there may be extension of forelimb, there may be full extension of the forelimb, and the end point is tonic hind limb extension. That is supposed to be the end point of this type of the model. So you can see in this video that the earpiece, it has been applied to the pinna of the animal and a fixed amount of current has been delivered. So you can see here that the animal is passing through the different stages of seizure and ultimately there is extension of the hind limb. Okay. So the extension of hind limb, it is supposed to be the end point of this type of the seizure or MUS type of model. Okay. So thereafter what we will have to do is we will have to record the latency and the duration of tonic hind limb extension. तो हमने ये दो पैरामीटर को रिकॉर्ड कर लिया and we will have to make observation table like this, and thereafter we calculate it that whether there is any significant difference in between the different type of in the different categories of animals or not. Like group 1 and 2, we can also carry out this experiment in the different group of the animal, and thereafter by applying statistics we can find out that what is the actual significant difference or whether the result is significant or not. Okay. Now in case of the PTZ model, so it is usually carried out in case of rat because by injecting PTZ there is a violent type of the seizure. Okay. So in majority of the mice it may die. So we usually choose the rat for the PTZ induced seizure and the standard drug, that is sodium valproate. Like the previous experiment, we will divide our animal into different groups. That is, one will be given normal saline and second one it will be given a well established anti-epileptic drug like the sodium valproate drug in the dose of 300 milligram per kg IP. So after 30 minutes we can note the different stages of the seizure. That is, there may be no activity or there may be hind limb tonic extension depending on the nature of the drug. Okay. And by recording these activities or these stages, we can prepare observation table. So you can see in this that we have injected sodium valproate in the dose of 300 milligram per kg body weight, right, by the intraperitoneal route. Similarly, in case of So we will use two different class of the animal. In one group, we will simply inject a vehicle in the form of the sodium chloride or the NaCl. In the second group, we will inject sodium valproate, and thereafter a period of 30 minutes, we will observe that what are the changes that are visible. So in case of valproate and PTZ, as you know that valproate, it is a well established anti-epileptic drug, so it is going to provide a very good protection, that is even after injecting PTZ you will not be able to see any type of seizure activity, right. Now in case of vehicle treated animal, so this animal has only been given normal saline plus PTZ. So you can see here that those animals who are given only vehicle plus epileptogenic drug like the PTZ, in that condition you can see the different phases of the figure, different phases of the seizure, as you can see here. So the spectrum of the spectrum of seizure activity, it may varies from no seizure to intense seizure. Okay, so you can see here the tonic hind limb extension, it is not very much it is not very well seen in this particular case. Okay, so the spectrum of seizure activity, it may varies from no seizure activity to hind limb extension. It entirely depends upon the nature of the drug. Okay, so this you can see that in absence of any type of protection, PTZ, it is capable of inducing seizure. So ultimately, we will have to note down the different parameters, and thereafter we can prepare this type of observation table, and thereafter after applying statistics, we can find out that what is the possible significance level between the different group of animals. ठीक है? थैंक यू. यदि इंस्ट्रूमेंट को थैंक यू, आप इंस्ट्रूमेंट के बारे में प्लीज कम फॉरवर्ड.
Thank you everyone. So कुछ query है? नहीं.
इलेक्ट्रिकल एक्टिविटी रिकॉर्ड करने के लिए हमारे पास ऑप्शन है एनिमल में.
देखो जैसे क्या होता है, जैसे हम ह्यूमन बीइंग में करते हैं ना, तो फिर एनिमल के लिए भी हर तरह का इंस्ट्रूमेंट बना हुआ है. जैसे कि आपने एक देखा, जैसे ये तो इलेक्ट्रोकन्वल्सीमीटर है, तो उसी तरह से ईजी एक्टिविटी रिकॉर्ड करने के लिए भी इंस्ट्रूमेंट हमारे पास अवेलेबल है. इवन जो वेंटिलेशन हम जो देते हैं ना, उसके लिए भी इंस्ट्रूमेंट हमारे पास अवेलेबल है. जो कार्डियोवास्कुलर मतलब है, जो ड्रग के लिए हम टेस्टिंग करते हैं ना, तो उसके लिए मतलब है कि जो भी सपोर्टिव इंस्ट्रूमेंट होता है ना, वो सारे के सारे मतलब एनिमल के लिए भी अवेलेबल होते हैं. तो आप इससे भी मतलब है कि आप चेक कर सकते हो किस तरह की एक्टिविटीज हो कर रही. तो बेसिकली हमारा ये गोल क्या है कि जो भी हम ड्रग बना रहे हैं मतलब एनिमल के लिए थोड़ी बना रहे हैं, ह्यूमन बीइंग के लिए बना रहे हैं ना. ठीक है, तो बात है कि जिस तरह की एक्टिविटी है उस तरह की एक्टिविटी एनिमल में भी क्रिएट करना है आपको. उस मॉडल को आपको एनिमल में भी मतलब लाना है. जो भी एक्टिविटी हम मतलब ईजी एक्टिविटी ह्यूमन बीइंग में देखते हैं, यदि मान लो एक्टिविटी एनिमल में दिखाई देगा, तभी हम बोल रहे हैं कि ठीक है जो भी हम ड्रग टेस्ट कर रहे हैं वो मतलब हम ह्यूमन बीइंग में यूज़ कर सकते हैं. ठीक है, तो फिर एक बार इस इंस्ट्रूमेंट को देख लो. इसे दिस इज इलेक्ट्रोकन्वल्सीमीटर, और इसमें अलग अलग मतलब ये बना हुआ कितना करंट आपको देना है, क्योंकि मैंने बताया कि माइस चूज करोगे उसके लिए अलग है, रैट जो कि बड़ा होता है उसके लिए अलग होता है. कितना ड्यूरेशन देना है, ड्यूरेशन फिक्स करके रखना है. और सिंपली मतलब है इसको हम ऑन करेंगे ना, तो उतना अमाउंट या फिर उतने ड्यूरेशन के लिए जो भी करंट देना होता है ना, उस ड्यूरेशन में वहाँ पे वो चला जाएगा. इसके बाद फिर जो भी मतलब है चेंजेस आएंगे, एक्टिविटी आएगी, उसकी अकॉर्डिंगली एक्टिविटी आप देख पाओगे. ठीक है। तो एक्चुअली ये थोड़ा सा है, ये थोड़ा शार्प भी है, तो थोड़ा ध्यान देने वाली बात ये भी होती है, क्योंकि यदि इसको भी हम लगाएंगे तो डायरेक्ट मतलब है कि कट हो जाएगा। ओके। और मतलब इंपॉर्टेंट पॉइंट ये है कि जैसे हम इलेक्ट्रोकन्वल्सिव इंड्यूस जो सीजर इंड्यूस करते हैं ना, यूजुअली हम माइस में करते हैं, और पीटीजेड एक्चुअली क्या है ना, केमिकल सब्सटेंस है, बहुत ही वायलेंट किस्म का सीजर इंड्यूस करता है। तो बेसिकली हम कोशिश करते हैं उससे रैट या फिर बड़े रैट में ही हम यूज़ करें। इससे मतलब है कि जो भी मतलब है कि वायलेंट जो मेरी कंट्रैक्शन के चलते जो भी प्रॉब्लम हो सकती है ना, उस तरह की ज़्यादा प्रॉब्लम मतलब है ना आया उसमें, ठीक है। क्योंकि इसमें करोगे तो ज़्यादातर केस में तो डेथ ही हो जाता है। ओके, चलो।
ईजी रिकॉर्ड करने के लिए पटना में मैं भी फ्रेंड्स से हूँ।
हाँ हाँ
पाल सर इस्टेब्लिश कर रहे थे हमारे एनिमल में।
हाँ हाँ।
एक बीच में प्रोजेक्ट सर आपको पता होगा रुक गया, तो मैं प्लान कर रहा था कि मैं ईजी रिकॉर्ड करूँ एनिमल में। तो
अभी तो
जानना चाहता हूँ कि क्या यहाँ पर रिसोर्स अवेलेबल है पटना के आसपास।
ये तो जो भी है बाहर से ये करना पड़ेगा ना, दिल्ली में तो है सब कुछ। जैसे जो मैं सपोर्टिव इंस्ट्रूमेंट की बात कर रहा हूँ, जैसे मतलब वेंटिलेटर या फिर मतलब है कि जो भी ऑक्सीजनेट करना हो, तो सारे के सारे वहाँ पे हैं अवेलेबल। आप कौन से डिपार्टमेंट में हो?
इंटरनेशनल फिजियोलॉजी।
अच्छा अच्छा, चलो ठीक है, ओके, थैंक यू।
Thank you, sir. Now I request Professor Dr. Manish sir to please felicitate Dr. Sunil Kumar Singh.
Thank you.
And I request sir to please felicitate Dr. Ravi Roshan. Thank you. You can move for your next session.
चलो, थैंक यू वेरी मच।
ये वाले पॉकेट में मोबाइल से कोई दिक्कत नहीं। शुरू करें? Very good afternoon, everyone. Today I am going to discuss about the analgesic activity of drugs. Okay, so here we are discussing how the analgesic activity we will see in rodents. So first, before that, we will discuss about what is the analgesia and what are the analgesic we use for this experiment. Use for this experiment thermal method, that is Telflick method and Aegis hot plate method. Aegis hot plate is discovered by Aegis and Lambach in 1953 and modified then for this thing. And chemical method we use acetic acid method or bradykinin. And electrical method is earlier it was used in monkey but is not currently used. And in other method, tail immersion method also there, that we use as a chemical method. So we put the ethanol and put the immersion of tail inside that, and then we see the response, which we don't do here. And a physical method, we use a tail compression method. So we have seen that one rat tail catcher, so we compress the tail. So that also we can use, but main is today I am going to demonstrate that Aegis hot plate method. And so Aegis hot plate method, we will use Aegis hot plate, and here we see that hot plate is there, and the instrument we have to set the temperature of around 50 to 55 degrees centigrade, and then we will put the mice on the hot plate and then we see the reaction. So here we put the mice here on the hot plate and see the reaction time. So either mice will jump out, try to jump, or try to lick his paw. So we noted down that reaction time. So first we will take weight around 18 to 22 milligram weighing mice, and then we mark them properly. Marking is very important. We have to put in two different groups. One group is for control and one for test. So marking is very important. So we divide after that, we put the mice on the plate and we noted down the basal reaction time. So once we put it and then we see what the reaction time. More than 6 seconds is not considered. Normally reaction time response will come in between 6 seconds. Okay. So if it is more than 6 seconds, we exclude that mice and then we take other mice for this test. And we will give either subcutaneously or oral route. Mainly in chemical method acetic acid we give intraperitoneally, but here we will use subcutaneously or oral route. So earlier sir have demonstrated how we give orally. So 0.5 milligram per ml we calculate the dose and we administered the drug in the orally. And then we repeat the same procedure in between 15, 13, 60 minute and 90 minute, and we noted down the reaction time. And we have to set the cutoff time as 15 seconds, means after 15 seconds we won't record the response, because mice will get irritated or might get thermal injury also. So risk is there, so we just put it the cutoff time of 15 seconds. So this is a Aegis hot plate method, and we compare the value and we calculate the percentage time, reaction time, and in centrally acting analgesic we can evaluate the hot plate method, and whereas peripherally acting analgesic is not effective. So for centrally acting like morphine and pethidine, we will see this response in mainly Aegis hot plate method. So this is a video of a response. So you see that how the response is there. Mice started tried to jump out, tried to lick the paw. See the response we have to record and we have to note it down. मेनली इसमें है कि तुरंत रिस्पांस देखना है, ज़्यादा देर हमको वेट नहीं करना है, नहीं तो फिर रीडिंग जो है गलत आ जाएगा, इसलिए हम लोग जो है इसको तुरंत जो है ये आप लोगों को दिखाने के लिए हम लोग जो है ऑलरेडी करके वीडियो बना के रखें, बिकॉज वी डोंट वांट टू कीप दैट माउस रिपीट नहीं कर सकते, माउस को रिपीट हम लोग करेंगे तो इरिटेट होगा तो रिस्पांस सही ना आए, इसलिए क्योंकि डिफरेंट ग्रुप में जो है ऑलरेडी डिवाइड करके रखना होता है इसमें. ये पॉल लिख करना शुरू कर दिया है, ठीक है. Now come to the tail flick method. We will use analgesiometer, ठीक है. In that there is a strainer where we keep the mice or rat, ठीक है. और प्रॉक्सिमल जो है हम लोग टेल का यूज नहीं करते हैं, डिस्टल एंड यूज करते हैं. प्रॉक्सिमल एंड जो है ज़्यादा कैरेटेनाइज रहता है तो हीट रेसिस्टेंट रहता है, और डिस्टल जो है थोड़ा हीट सेंसिटिव रहेगा, रिस्पॉन्स अच्छा आएगा. तो इसलिए हम लोग स्ट्रेनर में जो है उसको रैट को या माइस को प्लेस करके उसको फिक्स कर दें और फिर इसका रिस्पॉन्स हम लोग देखते हैं. विल डेमोन्स्ट्रेट देयर ऑल्सो कैसे ये करेगा काम. So the placing the tip of last to 1-2 centimeter of the tail on the radiant heat source, that is heating filament, just like हीटर में जो फिलामेंट रहता है वैसा ही रहेगा, उस पर हम लोग प्लेस करेंगे. एंड टेल विड्रॉल फ्रॉम हीट, रिएक्शन टाइम जो फ्लिकरिंग जो है हम लोग देखेंगे कितने सेकेंड में वो रिस्पॉन्स दे रहा है, फ्लिक कर रहा है. देन नॉर्मली विड्रॉ इज टेल टू 3 to 5 सेकेंड जैसा बोले. रिएक्शन टाइम हम लोग 6 सेकेंड से ज़्यादा नहीं रहना चाहिए, 3 to 5 में ही रिस्पॉन्स आ जाता है. एंड अगर इफ एनिमल फेलिंग इन बिटवीन नॉट इट टेक मोर देन 6 सेकेंड, देन वी विल रिजेक्ट दैट एक्सपेरिमेंट. तो एटलीस्ट बेसल रिएक्शन 3 to 5 सेकेंड जो है ये रहता है फॉर 5 मिनट से गैप में जो ईच माइस का जो है हम लोग गैप देते हैं. एक को हम प्लेस किए, फिर उसको जो है 5 मिनट के बाद जो है फिर हम लोग दूसरे को जो है ये करके रिएक्शन हम लोग देखेंगे. सो वेट इज ऑल्सो सेम 18 to 20 ग्राम रहना है. एंड देन वी मार्क इट प्रॉपरली एंड डिवाइड इनटू टेस्ट ग्रुप एंड कंट्रोल ग्रुप एंड प्लेस द माइस इनटू द स्ट्रेनर, जो अभी दिखाएंगे. स्ट्रेनर में डालेंगे और टेल को बाहर जो है प्लेस करते हैं. एंड देन जो टेल का जो डिस्टल एंड है 1 to 2 सेंटीमीटर वो फिलामेंट में रहेगा. एंड देन विल सी द रिस्पॉन्स. ओके, so same will administer orally or subcutaneously drugs and repeat same after 15 minutes, 30 minutes and 60 minutes and 90 minutes. And same cutoff time will be 15 second, and compare the value and calculate the percentage, this much. So here is inside the strainer, tail का जो है इसमें थोड़ा सा प्रॉक्सिमल नहीं रहा है, बट विल कीप द प्रॉक्सिमल इन 1 to 2 सेंटीमीटर, ठीक है. एंड देन सी हाउ वी सेट. फर्स्ट वी टेक द मेजरमेंट के बाद जो है नॉब को इसको सेट करते हैं और फिर टेल को बाहर करते हैं हम लोग. इस तरह से प्लेस कर देंगे फिलामेंट में. सबसे इम्पोर्टेंट है कि इसमें जो है माइस या रैट को जो है आराम से रखना होगा. अगर वो काम नहीं रहेगा तो रिस्पॉन्स हम लोग सही हो, बार बार टेल जो है ये फ्लिक करेगा, तो इसलिए उसको थोड़ा सा काम होने देना है और उसके बाद हम लोग जो है फिर शुरू करेंगे एक्सपेरिमेंट। ऐसा नहीं कि रेस्ट्रेनर में डालें और तुरंत रख दिए, नहीं, यू हैव टू वेट फॉर फ्यू मिनट और फ्यू सेकेंड, ये वो काम हो और रिलैक्स हो जाए, उसके बाद जो है फिर हम लोग रिस्पॉन्स देखेंगे। जैसे अभी आप यहाँ पे देख रहे हैं कि अभी जब रेस्ट्रेनर में हम लोग इंसर्ट किए हैं और जब प्लेस किए हैं तो कुछ सेकंड के लिए जो है, फ्यू सेकंड के लिए वो जो है टेल फ्लिक कर रहा था, तो उसको रिलैक्स होने देंगे। थोड़ा रिलैक्स हो जाएगा उसके बाद ठीक है, अभी रिलैक्स है, रिस्पॉन्स आया, ठीक है। So false positive response may be there due to other drugs like sedatives और muscle relaxant रहेगा, या फिर जो है minimal animal variation in tail flick analgesiometer, हो सकता है कि किसी का जो है response time due to keratinization of tail it will come delay, तो जो है रिस्पॉन्स टाइम डिपेंड करता है वेरिएशन में, कि जो हम लोग ग्रुप में डिवाइड किया उसमें जो टेल का ज़्यादा कैरेटिनाइजेशन ना हो, जैसे कि वो रिस्पॉन्स हो। थैंक यू। अब हम लोग एक बार वहाँ पे डेमोस्ट्रेट करेंगे, आई विल डेमोस्ट्रेट यू देयर। ये दिस वन इज एडिस हॉट प्लेट, 1953 में डिस्कवर किया गया था, एंड दिस जार वी यूज टू कीप इट देयर कि जो है बाहर जंप करके ना आए, ठीक है। ये स्विच ऑन ऑफ स्विच है, इसको हम लोग स्विच ऑन कर देंगे, ठीक है। एंड देन वी हैव टू सेट द टेम्परेचर 50 to 55 डिग्री, आइदर वी विल see the temperature from this also or we can use the thermometer also, ठीक है। तो ये टेम्परेचर सेट करने का ये this button we will press it and we will set the knob to 50 डिग्री और 55 डिग्री, उससे ज़्यादा नहीं और उससे कम नहीं, ठीक है। 51 बनाने देते हैं, ठीक है। अब छोड़ देते हैं और टेम्परेचर जो है राइज होने का वेट करेंगे। तो जब टेम्परेचर राइज होगा देन वी विल प्लेस द माइस इनसाइड द प्लेट। किसी को वीडियो बनाना है तो बना सकते हैं देखने के लिए। प्रीवियस ग्रुप को जो है शो किए हैं इसलिए थोड़ा सा टाइम लगेगा गर्म होने में, क्योंकि बंद कर दिए थे। गर्म हुआ है, हल्का सा हुआ। टेम्परेचर सेट होने के बाद डालते हैं, अभी तो पहले डाला गया है, टेम्परेचर सेट होने में। पॉल किया है अभी हल्का सा, ठीक है। एंड सो दैट वी हैव टू नोट इट डाउन द रिएक्शन टाइम। ज़्यादा गर्म हो जाएगा फिर। Now come to the analgesiometer. ये रेस ट्रेनर है. सबसे पहले तो है कि हमको मेजरमेंट लेना है. ठीक है, वेट ले लिए, मेजरमेंट ले लिए. वी हैव टू जस्ट सेट दिस थिंग. अगर ज़्यादा लंबा है तो इधर कर देंगे, और छोटा है बिकॉज टेल हैज़ टू कम आउटसाइड. सो विल फिक्स इट लाइक दिस. कौन सा डाले थे, ये वाला या ये वाला डाले थे? एक्चुअली सब इधर डिस्ट्रीब्यूटेड है ना, इसलिए दो ही हैं, तो ग्रुप में डिवाइड रहना जरूरी है. कोई बात नहीं, वीडियो में तो देखेंगे. इस तरह से टेल पकड़ के हम लोग जो है इंसर्ट करेंगे. ठीक है, और फिर हम लोग यहाँ पे कैप लगाएं. कहाँ गया? अच्छा, उल्टा हम किए हुए, सॉरी. तो इस तरह से इसको ये करके, ठीक है, कैप लगा दिया. टेल जो है बाहर रहेगा, और फिर हम लोग प्लेस कर देंगे. ये ऑटो कट के लिए, और यहाँ पे फिलामेंट लगा हुआ है. ठीक है, दिस वन इज अ फिलामेंट. तो वंस वी विल स्विच इट ऑन. ठीक है, ये दिस वन इज टू सेट द करंट फ्लो. ठीक है, ज़्यादा नहीं रहना है, 3 to 4 एम्पियर, नॉट मोर देन दैट. ओके, उसके बाद देन वी विल, दिस इज अ सेफ्टी टाइम. ओके, we have to press it. Before that we have to set the timer. 20 second 15 second timer set करेंगे, ये ऑन कर देंगे, and then we will start. तो यहाँ पे जो है फिलामेंट जो है हीट होना शुरू हो गया है. we will see here, इधर दिखाएं. तो जैसे हीट होता रहेगा, बंद होगा, हीट होगा, बंद होगा. बस इन बिटवीन माइस और रैट, any response will come, start flicking tail. So we'll note it down the reaction time, and then we'll calculate the percentage. ठीक है, समझ में आया? क्लियर? ओके, थैंक यू. ठीक है ना? ये तो बता दिए, डाल के दो, डालना दो. अच्छा, डाल के नहीं. एक मिनट, कम प्लीज. कैसे डालते हैं अंदर इस ट्रेनर में, विल शो इट टू यू. इसको इधर शिफ्ट करना हो जाएगा. करके भी दिखाई ही देते हैं. रिस्पॉन्स है, आ गया ना? ठीक है, अब यहाँ पे हम जो है ये ऑन कर दिए और स्टार्ट कर दिए, हटा लिए. अभी फिर से करना होगा. तो इसी तरह से we'll do it as we shown video, same process we'll do it. क्योंकि यहाँ कॉपरेट अभी नहीं गया, बार बार ये रिपीट कर रहे हैं ना सेम, तो थोड़ा सा डिफिकल्ट हो रहा है. तो जो है
करंट,
हाँ, यहाँ ये फिलामेंट है. ये
एनाल्जेसिया की डिग्री को मेजर करेगा.
हाँ, मीन्स विल गिव एनाल्जेसिक ड्रग. ठीक है, तो जो है वंस कंट्रोल में जो है और टेस्ट में डिफरेंस हो है. टेस्ट ड्रग जो है हम लोग दे देते हैं, और कंट्रोल में तो है कि इफ इट रिस्पॉन्स विल कम इन 5 सेकेंड, and after that in test the animal will give analgesic, and then we see the response. Might be it will come after 7 seconds or 8 seconds. That's why we give cut-off time to the 15 seconds. Then we see the response. Then we make the table for control and test, and then we see the response by seeing heating filament heat, and once the mice get heat, then start flicking the tail. See, filament heat हो रहा है. ये ऑटो कट हो जाएगा, जैसे ही वो फ्लिक करेगा ऑटो कट हो जाएगा, तो करंट नहीं होगा, हीट नहीं होगा. Thank you. ठीक है सही था ना समझ आया कुछ है नहीं बहुत आसान है मेन है कि पढ़ना है और देखना है
देख के अपनी पढ़ाई अच्छे से होगी
अब समझ में आया होगा हम लोग भी जब पढ़े थे तो आवाज नहीं लगा रहा होगा हम लोग भी जब पढ़े थे हम लोग देखे थे
Okay. Be seated, please. Sir, thank you, sir. Now I would like to request Professor Dr. Hitesh Mishra, sir, to felicitate Dr. Amritanshu. I request sir to felicitate Dr. Pallavi ma'am.
Thank you, sir.
Thank you, sir. Thank you, everyone.
first aid PG resident, SKMC as well. Okay. So, okay, got it now. So, welcome to IGIMS Patna, Department of Pharmacology, IGIMS Patna. Myself is Dr. Lalit Mohan, Professor at Pharmacology, IGIMS Patna. So, today we can go to demonstration of screening anti-anxiety activity. It is a generally 10 marks question during the PG exam, or we are generally here knowing the behavioral method. We are generally discussing the behavioral method of anxiety of rodents. Rodents means rat and mice. So, any day-to-day work hindrance, it may be more, or it is mainly diagnosed by psychiatrist anxiety, by the DCM, different version of DCM, Diagnostic Statistical Manual, different versions, like 5 maybe. Okay. So, there are different types of behavioral model for anxiolytic agents. So, extraceptive stimuli model, intraceptive stimuli model. Extraceptive means you are not giving any drug, you are not inducing by any, means you are not touching them. Simple to say, you are not touching them, or not introducing any drugs to produce anxiety. Okay, but this is a extraceptive stimuli, means instrument itself causes anxiety, means stimuli originating outside body, stimuli originating outside body. Suppose that is elevated plus maze, means this is elevated from ground level. If you are keeping the animal here in center, due to this elevation itself produces anxiety. Due to open space itself produces anxiety. By giving the drugs, you can reduce the anxiety. By giving the drugs, you can reduce the anxiety. So, you have to remember the some of models, but exam time 1 to 2 models you can describe it, that is enough. We are commonly in our lab, so even caffeine induced anxiety, if more than 10 times, even in humans, if more than 6 times you are consuming the caffeine, coffee simply, you have tachycardia. Okay? So, like the various drugs can produce anxiety, we have the animals models, but in our lab we are generally doing the experiment in 2 models. One is elevated plus maze, another is a bright dark arena. So, this is elevated plus maze that can be dealt with the Mukesh in details, what to do with the just I can demonstrate the dimension of the elevated plus maze. This is 2 open arm, 15 to 10 for rat, 16 to 5 centimeter for mouse, 2 closed arm. So, if remember dimension properly, write in the exam, definitely we will get more marks. So, 2 closed arm, 15 to 10 into 40 centimeter for rats, and 16 to 5 into 12 centimeter for mouse. There is open roof. Roof is a open. Roof is a open. And elevation 50 centimeter for rat. This is elevation from ground, 50 centimeter for rat and 25 centimeter for mouse. So, here we are doing the experiment, we have the animal will be acclimatized. If you are doing the experiment in animal house, there is a dark room. Why dark room? Why we are not doing in the light room experiment?
Because light can cause anxiety to the.
Okay, but every animal they can cause the anxiety. Nothing is too much difference. Rodents are nocturnal animals. It is a proper behavior will we get in night time, but you are not performing experiment in night. What we are doing? We can keep in the dark room and reverse the cycle. Daytime we can keep in dark room. When we are leaving the department, we can on the light. So they can be, we can do like 5 to 6 days like that only. Each and every group like that. So animal can be acclimatized properly because we are performing experiment in the daytime. Otherwise, performing experiment in night time, you can do in the nothing required to change. Okay, but if generally we are performing an experiment in daytime, so we can reverse the cycle. Light dark cycle, we can reverse it. And before any year, once if you can use the animal, you cannot use. So suppose they can use the animal for control group. You cannot use same animal for test group or a standard group because animal learns. Means one animal exposed for only one time in the same instrument. Okay? दूसरे इंस्ट्रूमेंट में कर सकते हो लेकिन सेम इंस्ट्रूमेंट में एटलीस्ट वन टाइम because they can learn the things, so there is a problem in the results. So generally we can clean the because there is sometimes defecation and all these things, so clean the chambers properly like similar way कि उसमें differences नहीं होना चाहिए cleaning में. So I told laboratory should be dim light and noise free, animal should be marked properly to avoid mixing, वो तो mark कर देते हैं, 6 animal का group बनाते हैं. The step bathe the animal in mark properly, divide animal into 3 groups, 2 group को अगले बार 3 कर देना जी. Number of 6 in each group, one group is a control, another group is a standard, third one is a test group. You may more group in test. Suppose that 5 milligram per kg, 10 milligram per kg, 15 milligram, like that. So a more in a standard, but definitely 2 group. One is a control, another is a standard. Generally we are using here a standard is alprazolam. That is a 0.05 milligram per kg body weight, like that. Okay. So minimum 18 animals is required. Sometimes we can make the 3 animal groups also if the animals availability is less. So 3 group definitely. One is a control, another is a standard, another is a test group. Test group have maybe different one maybe more. Okay, for different dose. So if you are using the control as a using the standard is alprazolam. Alprazolam generally given by the oral route. So alprazolam you have to prepare with the you have the break the alprazolam tablet, mix properly with the gum acacia. So control group of all 6 animals can also expose with the gum acacia and test group with the gum acacia. Sometimes like lorazepam, if you are using the intraperitoneal, test group maybe you have to with the intraperitoneal, like that only. Normal saline, whatever. So generally we can wait for the, we are doing 2 types of experiment. One is acute study, another is a chronic study. In acute study, we can give the drug and wait for 30 minutes. Afterwards, we can perform the experiment. In chronic study, we can give the drug for 10 days. Control group, a standard group and test group. And 10th day, we can perform the experiment. Before we can give the drug and after 30 minutes, we can perform the experiment. That is a chronic study. So we are keeping the animal in here in center of this elevated plus maze. So if I spend more time in open arm, means less anxiety or more anxiety? Less anxiety. Means if I open arm, if more time, less anxiety. If anxiety is more, they are moving towards the closed arm. So number of entries towards the open arm, again less anxiety. So the number of entry towards the open arm, less anxiety, time spent in open arm, less anxiety, time spent in more closed arm, more anxiety. This is somehow this is a brighter, is a fear provoking. The rationale is that open arm is more fear provoking. The instrument itself produces anxiety, so extroceptive. हम लोग एनिमल को कुछ दिए किए नहीं, कोई ड्रग नहीं दिए, कोई टच नहीं किए, इंस्ट्रूमेंट में रख दिए, उसको एंजाइटी होता है, बिकॉज दे आर रिलेटेड फ्रॉम द ग्राउंड लेवल. So हम लोग we can see the first arm preference by putting the center, they are moving the closed arm or open arm, we can note down. Previously we are doing with the stopwatch, they are now with the video recorders. Time spent in open arm, closed arm, and we can see the rears also. This video will show by the Dr. Mukesh. So this is the rears. Vertical movement, this type of called rears. If more rears means less anxiety. More ears means less anxiety. Even in open arm or even closed arm. मान लो स्टैंडर्ड में more ear होना चाहिए compared to the test drug generally. नहीं तो टेस्ट ड्रग ये superior हो जाएगा अल्प्राजुलाम से. So more ears means less anxiety. So we can note down for each animal for 5 minutes. Generally here is a group are more than 3. So we cannot do any S20 test or unpaired T test. So we can do ANOVA. ANOVA for to know the significance among full. And afterward each and every one we can do the compare with each and every one with the post hoc test, generally do not T test. So here you can see alprazolam. Percentage of open arm and total arm entry ratio is a more. Time spent in open arm is in second is a more. Here is a less. Closed arm is less. Okay, so this type of things we are noting down with the control, standard and test drug. Here is my test drug is a NRNXC. That is a polyherbal compound of the Osmum sanctum, means tulsi, and Bithania somnifera, etc. Next is a bright dark enna. Here this is a bright dark enna. Okay. So there are two compartments. One is a bright compartment, another is a dark compartment. That is colored by black color. Bright compartment is colored by white color, illuminated by the white light. Bright dark compartment illuminated by dim light. You can say dim light. You have to see also. So there is a illuminated by red color ka dim light. This is an open tube or wooden box, 45 into 27 into 27 centimeter. Is mainly for it is for rat. Back it is generally done for the rat only. Black chamber 2 into 5th of the box illuminated with a dim red 70 centimeter above the floor. Bright chamber, the two chamber is connected through the doorway. There is a chamber is connected through doorway, and floor is a porous. Okay. Here same thing again we can do the acclimatization of animal, bright and dark cycle, maintain for 12 and 12 hours like that. Generally we are doing reverse way to performing daytime experiment. Laboratory should be dim light, noise free, handle with animal with care, avoid stress and pain. Same thing again we can make the 3 groups, group 1 and 2 and 3 and test drug more group. Wait for 30 minutes after administration. Again 2 types of study we can doing, acute studies and chronic studies. Chronic study for 10 days, more than 10 days. So after day 10 we can performing the studies. Acute studies, same day we are performing the studies. After 30 minutes we can perform the experiment. And chronic study 10 days we can performing the experiment. Here again observe for 5 minutes. Here what is the observation parameter? Number of entry in the bright enna, time spent in rears with except. So time spent in bright and dark enna. If time is spending more in bright enna means less anxiety. Number of rears in bright and dark enna. Again same statistical test we applied here, ANOVA and post hoc donate T test. See here, number of the entries to bright area, time spent in bright area, in second, rears in bright area, rears in dark area. Okay. So today we have also released the book. So after you last, इसके बाद last हो जाएगा. You have to collect the book from the this floor only, right side. Thank you. Now he can demonstrate the video through video how they can place the animal, where it moves like that. Thank you. अभी है.
First we will see how to place the animal. We will place animal facing towards the light at the center of the maze, this way. Now see a close arm entry of the animal. When both hind and forelimb intersect, क्या हो गया? When both forelimb and hind limb enter inside the dark area, then we will count it as one entry, this way. This is a partial exit. So we will not count this number. This is exploring. This is head dipping when animal see from the floor. This is rearing. Exploring open arm. This is rear. Rear is nothing but standing on the hind limb. This is rear. This is rear. And this is rearing in the closed arm. And this is a plus mesh controller, we will see later on. Similarly, how to place animal in the light and dark arena. We will place in the light area at the center of the chamber, and we will give opportunity to enter inside the any chamber. This is entry into the dark arena. And this is entry into the bright arena. Now please come forward and see how to see the plus mesh controller. This is plus mesh controller. We can set timing at from this switch. This is time. We can increase the 14, 15 and by this down. Okay. And we will push the start button when we will place the animal at the center of the maze. Okay. We will hold at the base of the tail. so that it may not hurt him. Placing at the center of the image, facing towards the light. This is dipping, and this is sensor. We will count how much time and how many times it is present in the dark and light. स्टार्ट दबा नहीं था. देखिए ये ओपन आर्म है, ये डार्क वाले का. अभी ओपन में देखिए इंक्रीज कर रहा है 6. ठीक है, टाइमिंग है ये. अगर 13 सेकंड है, 9 सेकंड इसमें चला गया. अब इधर डार्क वाले एरिया में बढ़ रहा है. अब ओपन, ठीक है, अब ओपन में आ गया. यहाँ इंक्रीज कर रहा है. एक्चुअली वो बिल्कुल डार्क एरिया में हम लोग जब करेंगे कैमरा लगा के, और आइडियली वही अच्छा होगा. एक तो काउंटिंग जो है हम लोग वीडियो रिकॉर्डर से कर लेंगे कितनी बार एंट्री हुई है, और टाइमिंग जो है इससे हमारे पास आ जाएगा, और जो अपना एक्सपेरिमेंट है उसके अकॉर्डिंग रिजल्ट ले लेंगे हम. ठीक है, थैंक यू. कोई आप लोग करना चाहेंगे?
खुद ब खुद काउंट कर लेता है कितने देर डार्क कैमरा और कितने देर.
हाँ, टाइम यहाँ से सेंसर से ही काउंट कर ले रहा है, और कितनी बार एंट्री हुआ है तो उसके लिए रिकॉर्डर हम लगा के छोड़ दे रहे हैं. तो हम लोग ये कर टाइमिंग ये देख लेगा. काउंटर का भी इसमें है फंक्शन, लेकिन अगर काउंटर करेंगे तो फिर टाइमिंग हमको खुद देखना पड़ेगा. तो टाइम देखना ज़्यादा ये जरूरी है, तो इसलिए हम लोग इसको यूज़ कर रहे हैं.
ओके, थैंक यू.
आप लोग बैठ जाइए थोड़ी देर.
कितना, how many topic covered today? 7. Even this demonstration. Yeah, this one is 7. On 3 lectures. One lecture is too important. See. How to fill the fonts. And yes, sir. You have to know the institutional animal access committee, who is the member. Yes, and how to fill the fonts. Okay.
Thank you, sir, for this session. Now I request Professor Dr. Hitesh Mishra sir to felicitate Professor Dr. Lalit Mohan sir.
इधर आइए सर, इधर आइए. उधर ही घुमा ना.
Now I request sir to please felicitate Dr. Mukesh Kumar sir.
शर्ट का कलर मैच कर रहा है.
Now I request sir to felicitate Dr. Syed.
पूरा नाम बोलो.
Syed Sarjil Anis sir.
ठीक.
Thank you everyone. Now you can move.
Move for next.
हम लोगों ने जाता तो जाते बैठ दो पर इधर रख के वहां ऐसे निकाल दो. तुमने सारे सेशन के लिए बाकी
बुलाओ इससे.
उसके अलावा
हाइटिंग है ना.
जी सर.
from?
PG student.
PG student?
Life science zoology.
Very good. From Patna University.
PWC.
Hmm?
PWC.
Patna Women's College.
So my name is Martina. I am from Patna. I have done MBBS from Patna Medical College.
Okay.
I'm from PMC. I'm doing MD Pharmacology from
I am Vishwakuran from BR Medical College.
Sir, I'm Dr. Surya C. I did my MBBS from National Medical College, Kolkata, and now I'm pursuing
So, good evening all of you. In this session, we will see the demonstration of a screening of effect of drug on neuromuscular coordination. So, the aim of this experiment is to study the muscle relaxation property of diazepam, that is muscle grip strength in mice using rotor rod apparatus. So, as we know, the diazepam is basically a benzodiazepine. It has muscle property, and apart from muscle relaxation property, there are some other properties also, like anti-anxiety property, anti-convulsant property, and sedative property also. And sometimes we use in withdrawal syndrome and delirium tremens in case of chronic alcoholics. Okay. So, for the commencement of any experiment, we have to need some basic requirements. That is, in this experiment we need animal, that is mice, and having weight 20 to 25 grams of each mice. We will divide the mice into two groups, and each group contains 6 mice, and each mice is labeled to so that it cannot mix with the experimental group with the control group. So, like red colored is done for control group, and blue colored is done for experimental group. Now the syringe or needle we will use of 20 gauge, and basically the syringe most probably we use is tuberculin syringe or insulin syringe, and the drug solution is diazepam, and the instrument is rotor rod apparatus. Apart from rotor rod apparatus, and another apparatus is inclined plane. We will discuss about here regarding rotor rod apparatus. So, this one is mice, and this one is diazepam. It is marketed as tablet, in the form of tablet form, syrup form, as well as injectable form. We will use here in injectable form. This one is rotor rod apparatus. In this apparatus there are so many parts like fall sensor, latency to fall timer. This is a speed adjuster, and this is a spacer disc, and this rotating belt, drive belt. And what is the principle of this experiment? So basically the diazepam is a benzodiazepine, exert anxiolytic, sedative, muscle relaxant and anticonvulsant and amnestic effect, as we talked about previously. And the skeletal muscle relaxant together with priming or calming effect, these agents reduce anxiety as well as tension. So the loss of muscle grip is an indication of muscle relaxation, and the effect of that is loss of muscle grip can be easily studied in animal using either inclined plane method or rotating rod method. So in the rotating rod method, the mice is this is of control group that is already acclimatized, is a step for 3 to 5 minutes on the rotating pulley, that is rotating rod. So for any before doing any experiment, we have to acclimatize first the experimental animal. Before acclimatization we cannot do the experiment. जैसे अगर इस पे अगर अचानक से अगर एनिमल हाउस से इस पे रखा जाए तो हो सकता है कि जंप कर जाए या फिर आपके हाथ में बाइट भी कर ले, तो उसके लिए एक्लिमेटाइजेशन जरूरी है, लाइक इन प्रॉपर डीम लाइट इन्वायरमेंट, नॉइज फ्री इन्वायरमेंट, एंजाइटी फ्री इन्वायरमेंट, so that we can do the experiment easily. And the difference in the fall of time from the rotating rod before and after diazepam treatment is taken as index of muscle relaxation. So what are the precautions before the experimentation? As we talked that animal should be appropriately labeled to prevent mixing the group, that is control group and experimental or study group, and treat the animal with kindness, that is holding is delicately so that it cannot become irritable, or the mice will become anxiety free. So the procedure, weight the mice and count the mice in 6 in each groups, and put on the rotor rod apparatus each by each and see the retaining time on the rotating rod. Note down the fall of time when the mouse fall from the rotating rod. A normal mouse generally falls off within 3 to 5 minutes, and when the injection is given, that is diazepam, then we see the fall of time is decreased in compare to control group. So before giving the diazepam injection, we have to know about the drug dose titration. So in the market the dose of the in the market the diazepam is marketed as in the concentration of 1 ml contains 5 mg. So the dosing in mice is 4 mg per kg, means to say 100 gram requires 4 mg, and this is for that animal, this is highly concentrated because only 1 ml contains 5 mg, and the dose required only 4 mg for 1000 gram. So we have to eat diluted first with normal saline. So The dilution method will be discussed here by Dr. Rajiv sir. See later. So, after 30 minutes, the giving of the diazepam injection, we will repeat the experimentation and see the retaining time of the rotating pulley and compare the fall of time before and after giving the diazepam. This is the representation, weight and number of the mice, and we are going to see in this experiment later. So, weight and number of the mice, turn on the rotor rod and select an appropriate speed. Starting from 5 RPM and goes up to 20 to 25 RPM is ideal. Place the animal one by one on the rotating rod and note down the fall of time when the mouse fall from the rotating rod. A normal, that is control group, that is untreated mouse, generally stay for 3 to 5 minutes, and after injection of diazepam, after proper dilution, when given intraperitoneally, and wait for 30 minutes and repeat the experiment, that is put on the rotating pulley and see the fall of time. मतलब कितना देर तक स्टे करता है उस पर, कितने देर पे उस पर फॉलो अप कर जाता है. ठीक है. In this slide we will see the before treatment, the staying time is 30 seconds, and after treatment it is stay only for 9 seconds. This one is observation. In this table, the weight of the mice, the diazepam treatment and the before drug and the after drug, the fall of time is given, and the we conclude the percentage time change in time. So, percentage, what is the percentage change in time, how we will calculate? So, before giving the drugs, the time taken to retain on the pulley minus after giving the drug, the time retained on the pulley, divided by before giving the drug, the time staying on the rotating pulley into 100, that is percentage change in time. Yes, this one is percentage change in time, that is fall of time before treatment minus fall of time after treatment by fall of time before treatment into 100. This one is graphical representation. Before the drug, the average staying time is about 30 seconds, and after drug treatment is about 9 seconds. So, the drug dilution is very, very important. So, Dr. Neeraj sir, please say about the drug dilution. पहले डाइल्यूशन बता दें, उसके बाद इधर आ जाए, यहाँ दिखेगा.
So, dilution is the drug dosing is the most important. So, how to dose it? So, concentration. So, strength of the diazepam is given 5 mg per ml. So, very much concentration. So, how can we dosing? So, suppose a average 25 gram of mice I has taken. So, dose of mice 4 mg per kg body weight given. So, we have calculated 0.1 milligram diazepam required for 25 milligram, 25 gram of mice, and volume needed 0.02 ml. So, how to measure? So, this is the challenging. So, pipette is required. So, for the precision of dosing. So this is the most important thing. So how to solve this problem? So we have to dilute. Suppose if we dilute the 10 times of the dilution, so strength become 0.5 milligram per ml. For 25 milligram, 25 gram of mice, volume required is 0.2 ml. So answer will be possible with the help of the insulin syringe. 1 ml insulin syringe 40 unit. So 0.2 ml 8 unit. We can solve this problem. If 100 times dilution, if we dilute 100 times dilution, so for 25 gram of mice, 2 ml of volume needed, and these 2 ml of volume is the max value for the intraperitoneal route. So this is the limiting factor another. So to these problems solve, so we have to use either insulin syringe and another alternative tuberculin syringe. 40 units in insulin syringe 1 ml, but tuberculin syringe is graded with the up to the 100 Mars and 10 every 10 0.ml. So we have to calculate this. So problem can be solved by this method. If 1000 times dilution, so volume 20 ml. So another is the big, here limiting is the 20 ml. So this is the most important. The dosing part is the very important, and we have to choose intraperitoneal route. Why? Intraperitoneal route has highly absorptive surface area, and tablet is also available there, but absorption is the another thing, variation, so many variations, every mice to every mice variations. So first pass metabolism, all those things regarding. So this is the another things. So highly absorptive surface area provided the good level of the drugs to work on the central nervous system to see the effect for the muscle relaxant property. So finally we have chosen the two groups, group A and group B. In group A there are 6 mice, another group B 6 mice, and in every mice at least 3 readings should be taken, and the mean value chosen, and in every group, like group A, in group A before and post experiment, before A जैसे कंट्रोल चुना, कंट्रोल में मैंने क्या दिया, नॉर्मल सलाइन दिया, नॉर्मल सलाइन भी उसको भी किया, पहले नॉर्मल किया, फिर पुट देने के बाद एनएस किया, उसके बाद कंपैरेटिव स्टडी किया, तो उसमें भी हम निकाल लेंगे, है ना, यानी पेयर टी टेस्ट. उसी तरह ग्रुप बी में भी करेंगे, है ना, डोज दे करके, डाइजिपाम दे करके, तो उसी तरह से निकल जाएगा. तो अनपेयर टी, फिर जब ग्रुप ए और ग्रुप बी में करेंगे तो क्या होगा, अनपेयर टी टेस्ट, है ना, अनपेयर टी टेस्ट निकाल लेंगे. तो इससे हमारा ड्रग सिग्निफिकेंट है कि नहीं ग्रुप ए एंड वर्सेस ग्रुप बी में, जो कि हमको पता चल जाएगा. पी वैल्यू है लेस देन 0.05, तो सिग्निफिकेंट हमको मिलेगा आंसर. हाँ, एक साथ 6 चैम्बर बना हुआ है, तो एक साथ क्या कर सकते हैं, जैसे अल्प्राजोलाम भी दे दिए, डाइजिपाम भी दे दिए, लोराजेपाम भी दे दिए. तो अब हम देखना चाहते हैं लोराजेपाम वर्सेस डाइजिपाम, वहां भी हम कर सकते हैं. और सभी ड्रग्स के बीच में हम इफेक्ट साइज को मेजर भी कर सकते हैं, है ना. अरे ठीक है भाई, सिग्निफिकेंट तो है, लेकिन कितना सिग्निफिकेंट है, तो दिस इज द इफेक्ट साइज. आंसर इज इफेक्ट साइज. तो इफेक्ट साइज मेजर, तब हम कंक्लूड कर सकते हैं कि नहीं, ये मेरा ये ड्रग जो है ज्यादा सिग्निफिकेंट है, अच्छा रिजल्ट दे रहा है, इसका गुड मसल रिलैक्सेंट प्रॉपर्टीज है, ये एस्टेब्लिश हो सकता है. ठीक है. So this is the main motto of the experiment, है ना, to see the muscle relaxant properties. और अनदर थिंग, इसको हम क्या कर सकते हैं, हम डिजीज मॉडल्स भी बना सकते हैं इसका, है ना। जैसे चूहा में माइस में हम लोग सिलेक्ट किया तो माइस का हम लोग डिजीज मॉडल भी तैयार से, जैसे पार्किंसन मॉडल बनाते हैं, उसमें भी ग्रिप स्ट्रेंथ को देखते हैं, है ना। फॉल ऑफ टाइम को नोट करते हैं, जैसे परसेंटेज ऑफ फॉल ऑफ टाइम निकाले कि भाई ये डिजीज मॉडल बनाए तो इसका कितना असर है, इसमें स्ट्रेंथ है कि नहीं है ना। जो टॉक्सिन्स जितना चुना है, जैसे एमपीटीपी है ना पार्किंसन के लिए, तो देखते हैं उसका कितना फॉल हो रहा है कि नहीं हो रहा है। ये चीज देखने को मिलेगा हमको, मसल स्ट्रेंथ, ठीक है। तो ये चीज हो गया। तो कैलकुलेशन इज द, डोजिंग इज द मोस्ट इंपॉर्टेंट थिंग। रेस्ट पार्ट
Now, please come here and see the demonstration. This is on-off button of this system. Just on the button and put the mice on the rotating rack.
आप लोग ग्रुप में है ना, है ना। आप एक काम कीजिएगा, ये फोटो आप ले लीजिएगा आप लोग, ठीक है ना, और शेयर मार दीजिएगा सबको। कैलकुलेशन वाला पार्ट है, ठीक है। ये नंबर, हाँ, ये तीनों का आप लोग आपस में शेयर कर लीजिएगा, ठीक है, ताकि इजी हो। किसी को चाहिए तो अपना अपना कर लेगा, ठीक है।
Initial speed is 5 rpm. Then we have to increase up to 20 to 25 rpm. And note the time, what is the staying time. And remember, this one is control group, not given any drug.
ये माइस लिए हम लोग रैड भी ले सकते हैं, ठीक है। और इसका लेंथ जो है 75 सेंटीमीटर है, और ये हाइट जो है 50 सेंटीमीटर है, ठीक है। और इसका डायमीटर जो होगा रैट के लिए वो ज़्यादा होगा, 60 mm, और इसका 30 mm लिया हुआ है। हाँ रखिए, लीजिए। ऑलवेज टेल से हैंडल करना, टेल से इसको पिक करना है, और जिस तरह से मोशन हो रहा है जिस अकॉर्डिंग में, क्लॉकवाइज तो क्लॉकवाइज उसी डायरेक्शन में पुट कर देना है उसको, ठीक है। हाँ पकड़ी पकड़ी, टेल से पकड़ो, टेल। चलो रखा, पकड़ सकते हैं, लीजिए। अब देखिए जैसे गिरेगा वहाँ स्टॉपवाच रुक जाएगा उसका।
ड्रग देने के बाद और ये दोनों को कंपेयर करेंगे। एनेक्सियस है, थोड़ा सा एंजाइटी में आ गया, ये चलते थोड़ा टाइम।
स्पीड बढ़ा रहा है, स्पीड
हाँ अभी 12 से ऊपर
बढ़ा 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 बढ़ा।
अगर फुल्ली एक्लिमेटाइज नहीं रहेगा तो जंप कर जाएगा। साइज वगैरह रेट और इसका बता दीजिए।
साइज तो हम लोगों ने ये वेट तो ले लिया इसका, कितना 20 to 25 है ना। और रेट में हम लोग ले लेंगे वही 150 से 250 के बीच बोलता है, तो 200 से 250 एवरेज ले लिया जाता है। 180 100 200 के आसपास लेता है, हाँ। और माइस के लिए 30 और रेट के लिए 60, ये डायमीटर, ठीक है। और हाइट फिक्स है 50 सेंटीमीटर, ठीक है।
आइडियल है 3 to 5 मिनट।
स्टार्ट करेंगे और धीरे धीरे बढ़ाते जाएंगे अप टू कितना 25 तक रेवोल्यूशन पर मिनट, है ना। तो धीरे धीरे क्या होगा, उसका मसल्स जितना एनर्जी पावर है डाउन चलते जाएगा, नहीं तो फॉल हो जाएगा उसका। और ड्रग देंगे तो क्या हो जाएगा, क्योंकि मसल रिलैक्सान प्रॉपर्टी है तो पहले गिर जाएगा वो, है ना। तो उसी का परसेंटेज रेशियो हम लोगों ने निकाला था। और हम लोग कट ऑफ टाइम लिए थे, कितना लिए थे, 3 to 5 मिनट तक लिए थे। 3 3 मिनट लेते हैं जनरली, ठीक है। तो उसके अंदर वाला चूहा और ऊपर वाला नहीं लेना है, 3 to ऑनवर्ड नहीं लेना है, ठीक है। अपना है स्ट्रेंथ है, 3 to 5 मिनट तक है ना। अभी कौन सा ड्रग दिए उसको?
तो पहले गिर जाता
तब
चलिए आइडियल पहुंच गया, 20 से 25 किलो रखते हैं और ऑलमोस्ट 3 मिनट्स होने जा रहा है इसका। 3 to 5 आइडियल है, इसका मतलब अभी करीब फुल्ली एग्नोटाइज है, नॉट इन एक्सेस। 35 तक करके देखते हैं।
लेकिन अगर क्रॉस कर जाएगा तो नहीं लेंगे इस चूहा को, हटा देंगे। हाँ एक्चुअली क्या है, इसके कैलिब्रेशन में भी प्रॉब्लम है, ठीक है। न्यू वर्जन वाला थोड़ा सा ये पुराना हो चुका है ना थोड़ा सा, तो ये भी एक थोड़ा सा ये है, अपडेट नहीं हो सकता है, कैलिब्रेशन प्रॉब्लम हो। बढ़ाया गया कुछ नहीं गिर रहा क्या पिलाया अरे कुछ दिया है क्या इसको? कौन चीज स्टिमुलेंट दिया है तुम उसको? मैक्स वैल्यू है, नहीं उतने तो
नहीं बढ़ रहा है,
ये तो उतने मैक्स वैल्यू है।
5 मिनट्स हो गया, अभी नहीं बढ़ रहा।
5 को चला गया वो, वाह
30 सेकंड और है।
जैसे ये हो गया टेस्ट, कंट्रोल ग्रुप, इसी में कंपेयर करेंगे हम लोग एक्सपेरिमेंटल ग्रुप से। जैसे 2 इसमें और डिवीजन मान लें 3-4 डिवीजन का होगा, तो एक कंट्रोल ग्रुप का इसमें डाइजेपाम हो सकता है, एक में लोराजेपाम हो सकता है, एक में मिडाजोलाम हो सकता है, अल्प्राजोलाम हो सकता है। तो हम लोग ड्रग के बीच का कंपेयर कर सकते हैं, कंट्रोल और डिफरेंट ड्रग का कर सकते हैं, ठीक है ना।
हाँ इसको डिसाइड करेंगे, नहीं लेंगे इस आपको। हाँ तो एक बार में तुमने किए इसको, 3 बार 4 बार 5 बार कर करके पहले से 15 दिन से पहले से कराएंगे इसको, है ना। और जिस दिन एक्सपेरिमेंट परफॉर्म करेंगे कम से कम 3 रीडिंग लेंगे।
बात इम्पोर्टेंट है,
हाँ 3 रीडिंग लेंगे, ठीक है।
जो मोस्ट इम्पोर्टेंट है वो ड्रग डोज डाइल्यूशन टाइट्रेशन है, मतलब कितना कंसंट्रेटेड अमाउंट को हम लोग किस तरह से डाइल्यूट करें उसको देने लायक सब है। सेकेंड बात ये है कि हम लोग इस मेथड से सेम ग्रुप के डिफरेंट ड्रग का कंपैरेटिव हम लोग मसल रिलेक्शन प्रॉपर्टी दे सकते हैं, साथ ही कंट्रोल ग्रुप से डिफरेंट ड्रग का कंपैरेटिव हम लोग दे सकते हैं, ठीक है।
चलिए ठीक है, समझ में आया ना? हाँ ठीक, गुड।
हाँ जस्ट फिनिश किए थे लोग, स्टार्ट है जी, कैमरा बंद नहीं किया, नहीं वो पॉज किया इधर लाइट
Thank you, sir, for this session. Now I request Professor Dr. Manish sir to felicitate Dr. Punendu Aras. Uday sir. I request sir to felicitate Dr. Rajiv Kumar Neeraj sir. Thank you everyone. Now you can move forward for high tea and snacks. And if you want to take the book that we have launched today, you can take from there.
पोस्टर प्रेजेंटेशन
अभी चल रहा है साथ में।
मम्मी बंद कर दीजिए इसको।